everyone, I hope you can see me and hear me fine. A very good morning to all of you. If you have joined, please come and say hi in the chat box. Hi Shubhajit, how are you? Good morning. Annie, we are not live. Okay, that's all right, Shubhajit. But welcome to this session of INICET, right? So we are going to have the marathon. INICET is just around the corner, so just last minute revision of the most important topics, high yield topics. And the way I've done is, the way I've brought this marathon for you is so that we can integrate all of the knowledge. So we are going to read a lot of the theory and we are going to integrate them in the form of tables so that we can quickly recap everything that we have read. Okay, so this is not a detailed class on individual topics, but this is going to be a marathon on very important theory topics. Hi, good morning everybody. Welcome to the session. So without wasting much time, let's begin because we have long 3 hours ahead of us, right? So when we talk about the phases of bacterial growth curve, you all know that there is a lag phase, then there is a log phase, and after that there is a stationary phase, and then there is a dip that is called as the death or the decline phase, right? So what is this one in the blue? This is the log phase. Log phase, right? I hope you all remember. So what all happens in the lag, log and the stationary phase? So in the lag phase, there is no cell division. There is no cell division. So only thing that happens in this phase is that the bacteria, they eat and they grow up in size. Because when we put them into a culture medium and we have given them lots and lots to eat, they are simply eating and growing big in size. So if they ask you at which phase the cells are the maximum in the size, then obviously at the end of the lag phase, the cells have attained the maximum size, right? This is where the cells have attained the maximum size. What happens in the log phase? I always tell you log phase is equal to jog phase. Log is equal to jog. So this is the youth of the bacteria. Bacteria is quite young right now, right? So what happens when you're young, when you're in your youth? So first of all, why does this log happen? Because the bacteria is multiplying very fast. So what is happening? There is exponential multiplication, exponential cell division or exponential multiplication of the bacteria. Right? And what happens when you're young? You're very active, you're very mingling, you talk to people, you interact a lot. Good morning guys. So what happens then? You interact a lot. So the bacteria is going to be extremely biochemically active, extremely metabolically active, right? So best time to do biochemical reactions. The question will be asked like this. Best time to do biochemical reactions would be log phase or the jog phase or the young phase. It's the most metabolically active. When you're young, you like to do a lot of makeup, you like to wear jazzy clothes and all, right? So the bacteria is going to take up all of the stain. So this is the best time to perform gram stain. This is the best time to perform gram staining in the bacteria. Right? And this is the time when you want like shiny cars. So what is a shiny car? The shiny capsule. So this is the time that the bacteria produces the capsule. So we confuse it a lot between stationary and log phase. So when is the capsule produced? The log phase. When is the spore produced? S for stationary, S for spore. So I always tell you, remember stationary phase as the war zone. This is war zone now. Okay, so what is actually happening? Just imagine there is a test tube in which there is some culture media. You have put 10 bacteria there. Initially in the lag phase, all the 10 bacteria is eating and becoming big fat. Then finally the log phase comes, jog phase comes. They're in their youth, they're in their teenage, they're active, they're interacting, they're taking up the gram stain, they're producing shiny capsules, they're doing biochemical reactions, metabolically active. Now they are multiplying, okay, they are reproducing, there is a lot of exponential division. Now all the nutrition is lost, nutrition is depleted. Good morning guys. All the nutrition is now depleted, yes? So what is going to happen now? Whenever there is lack of nutrition, lack of resources, lack of oil in the world, what happens? War. So in the stationary zone, the war is happening. And always I tell you that in the war, there are two types of people in a war. One who hide. So either the bacteria is going to hide. So how will it hide? Inside bunkers. So it will hide under a blanket. What is that blanket? Spore formation. So in which phase does sporulation happen? S for stationary, S for sporulation phase. Then when they hide, they are going to store all the nutrition. They will pack big boxes of food. What are those big boxes of food? They are the storage granules. So if they ask you a question, Corynebacterium diphtheria produces the volutin granules in which phase? So this is very easy. The volutin granules of Corynebacterium diphtheria are produced in the stationary phase of bacterial growth curve. Right? Then there are the other type of people who are fighters. So the fighter bacteria, what will they do? They will attack. They will attack. How does the bacteria attack? By producing toxins. So in which phase are the endotoxin or the exotoxin both produced? Endo or exo, both the toxins are produced by bacteria in the stationary phase when they want to kill the others. In which phase is the antibiotics produced? Antibiotics is also produced by bacteria to kill other bacteria. Yes, correct, Shobhajit. Stationary phase. Yes, in the stationary phase, the antibiotics are also produced. Now, at this time, if I give you some makeup, will you care about makeup? You will not care about makeup, right? So, if I give the bacteria gram stain, they will not care. They are going to be non-uniformly gram stained. So, the bacteria is going to be non-uniformly gram stained here, right? Non-uniformly gram stained. Very, very easy for me to remember all the points. Then comes the death phase or the decline phase. What happens after wall? You have the mutilated bodies. So the mutilated bodies in bacteria, they look like the involution forms. Involution forms are the abnormally shaped, aberrant shaped forms of bacteria, right? So in which phase is the maximum viable count, guys? Come on, now you answer this in the chat. I'm reading the chat. You answer this. Maximum viable count, that is maximum live bacteria are present in which phase? Come on, chat box. In which phase is maximum viable count? Live bacteria. Alive bacteria is maximum when? Okay, and when is the maximum total count? Total count means body count. Total count, dead plus live. Total body count. Yes. End of log phase. End of log phase is when you're going to have maximum viable count. Over here you have maximum live bacteria. Because in the stationary phase, why is it stationary? Because the number of cell divisions, that is the number of new cell divisions, is equal to the number of deaths. The number of deaths. So equal number of people are dying as well as multiplying. So the total population remains the same. But what happens to the total body count? It keeps on increasing. So when is the total count? Yes, correct, Shubhajit. Total count is at the end of stationary phase. End of stationary phase. Yes, so obviously all the second year knowledge is going to help you crack INI-CET and NEET PG as well. Now I have a favorite question which I always ask all my students, and this is probably the 5th or 6th live class that I'm asking the same question, that generation time is maximum when? Please understand that the generation time is independent of the growth curve. So generation time is constant throughout. It is independent of the phases of the growth curve. Right? So that finishes the bacterial growth curve very quickly and the potential possible questions that can be asked. What is happening here? Immunology. Let's dive into immunology, right? What is happening here? You all know that there is a T cell. A T cell has a T cell receptor. The T cell receptor is made up of two parts, the alpha unit and the beta unit. And just like everybody else, they also have variable regions and constant regions. So if you look at this blue part, can you focus over here on this blue part? If I divide it into two, then the upper part can be called as the constant region of the beta region, and the upper part over here can be called as the constant alpha region, and the lower parts can be called as the variable beta region, and this can be called as the variable alpha region. So this is a T cell receptor. Yes. How does a T cell see the antigen? There always has to be an antigen presenting cell which keeps the antigen on its bicep. What is the bicep? The major histocompatibility complex, the MHC. The MHC is going to present the antigen to the T cell receptor, and that is how the activation happens. What happens whenever there is a super antigen? A super antigen is a super irritating guy. This guy is going to, yes, V beta region. This guy is going to stimulate the T cell receptor from the side. It's going to poke the T cell receptor from the side over here. What is it going to stimulate? It is going to non-specifically stimulate the V beta region of the T cell receptor. As a result of which, what is going to be there? There is going to be a massive stimulation of the T cell leading to hypergammaglobulinemia. Hypergammaglobulinemia. I hope there is no problem in you writing this. And there is going to be a cytokine storm, right? So they might give you an image and they might ask you this. What else can they do? They can ask you the examples. So your super bacteria, super sir Staph aureus, right? Staph aureus is a supreme guy. Why? Because all the toxins produced by Staph aureus are super antigens. So the toxic shock syndrome toxin 1 of Staph aureus is an example of a super antigen. Then the scalded syndrome, that is the exfoliative toxin of Staph aureus is an example of your super antigen. Then what else? The other toxin, the enterotoxin for the food poisoning produced by Staph aureus is an example of your super antigen. Yes, malassezia from yesterday. So we are coming on to it. Wait. So the second guy amongst bacteria is your Streptococcus pyogenes. Streptococcus pyogenes, which is group A Streptococcus. Group A Streptococcus. What does it produce? It produces the streptococcal pyrogenic exotoxin A and C. A and C. They are responsible for causing scarlet fever, toxic shock syndrome. That is also a super antigen. Another bacteria, tell me another bacteria which can produce. You know, mycoplasma, the agent of community acquired pneumonia, the agent of atypical pneumonia, that can also produce super antigen. Another bacteria, tell me, Yersinia, but mind you, not the plague causing one, but the ones which are responsible for causing GI diseases. So Yersinia pseudotuberculosis, Yersinia enterocolitica, they are the ones which are responsible for producing super antigens. What else can produce? Fungus. Which fungus? Yes, correctly said by Shubhajit in the chat box. Malassezia furfur. Malassezia furfur. We are going to talk about Malassezia furfur today as well. And number 6, you have some viruses. Which viruses? Cytomegalovirus, Epstein-Barr virus, few components of the HIV virus. They can all act as super antigens. Right? Is it clear? Safety pin kahan se aaya? Okay, Yersinia safety pin. Excellent demonstration of knowledge. Good job, guys. Right? Then we move ahead. Then we look at the next thing, that let's look at the antibody. So let's finish a little bit of immunity right now. Can you see this antibody? A single Y-shaped molecule. This is the fabulous fab portion which binds my antigen. And this is the constant region which is responsible for the different classes of antibodies, right? So how do I remember the classes? I remember GAMDE. Why do I remember GAMDE? Because this is the concentration. So which is the maximum concentration? This is IgG is present in the maximum concentration inside my body. So whenever I see a antibody which is a monomer, a single unit, right, a single unit, so this could either be an IgG, or this could be an IgE, or this could be IgD, right? The only possibilities are these three. If it is an IgG, what are all the features? The features is that it is going to be present in maximum concentration. Right? This is what is responsible for indicating past infection. What else? This activates the classical complement pathway. Right? Classical complement pathway. And what else does it do? It crosses the placenta. So these are all important points about IgG that we have to remember. What about IgE? I always tell you, E is equal to U. U means allergy and parasites. So this is the one which is responsible for causing allergies and type 1 hypersensitivity. This is the main antibody produced. We will talk about it. This is the one which rises in the parasitic, especially the helminthic infections. Okay, it rises in the parasitic or especially the helminthic infections. EBV-CMV, correct. Okay, right. Then E, if you touch something hot, you will say E. So IgE is actually going to be heat labile out of all the antibodies, right? Then what about IgD? We only know one importance of IgG. You know the B cells? The B cells have these B cell receptors on their surface. You know what are these B cell receptors? These B cell receptors are nothing but IgD or IgM. So this is the only thing we know, importance of IgD, that we know, right? Now suppose they give you this pentamer. Suppose they give you this pentamer in the diagram. So this here is the J chain, which is called as the joining chain. So I can see that this guy over here is a pentamer. So what am I dealing with? I'm obviously dealing with IgN. So So what are the features of IgM antibody? IgM is the first antibody to rise. So this is a marker of acute infection. This is also present as a B cell receptor. So this is your B cell marker as well, just like your IgD antibody. What else? This is a pentamer. So obviously this is going to be the heaviest. This is going to be the heaviest antibody out there. Anything else which is important? It is joined by the J chain. Similarly, this joint at the hip, with the FC portion, the J chain present and the secretory protein also present. This in fact is a PYQ of INICET. They asked this in 2025. They simply gave this image and they said which type of antibody this is, right? So this secretory protein has been borrowed from the host because this makes this antibody a secretory antibody. Secretory antibody, and this is the only antibody which undergoes mucosal transport. Right? It undergoes what? It undergoes mucosal transport. Right? So what is this? This is IgA. IgA is always present as a dimer. Right? And this is A for alternate. So this is the one which activates the alternate pathway of complement. Alternate pathway of complement is activated by the IgA antibody. Right? Excellent. Done. Now, what is this? This is an update. This is an update, guys. Yes, okay, IgM, correct. What is the update? Nobel Prize in 2025. So they might ask you, they might not ask you, but since this is an update, so we have to predict this. So what can they ask? 2025 Nobel Prize in Physiology and Medicine. Nobody can remember these names. Shimon Sakaguchi, Mary Brunkow, and Fred Ramsell. Just remember, 3 people got the Nobel Prize. What did they get it for? For identifying that the T regulatory cells, which are controlled by the FOXP3 gene, they are the defining problem of autoimmunity. They are the pace keepers to prevent autoimmunity, right? Master switch control. That means they said that the peripheral immune tolerance. So you look for these words in the question, whether they are saying that the peripheral immune tolerance is defined by the T regulatory genes and the FOXP3 gene is responsible for regulating these T cells. So this brings me what all concepts we had read about peripheral immune tolerance. If they want to ask you a question on peripheral immune tolerance, chat box is open. Come on, get the answers flowing. What were the methodologies of peripheral immune tolerance? In fact, let's do one thing. Let's enumerate both. What were the factors for central immune tolerance? The factors for central immune tolerance, it was either happening in the thymus or in the bone marrow. So what was happening to the T cells in the thymus? If at all a T cell makes the receptor against a self antigen, that T cell had to die. That T cell had to die. What is that called as? Clonal deletion. Clonal deletion. So if they ask you a question, clonal deletion is a method of central tolerance, right? What happens to the B cells in the bone marrow? In the bone marrow, B cells undergo B cell receptor editing. So they undergo receptor editing. So if they ask you, receptor editing and clonal deletion are methods of central immune tolerance. Correct? Then if they ask you, what are the methods of peripheral immune tolerance? Peripheral immune tolerance. Right? That is what they might ask you. So you know the T regulatory genes, just told by all of these guys, the T regulatory cells, they go about patrolling and killing any any T cells which have produced receptors against autoantibodies, right? They do that. Secondly, the T regulatory cells, then you have some sequestered antigens. Sequestration of certain antigens such as in the brain, in the testes, in the cornea, that also protects against autoantibodies. Then what was there? There was the energy, T cell energy. What happens? There is no co-stimulatory receptor binding. What is prevented there? We don't allow. We prevent the B7 and which ligand binding? Come on guys, chat box. Nobody is answering. We prevent the B7 and the CD, which receptor binding is prevented? Nobody remembers peripheral tolerance. B7CD28 receptor binding is prevented over here. So the co-stimulation of the T cell does not happen, which is anergy. Then even if anergy fails, even if this receptor binding happens, there is phenotypic skewing. Phenotypic skewing, where the cytokines which are produced after T cell activation, they are of less pathogenic. So you have the low pathogenicity cytokines which are produced after this. Yes, then you have another way of peripheral tolerance. What's that? You have anergy, you have phenotypic skewing, you have sequestration of antigens, you have T regulatory cells, and what else happens? Yeah, more or less. Am I missing anything? If I'm missing anything, maybe I can think of it later. So these are the major events which happen. T cell anergy. So if T cell anergy fails, then there is going to be a problem. If the phenotypic skewing fails, then there is going to be a problem. If the sequestration, if there is any trauma, the antigens come out, autoimmunity can start. If the T regulatory cells they fail, then autoimmunity can also start, right? So these are the potential questions because of this one update. Or of course there is a direct question, which is the FOXP3 gene that they can ask you getting activated and protecting us against autoimmunity. So this was an update of 2025. So that's why they can ask you. Right, guys. What else? Now let's talk about hypersensitivity a little bit. So I want to test your knowledge first. The way I have written: serum sickness, contact dermatitis, SLE, erythroblastosis fetalis, bee venom, and farmer's lung. What are these all examples of the hypersensitivity reactions? You tell me in the chat box. I am waiting. Come on. And then we will discuss all the individual types of hypersensitivity. Come on, come on. Tell me. Serum sickness is an example of what? Nobody is writing. Hypersensitivity reaction 3, correct. Okay, we forgot to add one thing over here. AICD, or activation induced cell death, very important, which is mediated by the FAS ligand. FAS ligand upregulation leads to activation induced cell death of the T cell. So the T cell dies. So now we have the list is completed. Alright, okay, let's go back. Serum sickness type 3, okay. What contact dermatitis you are saying is type 4 hypersensitivity, correct. What about SLE? SLE, come on, what happens in SLE? You have the red rashes. Yes, it is type 3, correct. Erythroblastosis fetalis, what do you think this is? Erythroblastosis fetalis, it happens because of RH incompatibility. So it has something to do with cross reaction of the RBCs. I'm getting answers, very nice. So this is type, come on guys. Nobody? SLE is type 3, correct. What about erythroblastosis fetalis? What about bee venom? So bee venom, you will have immediate reaction, right? Wheal production. So that is type 1. No problem there. Farmer's lung. EB2. Erythroblastosis is 2. Type 2. Excellent. What about farmer's lung? Bee venom is simple. What about farmer's lung? Farmer's lung? 2. Correct. What about farmer's lung, guys? Farmer's lung, there's going to be some granuloma production around. Bee venom is type 1. So type 4, farmer's lung is type 4 because of the chronic nature. Farmer's lung is type 4, beta. Right. Steven-Johnson syndrome, type 4. Good pasture syndrome, type 2. These are all just names. Lepra reaction 2. So one of my students itself gave me the beautiful mnemonic to remember lepra reaction. He said that, ma'am, the sum total has to come to be 5. The sum total has to come to 5. So if it is lepra reaction 1, then the hypersensitivity has to be 4, because then the total will become 5. Yes. If it is lepra reaction 2, then the hypersensitivity has to be 3, because then the total is going to be 5. So my student itself gave me this beautiful mnemonic. So I have learned from my students as well. So lepra reaction 2 is going to be hypersensitivity 3. No problem there. And if it was written lepra reaction 1, it would have been hypersensitivity 4. Great. Cassoni's test. Who is going to tell me this? Yes, Shubhaji. 2 plus 3 and 1 plus 4. Cassoni's test. Come on, guys. Cassoni's test. Who will tell me Cassoni's test? What is Cassoni's test? What is it performed for? You remember hydrated disease. In that we are injecting the hydrated fluid into the person and we are looking for allergic response. So this is type 1. So not every skin test is a type 4 test. There is Monte is similar to Montux, and Montux we do know it is for TB and it is the type 4. So Montenegro is Kala Azar, and this becomes your type 4 hypersensitivity reaction. This is a delayed type hypersensitivity skin reaction. So not every injected skin reaction is your type 4. So what is the antigen injected? Leishmanian antigen is injected in this. Okay, quick revision. Very nice. Now, hypersensitive 1. Quickly, we will do. Yes, what will come? An allergen. An irritating allergen will come in hypersensitivity 1. Who will see this allergen? Your T cell. T cell is going to see this allergen. Imagine T helper 2 or whatever. What is T helper going to release? T helper, okay, APC. I'm sorry. Your APC is going to see this. APC is going to present this on its bicep to what? To the T cell, right? T cell has seen it. T cell gets activated. Now this T cell converts into a delayed type hypersensitivity T cell. Why? Because this T cell is sensitized. It has now produced these receptors against this allergen. So I call it the sensitization phase. Sensitization phase is complete. Now, sorry. Guys, wait. Let's do it again. Yeah. So the allergen has been shown. What will the allergen do? It will be interacted and taken up by the T helper. What is T helper going to produce? It is going to produce interleukin 4. What is interleukin 4 going to do? It is going to activate the B cell. B cell is going to produce plasma cell. What will plasma cell produce? Ultimately, there is production of IgE antibodies. Right? These IgE antibodies. And I know from the Fab portion they bind to the allergen. But who do they bind to from the FC portion? From the FC portion, they bind to the mast cells. So over here, they will bind to a big mast cell. And this mast cell is a mast cell. He has many granules inside, jinki wajah se it can burst. Yes, correct, Shubhajit. 1, 2, ka 4. That is a mnemonic. I will come on to that mnemonic very soon. So this is the mast cell which is there. Right? This mast cell is now sensitized. Now the sensitization phase is complete. This is called as sensitization of mast cell. This sensitized mast cell has produced many receptors for the FC portion of these IgE antibodies. And now these IgE antibodies are sitting there bound to the FC portion of these cells, right? See, they're sitting here bound to the FC portion. Now they're waiting. They're waiting for this idiotic allergen to come again. This allergen will come again and it will come and bind to 2 simultaneous IgEs together. So when it will bind to 2 IgEs together, it is like a head butting. So it has picked one IgE, another IgE, and it is butting their heads together. What is going to happen? The mast cell is going to lose his mind and the mast cell is going to degranulate. So what is the second phase which happens, which is the actual phase? This is the degranulation of the mast cell which actually happens. Degranulation of the mast cell. What does degranulation do? It produces prostaglandins, it produces leukotrienes, it produces histamines. And what do these histamines, leukotrienes, prostaglandins do? They bring about the allergic response, the edema, the smooth muscle contraction, etc. Yes. So when does the degranulation happen? It happens on re-exposure. So first there is exposure. Yes. there is exposure, there is sensitization, then there is re-exposure, and then there is degranulation of the mast cell. Correct? How do we remember which helper cell, which interleukin? So our mnemonic, like my students will remember, 1, 2, ka, 4. 1, 2 ka 4, 4, 2 ka 1, my name is Lakhan, right? Very bad singer, please don't mind me. So 1, 2 ka 4. So if it is it is hypersensitivity reaction type 1. So which is the T helper which is going to be there? T helper 2. So which is the interleukin which is going to get produced? Interleukin 4. Easy, easy. Which are the main antibodies responsible? IgE. How do I remember type 1, 2, 3, 4? It is very easy to remember. Okay, let's try to remember. If I say hypersensitivity reaction 1, 2, 3 and 4. Can I also call them as A, B, C and D? Yes. So A is allergy. So I know that hypersensitivity 1 is allergy. The main guy which are going to act is IgE. What is the final step that happens is mast cell degranulation is the final step that happens over here. Right? Thank you. Thank you. Final step that happens is mast cell degranulation. Right? And it happens immediately. So one looks like I. So this happens immediately. This is an immediate reaction. Done? Done. What about type 2? B. Can I write it as antibody? So this is the one which is antibody mediated. Can I also write it as cytotoxic? Cytotoxic. So what is actually happening? Antibody mediated cytotoxicity. So what is going to be the main cellular events here? Antibody dependent cellular cytotoxicity. What is going to happen here? Mainly IgM or IgG mediated killing. How do these IgM, IgG kill? They kill by calling the complement. So complement mediated killing and ADCC are going to be the mainstay of killing by type 2. Then we have type 3. The 3 rhymes with free and C can be written as complex. Right? So what is happening in type 3? There are free antigen antibody complexes which are formed in the type 3 hypersensitivity reaction. They go and they settle in different sites of the body. What happens in D? D is delayed type hypersensitivity. It takes many days, 2 to 3 days. Actually 2 to 3 days. Right? Delayed type hypersensitivity. This is the only one which is an example of cell mediated immunity. All of these three are actually your examples of humoral immunity. Humoral immunity. Alright? Excellent. These are examples of humoral immunity. This is an example of cell mediated immunity. With this overall understanding, let's go ahead. Let's look at type 2 hypersensitivity. Shall we look at type 2 hypersensitivity? How is ADCC happening? This here is an allergen. Allergen is bound to some target cell, some RBC, some cell of my own body. This will be identified by whom? This will be identified by my B cell. B cell is going to activate what? It's going to activate plasma cell, which is going to produce these antibodies. And you know, these antibodies can either activate the complement mediated killing, or these antibodies from the FC portion, they can bind the natural killer cells, NK cells. And what will these NK cells do? They will lead to antibody dependent cellular cytotoxicity. You know this, very easy. What happens in type 3? The complexes, they go and settle in different parts of the body. So if they settle in my basement membrane of the kidneys, they can lead to glomerulonephritis. Glomerulonephritis becomes an example. Then SLE also becomes an example, where then they settle in the skin. If they settle in the joints, then reactive arthritis, they all become your example. Serum sickness, they all become your example. Antibody dependent cellular cytotoxicity. Excellent. Right? Then what happens in type 4? Let's go back to the mnemonic. 1, 2, ka 4. So if the hypersensitivity reaction is 4, then my T helper 1 is going to produce interleukin 2 over here. So how do I reach T helper 1 first? Of course, I am going to reach with the help of antigen presenting cell. So suppose this antigen has been shown by the antigen presenting cell to whom? on the MHC to the T cell. Now, which T cell is going to come? T helper 1. T helper 1 is going to release interleukin 2, and we know interleukin 2 does what? It activates the cytotoxic T cells. Cytotoxic T cells are going to kill this guy, and they activate the interferon gamma, etc. is also produced. Interleukin 2 plus interferon gamma plus tissue necrosis factor. They're all going to call upon the macrophages. They're going to call upon the natural killer cells. So they're all going to create a what? This is the allergen, and they're going to create this granuloma around this allergen. What is this? Granuloma formation. So this T helper cell here, he got sensitized first. So this is the sensitization phase. Right? Because this T helper converts into a delayed type T helper cell. And then again, when this allergen comes and attacks on this delayed type hypersensitivity cell, this produces a lot of interleukin 2. This interleukin 2 further activates all of these cells over here, and ultimately there is granuloma formation. So what is the ultimate step of type 4 hypersensitivity? It is granuloma formation. So your chronic diseases like tuberculosis, your skin tests such as your Montenegro's test, your Montague's test, they are all examples of type 4 hypersensitivity. Alright, spent lots and lots of time on hypersensitivity. Now we do what? Now let's talk about biosafety levels. Shall we talk about biosafety levels? So you have BSL 1, 2, 3 and 4. This is in the increasing risk of the microbes. So biosafety level 1 means simple benchtop working. I am working on a benchtop, so I am dealing with simple commensals such as Saccharomyces yeast. Yes, simple day-to-day bacteria such as your E. coli, such as your Klebsiella, Staph aureus, the day-to-day bacteria. Then what happens in BSL 2? In BSL 2, I need to at least wear a surgical mask, and I need to go into a biosafety cabinet. I cannot do it out in the open. So your pathogenic guys such as hepatitis, yes, such as salmonella, such as Shigella, which have a low infective dose and which can lead to serious diseases. BSL 3, now I am talking about N95 masks. Now I am talking about HEPA filtered air. Right? HEPA filters. So now come your respiratory guys. So always remember BSL 3 is all the respiratory pathogens. And they like to ask you BSL 3 for this reason, because this is the only category of classification which can be classified properly. So BSL 3 agents include your SARS-CoV-2. So you needed a proper room at the end of the laboratory, you needed HEPA filters, you need biosafety cabinets. Then your Mycobacterium tuberculosis. This is a previous year question, hai na? They asked which category agent is Mycobacterium tuberculosis. This is a PYQ. So respiratory hai, so BSL 3 ही होना पड़ेगा उसको. If it is respiratory, it is going to be BSL 3. Then Yersinia pestis, which causes plague. This is also your category 3. BSL 3. What about BSL 4? These are the rare guys, right? Ebola. Smallpox. Imagine if smallpox comes back. It is very, very dangerous. If we are talking in India, yellow fever. These are all very, very important BSL 4 agents. Okay. Then, what about bioterrorism agents? So let's cover two types of agents together. BSL, which was biosafety level. Now I'm talking about bioterrorism. Bioterrorism is category A, B and C. What is category A? Maximum priority. These are highly dangerous, high mortality, and they can disseminate very, very easily. So they need a lot of special attention. Right? Then we have category B, which are moderate, and category C, they are easily produced, but they are high morbidity and there is high morbidity and mortality associated with them as well. Are we ready? So what is the mnemonic for category A agents? The mnemonic for category A agents? ABP small tv. Yes, ABP small tv. So ABP news, These days news is what? Very, very loud, very, very like that. So for the category A agents, we have to be very hyper. So we have the ABP. What is A? Anthrax. Anthrax. Remember, the spores of the anthrax were used as bioterrorism agents. Botulism. Botulism, because it's a neurotoxin. Then plague. Plague is a category A agent, right? Smallpox. Smallpox. TB, T for tularemia, tularemia, and V, viral hemorrhagic fevers. So what will come in viral hemorrhagic fever? Ebola, Lassa, they will fall into your viral hemorrhagic fever category. Okay, excellent. What about category B? See, category B is BQP food. BQP food. So all food and water safety related. Food and water safety related. So salmonella, which is typhoid, Vibrio cholerae, all of these fall under this category. Vibrio cholerae, enterohemorrhagic E. coli, salmonella, they all fall here. Then B for brucella. Then you have Q fever, which is Coxiella, Coxiella. And P is what? Psittacosis, Psittacosis. This is category B. What about category C? All the new ones. Hantavirus, Nipah virus and Mycobacterium tuberculosis. This is a question which was asked. This is a PYQ. Especially your drug-resistant TB. Drug-resistant TB. Emerging. So C is emerging pathogens. This is what is important here. C, category C is actually emerging pathogens, the new ones which are coming up and we are not prepared for them. So new drug resistance is coming up in TB. So even though TB is an old disease, but new drug resistance is coming, so we are not prepared. Nipah virus new, hantavirus new, and when SARS-CoV-2 came into the world, it was also considered as a category C bioterrorism agent, right? So the new guys. So let's test our knowledge. Tularemia, guys, come on. What is the agent? Anybody? Yes, spore sent as envelope. Very, very nice. Tularemia is a ABP small TB category A agent. No problem there. But what is the agent? What is the agent? Francisella tularensis. And my students will remember the mnemonic. We had looked at a rabbit, Francis the rabbit. So we know that tularemia is zoonotic through the rabbits. Then glanders. Glanders is the disease caused by, it's in horses, the disease of horses by Burkholderia malayi. Burkholderia malayi. What is the agent? B. B. So every B is category B. Then cholera, food safety becomes food safety, Vibrio cholerae, right? So food safety, water safety. Where is water safety and food safety going to come? Category B. Okay, coronavirus emerging, especially the SARS-CoV-2 category C. MDR-TB category C. Yes, yellow fever, tell me. Yellow fever, the yellow fever virus, which is a flaviviridae, which is a flavivirus responsible for hemorrhagic hepatitis. Especially in India, it is not there. So if it comes, it becomes which agent? Which category agent? New virus. Category C. Right? For India, it becomes category C because it will be like a new disease. Anthrax. We have just seen. Anthrax is by Bacillus anthracis. Bacillus anthracis. Right? So category A. Yes. Ebola. Ebola virus. It is going to be category VHF. Yes, it is an agent of VHF. So it is going to be category A. Plague, ABP, small TB, Yersinia pestis. Even though it is a bacteria, it becomes category A. Smallpox. Who is the guy? Variola major. है ना? Variola major. Category क्या बन जाएगी? ABP small TB, smallpox category A. O157H7, enterohemorrhagic E. coli. Again we are talking food safety. So this becomes category B. Okay. So easily you can mark if you are faced with any such question. Shall we discuss vaccines? There is a recent update of vaccine. So of course very soon we are going to talk about dengue vaccine. Also in the fourth slide we are going to talk about dengue vaccines. Before that, let's look at our existing knowledge about vaccines. Come on, let's do this. Yes. What is our mnemonic? Excellent mnemonic, my students will tell. Japanese chick on a Bombay train. Japanese chick on a Bombay train. So, what are all the agents? Japanese encephalitis is a live vaccine. What is the strain used? SA14142 strain. Yes, chicken pox. What is the strain of vaccine used in this live vaccine? Oka strain. Yes, on a Bombay train. Bombay. Let's write Bombay. Okay, I'll write Y later. So B. B is what? BCG. Bacillus calmette-guerin for tuberculosis. O is what? OPV. What is the strain of OPV? Sabin strain. Then I have, yes, Japanese chick on a Bombay train. Excellent, Shobhaji. Then we have M. M is MMR. Actually, it is MMR. So we have measles. So measles I will write alag se. Measles, है ना? Just shift this. Measles I will write separately. Then we have mumps and we have rubella. Mumps का कौन सा strain होता है? Mumps. Gerald Lynn went up the hill, got mumps and came down. So we have the Gerald Lynn strain. Excellent. What about rubella? RA, not 24/7, 27/3. RA 27/3. What about measles? Measles is very special. There are 3 strains of measles. So how do we write? We write like this. Yes, we write measles like this. So we have the Moratan strain. We have the Edmonston Zagreb strain. And we have the Schwartz strain. Yes, the 3 strains of measles. Then Y. Y is yellow fever. Then we have yellow fever. Yellow fever vaccine is a live vaccine. What is the strain used? 17D strain. Right, guys? 17D strain. Thank you. 17D strain. Now we are going to use this 17D somewhere else also. When we talk about the dengue vaccine, there we are going to again talk about the 17D strain. Then train. T for typhoral. So not every typhoid vaccine is a live attenuated vaccine, but typhoral is a live attenuated vaccine. Who will tell me the strain of typhoral? TY21A. TY21A is the strain. R for rotavirus. Rotavirus, which is Rotatec. What is the strain? 116E strain. 116E strain. Then we have influenza. Influenza, right? You have the live attenuated influenza vaccines. The live vaccines, right? Which are used as nasal sprays for influenza. Alright, guys. Yes, so this was live vaccines. Then we have the toxoid vaccines. Very, very easy. You have the diphtheria toxoid, you have the tetanus toxoid, and you have the pertussis toxoid vaccine. So the DPT will become examples of toxoid vaccine. What about subunit vaccine? Can you tell me about subunit vaccine? The acellular pertussis. Acellular pertussis becomes subunit. What else becomes subunit? Hepatitis B surface antigen. The Hep B vaccine becomes subunit vaccine. Then coronavirus spike protein. Yes. Coronavirus is also spike protein. That is also an example of subunit vaccine. Now let's come to the actual topic which we were waiting for. The reason why we started talking about vaccines. The updates on dengue vaccine. Yes, so we know that dengue is present in the serotypes DEN 1, DEN 2, DEN 3 and DEN 4. These are the 4 subtypes, serotypes of the dengue vaccine. The existing vaccine is Dengvaxia. Dengvaxia. This is also a live vaccine. This also has the 4 serotypes. This also covers the 4 serotypes. But what is the backbone of this? Okay, right. What is the backbone of this? The backbone is the yellow fever vaccine. Yes, backbone is the yellow fever virus. Sorry guys. Yellow fever virus is the backbone of this guy. Right. Then Qdenga, recently released Qdenga, approved by India, still not given the schedule of vaccination. This is also live vaccine. This also has 4 serotypes. But what is the backbone? Backbone is a DEN 2. DEN 2 is the backbone. And on top of that, we have added proteins of DEN 1, 3 and 4. Right. What this is also 2 doses. This is also 2 doses. What is mandatory here? Mandatory is previous history of disease must be present. So this can only be given in previously diseased individuals. Here, no history of disease needed. Okay, so we do not need to elicit seropositivity. So over here we can say for Dengvaxia can only be given. So who are the candidates for Dengvaxia? Candidates are seropositive individuals. Seropositive people. Over here seropositivity is not needed. Seropositivity not needed. Then we have Dengue-Al. Dengue-Al. What about Dengue-Al? Dengue-Al is new. It is being made in India by ICMR. And it is currently in the phase 3 trials. What is special about it? This is also live, but this is 1 dose, single dose vaccine. This is a single dose vaccine. Alright guys, this is a single dose. Thank you so much Mazhar for your compliments. Yes, so now we have 3 important dengue vaccines. Dengvaxia, Qdenga, Dengue-Al. Right? Done. This was the update that I wanted to give you. Now let's talk a little bit about sterilization and disinfection. Yes, so if I quickly let's recap. What is sterilization? It is only sterilization when it can kill the spores. When it can kill the spores. Excellent. Now you tell me. There are the physical methods of sterilization and there are the chemical methods of sterilization. If I am saying physical methods, they can be heat-based methods or they can be non-heat based methods. So when I am saying heat-based methods, it can be dry heat. So whenever there is sterilization and heat, it has to be more than 100 degrees Celsius. Or it can be moist heat. Moist heat. So if you tell me examples of dry heat, hot air oven, flaming and incineration. Incineration. They are your dry heat. What about moist heat? The over romanticized pressure cooker, over glorified pressure cooker, autoclave. Autoclave. Yes, 10 raised to power 6 log reduction in the organic load of microbes. That is also sterilization. But they are going to ask you this: kill the spores, whether they kill the spores or not. Okay, excellent. Now, what about the What about the non-heat based methods? Non-heat based physical methods. So you have filtration, which can include what? Which can include your HEPA filters, ALPA filters and membrane filters. What is the pore size of membrane filters? 0.22 micrometers. Yes, 0.22 micrometers. Then there is radiation. And amongst radiation, you have the ionizing radiation. Ionizing radiation. So which ones? X-ray and gamma ray. Gamma ray. What do you sterilize with it? Catgut sutures, transplant tissues. Indicators, biological indicators, writing right here. What is the biological indicator for hot air oven or dry heat? Bacillus atrophius. Bacillus atrophius. What is the indicator for moist heat or autoclave? Geobacillus stereothermophilus. Yes, stereothermophilus. Excellent. What is the biological indicator for filtration? Bacillus subtilis. Bacillus subtilis. What is it for radiation? It is Serratia. Okay, sorry, ulta kar diya. What is it for radiation? Radiation is Bacillus subtilis. है ना. और फिल्ट्रेशन में तो स्पोर देखने की जरूरत नहीं है. दिस इज नॉट किलिंग, दिस इज सिंपली मैकेनिकली फिल्टरिंग आउट. सो वी कैन यूज Serratia marcescens और वी कैन यूज Brevidomonas diminuta. Brevidomonas diminuta. ओके, ये कोई पूछता नहीं है वैसे. ठीक है, दिस इज द फिजिकल मेथड्स. देन फॉर स्टेरिलाइजेशन वी कैन ऑल्सो हैव द केमिकल मेथड्स. Chemical methods. So you can have the ETO gas, which is gaseous chemical sterilant, or you can have the, what else can you have other than ETO gas? Plasma sterilizer. Plasma sterilizer, plasma is the fourth state of matter. Fourth state of matter. So what do we use as a for ETO gas? What is our biological indicator? Same as dry heat. So over here you have Bacillus atrophius. And what is the indicator for plasma sterilizer? Same as autoclave. Geobacillus stereothermophilus. What percentage of H2O2 do we use in plasma sterilizer? We use 6% hydrogen peroxide. Vapors. Yes, excellent. ETO and plasma sterilizer. अच्छा, अब देखते हैं जरा Spalding's classification. He says there can be critical items. Critical means always sterilization only. Sterilization only. There can be semi-critical. If it is semi-critical, then it can be sterilization or high-level disinfection. Or they can be non-critical. If they are non-critical items, then intermediate level disinfection or low level disinfection can work. So let's talk, come on. Correct, 6%. Laryngoscope blades. Are they critical, non-critical? What? Laryngoscope blades. They are made up of metal. They can cause trauma. Don't you think they should be considered as critical? Come on. Chat box, come on. Critical, non-critical, semi-critical, what are they? Nobody is writing. Critical? Yes. Sterilization method, obviously autoclave or ETO. What is the principle? How does they kill actually? It is the DNA destruction. Destruction of DNA is going to be the principle of all your sterilization methods. Oximeter. What about oximeter? बाहर ही है, नॉन क्रिटिकल है ना, सिंपली टचिंग द स्किन ऑफ द पेशेंट, नॉन क्रिटिकल. लो लेवल डिसइंफेक्शन. व्हाट कैन आई यूज? आई कैन यूज इथेनॉल. हाउ डू लो लेवल डिसइंफेक्टेंट्स किल? दे डिस्ट्रक्ट द lipid destruction. Yes, implants go well inside. They have to be critical. What is going to happen? Autoclave or ETO. We know the mechanism of action. Cardiac catheters going straight into the heart. Critical. Same. Okay. Cardiac catheters cannot be autoclaved. So you will do either ETO or plasma. What about flexible endoscopes? Flexible endoscopes are considered semi-critical. Because they are flexible, they only enter the mucous membrane. Yes, semi-critical only enter the mucous membrane. So a flexible endoscope is only entering there and it's not causing any trauma as such. So this is semi-critical. What can we use for this? We can use a simple high-level disinfectant. Let's talk about a high-level disinfectant. Glutaraldehyde. Yes, glutaraldehyde. What? Also called as Sidex. Sidex. How much? 2 to 2.4%. Or paracetic acid or hydrogen peroxide. Bedpans, they are also semi-critical. They will also be high-level disinfectant. Another example of high-level disinfectant which can be used, orthophthaldehyde. So there is a hyphen here. Hyphen means half a word. So what is the concentration? 0.5%. 0.5%. What else can be used? Paracetic acid. P for paracetic acid, P for parch. So P for point. This is 0.1%. And H2O2. 2 plus 2 is 4. 4% H2O2. All of these become your high level disinfectants. Yes. Then periodontal scalars. Don't you think this is a critical item? This is a critical item and this needs to be autoclaved or ETO. Right? Correct. So done. Done. Next what? What are we looking at next? Just a minute. Yeah. Match the following. Now you guys match the following. Come on. Formaldehyde, yes, correctly said, but formaldehyde is not used because of the carcinogenic fumes, toxic fumes produced by it. Now you tell me, match the following. Cheese, honey, seafood, eggs, poultry. We're talking about food poisoning now very, very quickly. Quickly, we're talking about food poisoning. Cheese. What should I match cheese with? So I am saying that this is Clostridium botulinum. Yeah. Clostridium botulinum. Poultry is 5. Okay, you're matching from below. You're matching from below. Poultry. Chalo, let me say poultry or raw milk. Now you tell me. Seafood is easy. Vibrios, the halophilic ones. The halophilic vibrios such as Vibrio parahemolyticus. Yeah. So seafood is 1. What about honey? Honey. Botulism. Yes. Canned food. So honey becomes 5. No problem. Eggs. Eggs. Reptile eggs. Salmonella. The non-typhoidal salmonella. Eggs become 3. Excellent. Now what about poultry raw milk? Remember Campylobacter? We were camping with cattle and little chicks and all. So poultry and raw milk becomes Campylobacter. Now only thing remaining, cheese is associated with listeria. Cheese and listeria. This is what I wanted you to know here. Okay. X33. Who will tell me about reheated fried rice syndrome? Yes. Who will tell me about reheated fried rice syndrome? Bolo. Caused by? Yes. Bacillus cereus. Bacillus cereus. Which toxin is produced? The emetic type toxin. Emetic type toxin. And this is a type of a preformed toxin. Preformed toxin. Who else produces the preformed toxin? Staph aureus. Staph aureus produces the preformed toxin. So if I am talking about milk or pastries or cream, then also Staph aureus They are both preformed toxins and they are both going to be 1 to 3 hours is going to be the incubation period for them. 1 to 3 hours is going to be the incubation period. Yes. Now, Bacillus cereus also produces the diarrhea type toxin. Also produces the diarrhea type toxin. Yes, that can lead to food poisoning in 18 to 16 hours incubation period. This is from eating chicken, poultry, etc. Done. Yes. So let's look at a few zoonotic diseases now. I said cows or milk. What all can happen whenever you have in the history you have the buzzword of cows or milk? Yes. Come on, you also keep writing. Brucella. Yes, Brucella abortus. Brucella. What about same as ETEC heat lab? Heat labile 1. Okay. Excellent. Then Campylobacter we have just seen. Campylobacter. Okay. Who was our cow? Bruce Anthers. Remember? Hi, my name is Bruce Anthers. So you had Brucella. You had anthrax. Anthrax. Especially which anthrax? Cutaneous anthrax. Cutaneous anthrax. What is it called as? Hyde-Porter's disease. Hyde-Porter's disease. Yes, anthrax. Then my name is Bruce Anthers cow. Cow for Coxiella. Coxiella. So Q fever. Q fever. Done. Hi, my name is Bruce Anthers. Remember our cow? Yes, done. Then we have poultry. What about poultry? Same as the above. Campylobacter, Coxiella, Brucella, then also non-typhoidal salmonella, especially from the eggs. So poultry and reptiles, they can transmit non-typhoidal salmonella. Then you remember another source of food poisoning from eating chicken 16 to 18 hours, Clostridium perfringens. So Clostridium perfringens doesn't only cause gas gangrene, it also causes an enterotoxin, necrotizing enterocolitis also can happen. No problem. So Clostridium perfringens, food poisoning can be because of eating poultry. What about pigs? What do the pigs transmit? First of all, the moment you say pig, you should remember pork tapeworm. Yes, you should remember pork tapeworm. Clostridium perfringens, Campylobacter. Okay, great, excellent. Pork tapeworm. So you have Taenia solium. Taenia solium. What will eating pork tapeworm cause? It will cause intestinal teniasis, not neurocysticercosis. Intestinal teniasis. Okay, so eating pork does not transmit neurocysticercosis, not neurocysticercosis. What else from pigs? They are the amplifier host of Japanese encephalitis. So not disease transmission, but they are the amplifier host of Japanese encephalitis. What else are they? They are the mixing vessels. Mixing vessels. In whom? In which disease do they act as a mixing vessel? Influenza, correct. Influenza. H1N1, etc. In influenza, they act as mixing vessels. What about cats? What do cats transmit? Very, very important, two diseases. One is, of course, toxoplasmosis. Toxoplasma. So we always remember that cat with the red eyes and the spotted head, and it was a baby cat. So that is toxoplasmosis. And the other disease which is called as the cat scratch disease. Cat scratch disease transmitted by Bartonella. हमने हंसल मेहता की बिल्ली देखी थी ना. So Bartonella henselae. Bartonella henselae. हंसल मेहता की बिल्ली, right? Henselae the cat. Bartonella henselae, cat scratch disease. Done? Done. What else? What? Who's the next animal? Dogs. Dogs भी कोई बीमारी करते हैं. They're so cute. Of course they cause diseases. कौन सी? Dog tapeworm, Echinococcus granulosus. Dog tapeworm. What is the name of the disease, guys? Chat box. Come on. Hydatid cyst. Yes. Hydatid cyst. Awesome. Rats. Rats. RRR. R disease. Rat urine, rainwater and rice fields. So of course, leptospirosis. Leptospira interrogans, also called as Weil's disease. Weil's disease. Yes. What else? Rat bite fever. Rat bite fever caused by spirillum. Spirillum minus. Okay, done. What else? Birds. What do birds spread? We have just seen the category B agent. The category B agent. Psittacosis. Psittacosis. Through parrots. P for parrots. Then bird droppings, nitrogenous soil. Remember, histoplasmosis. Histoplasmosis. We will be revising histoplasmosis. Then the rats of the sky, pigeons, they transmit cryptococcosis. Cryptococcosis. Made a reel on it also. Rats of the sky, pigeons. Yes. Reptiles. Very easy now. Reptiles, I have said it so many times. Non-typhoidal salmonella. Fish. Fish tapeworm. एक तो क्लोनोर्किस, ओपिस्थॉर्किस. यस, स्पेलिंग को क्या हो गया? And the fish tapeworm. Who is the fish tapeworm? Diphyllobothrium latum. The guy who desperately wants to behave like a trematode with the D for Dhakkan eggs. Yes. Rabbits. What do rabbits transmit? Francis the rabbit. Franciella tulurensis. So we have tularemia. Done. All right. Done. Let's go ahead. Viral inclusion bodies. Let's talk about viral inclusion bodies. Now we have started virology. Are we ready? Can we start? Excellent. Viral inclusion bodies. So they can be the basophilic, so I am writing with blue, intranuclear inclusion bodies. Yes, they can be the eosinophilic, intracytoplasmic inclusion bodies. Yes, intranuclear inclusion bodies, intracytoplasmic inclusion bodies. And they can be the both intranuclear plus intracytoplasmic inclusion bodies, which are the both inclusion bodies. Yes, both is very, very simple. Chief Minister of both the states. Chief Minister of both the states. CM. So you have C and M. So with C you have cytomegalovirus and with M you have the measles virus. Yes, cytomegalovirus से क्या होगा? Owl's eye. Owl's eye. Owl's eye appearance. Measles, Warthin-Finkeldy cells. Warthin-Finkeldy cells. Alright, then you have intranuclear inclusion bodies. Yes, intranuclear. You have two types of intranuclear. Cowdrey type A and Cowdrey type B. Cowdrey type A and Cowdrey type B. Hey Bap. Hey Bap. Right? H for herpes simplex virus, which is Lipschultz bodies. And yellow fever. Yellow fever. Which bodies? Taurus bodies. Excellent. Then adenovirus and poliovirus. B is Cowdrey type B, A is Cowdrey type A. Okay. Now, intracytoplasmic. The most important, rabies, which is the Negri bodies. You can never forget it. Done. Then you have all the pox viruses, all the pox viruses. So you have the smallpox, right? In the smallpox, you have two types of viruses. The variola strain, which is the variola major, or the vaccinia strain, vaccinia virus. So vaccinia virus is the one used for giving vaccine, and variola is the one which causes the disease. So what is the disease? Smallpox. So the bodies are called as Paston bodies. Paston bodies. And vaccine. Vaccine was given in a cow. So this is guarneri. Guarneri bodies. I hope you can see I am not blocking the view. Yeah. Am I blocking? No. Yeah. Guarneri bodies. Done. Right? Paston and Guarneri. Excellent, excellent, excellent. Right. Then we also have fowl pox. Foul pox. So foul कब करते हैं हम? When we play with a ball. So you have the Bollinger bodies. Bollinger bodies of foul pox. I think you can't see. Okay. And then finally we have the molluscum contagiosum virus, which shows the Henderson-Peterson bodies or the HP bodies. Right? Henderson-Peterson bodies or the HP bodies. Done, guys? Foul pox and Bollinger. Done. So let's, okay, before that, let's talk about the symmetry and everything. So let's quickly revise our mnemonics about the viruses. So we have all the DNA viruses. All the DNA viruses are double-stranded DNA viruses, and they are all happy viruses. Happy viruses, double P, right? So you have the herpes viridae. The herpes viridae, which is the human herpes virus. You have hepadna, PADNA, which is hepatitis B. Then you have adeno, parvo, papova means human papilloma virus. Then you have pox and you have phage, which is bacteriophage. These are the happy viruses or double DNA stranded DNA viruses, except parvo. Parvo तो single stranded DNA virus है. Parvo तो smallest virus है. पर वो तो नॉन एनवेलप्ड वायरस है. इनफैक्ट इन तीनों में से ही एपीपी पाप वायरसेस आर योर नॉन एनवेलप्ड वायरसेस. क्या पाप है इनका? They are all non-enveloped viruses. So these are your non-enveloped viruses. When we talk about RNA viruses and we talk about non-enveloped RNA viruses, then we talk about three guys. एक तो वो जिसको सबने कैंसिल कर दिया बिकॉज इट डिड नॉट हैव अ एनवेलप. एक वो जो हमेशा ही रो रहा था, रोता हुआ वायरस. एंड देन फाइनली वी हैव द वन जिसके नाम में ही आरएनए आता है, जिसके नाम में ही आरएनए आता है. सो व्हाट आर द नेम्स? पिकोरना विरिडी, जिसका पार्ट है हेपेटाइटिस ए वायरस. Then cancel culture, calciviridi, जिसका पार्ट है हेपेटाइटिस ई वायरस. Yes, and we have rota hua virus, which is rotavirus. And we always remember why is rotavirus rota hua? Because इसके साथ धोखा हुआ है. It has double stranded RNA and इसके पास envelope नहीं है. So double stranded RNA, that is why it is rota hua virus. Okay, excellent. So let's look here. Herpes, come on, tell me. Herpes. What is the genome? What is the symmetry? What is the envelope? And we know all of these guys, all DNA viruses are going to be icosahedral. Yes. All of these viruses are also going to be icosahedral. No doubt. Okay. Now let's see. Herpes. Herpes belongs to human herpes viridae. It belongs to my happy viruses. Of course, it's a double-stranded DNA. Of course, it is going to be an icosahedral symmetry. Yes. What is it going to have an envelope? Yes, it is a happy virus. It is going to have an envelope. HIV. HIV has 2 copies of single-stranded RNA. The retroviridae, they have 2 copies of single-stranded RNA. Yes. What is the symmetry? Icosahedral, no problem. Does it have an envelope? Yes, sir, it has an envelope. Hepatitis B, hepadnaviridae, it's a happy virus, double-stranded DNA, icosahedral, envelope present. Hepatitis C, it is hepatitis C is a single stranded RNA. All other hepatitis viruses are single stranded RNA viruses. Yes, single stranded RNA. What else? Is it icosahedral? Is it going to have an envelope? Yes, it is going to have an envelope also. Only A and E are non-enveloped. HAV and HEV, they belong to non-enveloped single stranded RNA viruses. Icosahedral symmetry, no envelope. So no envelope. What are the special ones? I am writing in red, and the normal ones I am writing in black. ठीक है ना? Done. Picorna viridi. What about Picorna viridi? Picorna, pico RNA, name itself has RNA. This also belongs to my mnemonic. This is a single stranded RNA virus, icosahedral, no envelope. Yes, influenza. Who will tell me influenza viruses? They belong to the mnemonic Bira. So let's write that mnemonic also over here. Let's make a box and let's write our mnemonic of segmented viruses. And always segmented viruses are only ever going to be my RNA viruses. Segmented viruses are only going to be RNA viruses. So we had Bira, Bunyaviridi, influenza virus, rotavirus, and adenovirus. Arenaviridi. How many segments were there? 3, 8, 11, and 2. 3, 8, 11, and 2. Because 3 plus 8 is 11 and 1 plus 1 is 2. Yes, so these are my viruses. Okay. Then influenza virus becomes, what does the genome become? Segmented RNA. How many segments? 8 segments. Let's look here. ये रहा influenza, 8 segmented RNA virus है ना. So यहाँ पे क्या आएगा? 8 segmented RNA. 8 segmented RNA. But single stranded. 8 segmented single stranded RNA. Symmetry कैसी होती है इसकी? Influenza virus की helical. This is a helical symmetry, and if it is a helical, it has to be enveloped. It becomes enveloped. Then adeno. Adeno kahan dekha? It's a happy virus, but a non-enveloped happy virus. So it is a double-stranded DNA with an icosahedral symmetry. Because it is non-enveloped, has to be icosahedral. So and non-enveloped. ठीक है. What about parvo? Parvo तो single stranded DNA है, single stranded DNA. Parvo तो non-enveloped है, लेकिन सभी की तरह ये भी icosahedral ही है. रोटा वायरस रो रहा है, हमेशा रोता रहता है. क्यों? उसके साथ धोखा हो गया. क्या धोखा? डबल स्ट्रैंडेड RNA. और क्यों रोता रहता है? एनवेलप भी नहीं है उसके पास. सिमेट्री इसकी कैसी होती है? रोटा वायरस सिमेट्री इज icosahedral, no problem. Bacteriophage, complex. They are also happy viruses. तो double stranded DNA virus, happy virus. Envelope से कोई मतलब नहीं है. कैसी है symmetry? Complex symmetry. Done. All viruses we have revised. ये कौन है? पहचानो इसको. आइडेंटिफाई दिस वायरस. क्या दिख रहा है? एक सिंगल स्ट्रैंडेड आरएनए, दूसरा सिंगल स्ट्रैंडेड आरएनए, ऊपर कुछ 3 एंजाइम्स लगे हुए हैं, बाहर मैट्रिक्स है. ये दिख रहा है एक प्रोटीन सबसे बाहर का और एक उससे अंदर का प्रोटीन. कौन से प्रोटीन्स हैं ये? चैट बॉक्स, कीप इट कमिंग. जीपी सबसे बाहर वाला GP120, उसके अंदर वाला GP41, GP120 and GP41. Correct. What about GP120? This is binding. This is the one responsible for binding. What does it bind to? GP41 is responsible for fusion. Am I not getting any answers? Guys, you don't identify HIV virus? Some answers, please. HIV virus है ना. So GP120 binds to what? CXCR5 या CCR4 या CCR5 and CXCR4? चूज करो जरा. CCR4, CXCR5 और CCR5 and CXCR4. क्या चल रहा है? कौन सा वाला है? किससे बाइंड करना है? पिक वन. किसको काटूं? रिवीजन तो चलता ही रहेगा शुभाजीत, यस अभी भी बहुत रिवीजन करना है, चलता ही रहेगा रिवीजन। हाँ जी बोलो। CCR5 एंड CXCR4 कैसे याद रखें, हाउ टू रिमेंबर? चाय सिगरेट 5 रुपए, चाय सिगरेट ₹5, चाय सिगरेट ₹5, ठीक है, दैट इज CCR5। तो दूसरा वाला CXCR4 क्या करेगा, ये मेरा GP120 ये बाइंड करेगा CCR5 एंड CXCR4 को, और GP41 फ्यूज कर जाएगा मेरे सेल के साथ। उसके बाद क्या होगा, दीज पूल ऑफ एंजाइम्स विच आर द पॉल जीन्स, sorry, the pool of enzymes coded by the RIP, rest in peace. So R is for the reverse transcriptase, I is for the integrase, and P is for the protease, pool of enzymes. The Pol gene codes for the pool of enzymes. What is the gene? Pol gene, pool of enzymes. Done? Which of these is responsible for the window period, guys? Window period, this integrase is the one which is responsible for window period because it integrates the viral RNA into my or the person's genome. That is why, right? Pol gene does this. Then there are 3 important genes: Pol gene, GAG gene, and the ENV gene. Right? What was the ENV gene coding for? ENV gene was coding for the GP120 and the GP41, which are the binding units. What is the GAG gene coding for? GAG gene is coding for the matrix antigen, this matrix outside, and it is coding for the non-structural gene, which is the P24. P24 antigen, which we detect in the window period, that is by the GAG gene, coded by the GAG gene. This is the one which we detect in window period. Okay, done. Let's move ahead. Let's quickly look at the replication cycle of the virus. So we have this HIV1 binds to CCR5 or CXCR4, fuses with it, comes inside, reverse transcription starts, and it goes, uncoats inside the nucleus, integrates into my nucleus with the help of the integrase enzyme, transcribes, translates, assembles into proteins, buds out, releases, and goes out after maturation. Very, very simple. What is the important step? The reverse transcriptase step, where the RNA is converted into DNA. Excellent. What is happening over here? Please identify the phenomena. I want you to write in the chat box. Please identify the phenomena. What is happening here? Anybody? Transformation? No. Okay. Let's understand what's happening here. You have this virus. Okay. This virus has got the purple or the pink antigens. Yes. This has got the green antigens. This has got the yellow antigens. We took the viruses, we mixed them up like bhel puri. What do you have now? You have this bhel puri mixed virus here. So many antigens, all antigens have come together. What is this? This was one strain, one strain, one strain. Now a totally new strain of the virus has been produced here. New strain of virus has been produced. What is this called as? It is an assortment of mithai, assortment of bhel puri. This is genetic reassortment. Genetic reassortment. What is genetic reassortment called as? Genetic shift. shift. Shift is sudden. Sudden. Happens suddenly. But something which happens suddenly and a totally new strain is formed cannot happen very, very frequently. So this happens less frequently. Less frequently. So that can happen like once in 10 years. Yes, genetic reassortment leading to pandemics. This leads to pandemics. And what is happening here? One guy with round antigens, quickly there was a mutation. There was a point mutation quickly and suddenly we have these semicircular ones, semicircular ones. Okay, what is this happening here? Genetic drift. Genetic drift. Okay, this is going to be gradual. But this is going to happen more frequently. So this will happen 2 to 3 years. Over here, a new strain is not produced. You see, it is still purple, it is still purple. Purple has only changed to purple, only the shape has changed. So no new strain. No new strain has been produced here. These are all the phenomena of genetic drift and genetic shift or genetic reassortment. This is shown by the very famous influenza virus. Yes, correctly answered. Right, guys? Okay. Chale, let's talk about hepatitis virus a little bit. You tell me, how does the hepatitis virus serology work? You see, this is my virus. Inside this here is the partial double-stranded DNA. Partial double-stranded DNA. It is covered by a icosahedral core and outside that there is an envelope. So envelope is going to have the surface proteins. Okay. And this is the surface protein. So I am calling it the hepatitis B surface antigen. And this green one I am calling as the hepatitis B core antigen. Something which I can contract before the core, these pink ones which are floating about, I call them as the hepatitis B E or the pre-core antigens. Hepatitis B pre-core antigens. Yes. So when the bacteria is flowing inside my blood, this surface antigen is going to be the first antigen to rise. First antigen to rise whenever I have the disease. And surface antigen always means illness. Always means illness. So if you're a person who's simply vaccinated, you might have antibodies against surface antigen, but you will never show the antigen positivity. Whenever there is surface antigen positivity, by rule it means this is a disease. It could be acute disease, chronic disease, carrier disease, anything, but it is disease. Yes, hepatitis B pre-core. What does pre-core do? It is like a leaky antigen. It is the second antigen to rise. The core never comes out. We never leave our core, na? We never leave our identity. We are at a core, we are who we are. We will never let it out. We will never let it go. So that is the core antigen. What does the core antigen do? It never rises. It never rises. It can only be seen in hepatocytes. Only seen in hepatocytes. Do you think my body cannot make antibodies also against it? No, it can, right? Because it is present at the tissue level at least. So my body can make antibodies very much. So which antibodies will rise first and which will rise later? So the first antibody to rise actually is the anti-hepatitis B core antibody. Why? Because when you really want to kill somebody, you want to attack its core first. So the body attacks the core of the virus the first. Of course, the first to rise is the IgM of the core. Then the second antibody which rises is the surface antibody, HBsAg. Yes, because it is floating about, so then the body attacks the surface. फिर third क्या होती है? By the time it is time for the IgG to rise, तो अब core की IgG आ गई है सीन में. एंड फाइनली भाई एक जो लीकी सा घूम रहा था हमारा anti-HBe, इसके अगेंस्ट भी बना ही लेते हैं. सो दैट इज हाउ द एंटीबॉडीज है एंड दे आर म्यूचुअली एक्सक्लूसिव. अगर कभी किसी में सरफेस एंटीजन मिला है तो सरफेस एंटीबॉडी नहीं मिलेगी. अगर किसी में सरफेस एंटीबॉडी मिल गई तो उसमें सरफेस एंटीजन नहीं मिलेगा. दीज टू आर म्यूचुअली एक्सक्लूसिव ऑफ ईच अदर. ठीक है, तो अब आप जरा इस टेबल को देखते हैं. टेबल ने क्या पूछा कि कोई वैक्सीनेटेड इंडिविजुअल अगर है, सो वैक्सीनेटेड इंडिविजुअल, यू एंड मी, माय बॉडी हैज नेवर सीन हेपेटाइटिस बी वायरस, योर बॉडी हैज नेवर सीन हेपेटाइटिस बी वायरस, है ना। उसने सिर्फ किसको देखा है? सरफेस एंटीजन को। क्यों देखा है? बिकॉज मैंने वैक्सीन में सिंपली सरफेस एंटीजन को पिक करके, इट्स अ सब यूनिट वैक्सीन। थोड़ी देर पहले हमने देखा था सब यूनिट वैक्सीन में हेपेटाइटिस बी सरफेस एंटीजन पिक करके आई गिवन इन द फॉर्म ऑफ अ वैक्सीन, है ना। तो अब मेरी बॉडी ने एंटीबॉडीज बना ली है। नाउ यू गॉट वैक्सीनेटेड इन चाइल्डहुड, टुडे इफ आई लुक, विल आई बी एबल टू फाइंड सरफेस एंटीजन इन यू? एब्सोल्युटली नॉट. Surface antigen is going to be absent. Will you have anti-HBs antibodies? Absolutely, you will have these antibodies. Will you have core? No reason, na. Your body never saw the virus. If you were recovered from illness, if you were recovered, that means body has killed the virus. That means antibodies body must have produced. So anti-HBs definitely going to be there. Do you think now surface antigen is going to be there? No, right? Because the body has killed the virus. But body saw the virus. So is the body only going to produce anti-HBs? No, it is going to produce anti-core, anti-pre-core, everything. So yes, you will find, you may or may not find IgM, but you will definitely find IgG. Why? Because अगर recover हुए कुछ टाइम हुआ है तो IgM you might find, recover हुए बहुत सारे साल हो गए तो IgM चली जाएगी, but IgG तो persist करेगी ना कोर के अगेंस्ट. डन. अक्यूट इलनेस है, अभी अभी फ्रेश वायरस बॉडी में आया है, एंटीजन दिखेगा? बिल्कुल दिखेगा. फर्स्ट एंटीजन टू राइज. एंटीबॉडी बन गई क्या? अभी तो नहीं बनी है एंटीबॉडी. एंटी एचबीसी आईजीएम दिखेगा? बिल्कुल दिखेगा. फर्स्ट एंटीबॉडी टू राइज, एंटी एचबीसी आईजीएम बिल्कुल दिखेगा मुझे. आईजीजी अभी नहीं दिखेगा, राइट. एस्केप म्यूटेंट, व्हाट अबाउट एस्केप म्यूटेंट? बताओ जरा. डिजीज्ड है मेरा पर्सन, तो एंटी एचबीएस तो नहीं बना पाया है अभी तक. डिजीज्ड है, वायरस है बॉडी में, फर्स्ट एंटीबॉडी टू राइज, एंटी एचबीसी आईजीएम दिखेगी क्या? बिल्कुल दिखेगी. आईजीजी का अभी टाइम नहीं आया है. व्हाट अबाउट द सरफेस एंटीजन? इट इज एन एस्केप म्यूटेंट, इट वांट्स टू एस्केप डायग्नोसिस. These are the ones in which there is a genetic mutation in the S region of the genome, the spike coding, the surface protein coding genome. So it is going to be HBsAg negative. So when I do the diagnosis of hepatitis B, I only test for surface antigen in most of my patients. But if the patient has an escape mutant infecting him, this escape mutant is going to give me chakma. This escape mutant is going to escape the diagnosis. So that is why even if the person has illness, surface antigen is negative over here. Alright, that is escape mutant. Oh, Molshri is saying, oh, right. So that is the concept behind it. Chale, let's go ahead. Viral exanthems, super important, especially for INI exam, super important, especially the viral exanthems of childhood. Tell me, measles. Measles is caused by whom? Measles virus belongs to which family? It belongs to the Paramyxoviridae family. Paramyxoviruses. Yes, distant cousins of influenza virus. What is the rash type? What type of a rash is produced? Rash is head to toe rash, behind post auricular. So it goes in the way the girl puts her hair back. Right, so it goes post auricular, nape of neck, and then the trunk. That is how the rash moves, right? And it is maculopapular rash. So you have maculopapular rash, which starts from the post auricular region, and then it goes to the neck, and then it goes to the trunk. So you can also call it head to toe. Head to toe. Yes. What is the incubation period? Very simple. 7 से 14 दिन. 7 to 14 days. कॉम्प्लिकेशन क्या होती है? वेरी कॉमन कॉम्प्लिकेशन ऑफ मीजल्स, एसएसपीई सब एक्यूट स्क्लेरोसिंग पैनएन्सेफेलाइटिस, देन pneumonia, Hesch's pneumonia can happen because of measles, and then you have ear infections. Yes, ear infections. Excellent. Ear infections can also happen. Rubella, MMR से याद करेंगे अब. रूबेला क्या? रूबेला वायरस belongs to whom? It belongs to the Rubiviridie family. Actually, the only guy we are interested in the Rubiviridie family is the rubella virus. क्या बोलते हैं इसको? What is the disease called as? It is also called as German measles. So just like measles, the rash is going to be there. It's just going to be milder. Yes, what is the incubation period? Just double of that, 14 to 20 days. इसको into 2 कर दो, double कर दो, 14 to 21 days. What is the complication? Encephalitis very commonly happens. And why are we scared of rubella? What is the most dangerous thing about rubella? It is teratogenic. So you have congenital rubella syndrome, which is very, very bad news for the baby. Yes, congenital rubella syndrome. जिसमें क्या क्या होता है? पेटेंट डक्टस आर्टेरियोसिस, साथ में कैटरैक्ट, एंड देयर इज ऑल्सो माइक्रोसिफेली. ठीक है, डन. मोलस्कम, by the molluscum contagiosum virus, showing the Henderson-Peterson or the HP bodies. You see the umbilicated rash. ठीक है ना, एक रैश होता है जिसके ऊपर ऐसे डिप होता है, और डिम्पल्ड रैशेस और अम्बलाइकेटेड रैशेस. व्हाट इज 2 to 7? कितना? वीक्स. अब इसको बढ़ा दो इंक्यूबेशन पीरियड, अब वीक्स में चले जाओ. दिस इज अ बिनाइन डिजीज, दिस इज बिनाइन, नो कॉम्प्लिकेशंस. हैंड फुट माउथ डिजीज, HFMD is hand foot mouth disease. So this is telling me, where is my rash? My rash is in the hand, foot and mouth. What type of a rash is it? It is the blisters or vesicular rashes formed. ठीक है. It is caused by whom? It is Coxsackie virus. Coxsackie A16. Coxsackie A16 virus. Yes, it belongs to enterovirus 71. So if they ask you both, then Coxsackie is a better answer than entero 71. If Coxsackie is not given the option, then entero 71 you have to mark. 3 to 6 days, 1 week is the incubation period. What is the complication of hand foot mouth disease? All of these rashes, most common is encephalitis only, right? So it can involve the brain. Encephalitis can happen here. Viral meningitis can also happen. You know, it can also involve the meninges, not just the parenchyma. And viral myocarditis. What is the important thing here? Myocarditis. So not just the hand, foot and mouth. It can involve the heart. Roseola infantum, also called as the sixth disease. एक फिफ्थ डिजीज है, एक सिक्स्थ डिजीज है. सिक्स्थ डिजीज इज रोजियोला इन्फेंटम. व्हाई? बिकॉज ह्यूमन हर्पीज वायरस 6 and 7. सो सिक्स्थ डिजीज ह्यूमन हर्पीज वायरस 6 की वजह से होती है. यू हैव द पिंक कलर्ड ब्लांचिंग रैशेस. Pink blanching. So if you press on them, they vanish. They become pale. Pink blanching rashes are seen. कितना? 5 से 15 दिन. कोई बात नहीं, इतना इंपॉर्टेंट नहीं है. मोरलेस एक हफ्ता ही है. क्या इंपॉर्टेंट बात है? फिब्राइल सीजर्स. फिब्राइल सीजर्स है हमारी इंपॉर्टेंट बात. ओके, फिफ्थ डिजीज किससे होगी? अब आ गई फिफ्थ डिजीज. फिफ्थ डिजीज इज ऑल्सो कॉल्ड एज एरिदीमा इन्फेक्शियोसम. ठीक है, दिस इज कॉल्ड बाय पार्वोवायरस. पार्वोवायरस कौन सा? एक ही इंपॉर्टेंट है, B19. पार्वोवायरस B19. डिजीज इज ऑल्सो कॉल्ड एज एरिदीमा इन्फेक्शियोसम. एरिदीमा इन्फेक्शियोसम. क्या? स्लैप चीक अपीयरेंस. छोटा बच्चा विद चाटा पड़ा हुआ ऑन द चीक्स. वेरी क्लासिकल है ना, वेरी वेरी क्लासिकल. स्लैप चीक अपीयरेंस, पांचों उंगलियों का झापड़. ओके, फिफ्थ डिजीज स्लैप चीक अपीयरेंस. क्या होगा? 5 उंगलियां, 5 डिजीज, 5 से 15 दिन ही होगा इंक्यूबेशन पीरियड. बहुत सारे इसके देर आर many complications of this. Okay, I don't know how to zoom in. Maybe you cannot see this. Can I shift? Okay, now you can see. What all will happen? aplastic crisis can happen. Why? Because this virus loves to attack young RBCs. Erythroblastosis fetalis also it causes, but the non-immune type. So aplastic crisis is going to happen, going to kill all the young RBCs. Pure red cell aplasia, it is going to totally get rid of the red cells, pure red cell aplasia. It is going to attack the red cells specifically. Then hemolytic anemias, then hydrops fetalis. कौन सा hydrops fetalis? Non-immune. ठीक है. So the hydrops fetalis, which is the breakdown of the RBCs of the fetus, is not happening because of the RH incompatibility with the mother. It is actually happening because of the virus going and attacking. So you have the congenital hydrops fetalis or you have the non-immune type hydrops fetalis. What about chickenpox? Chickenpox is by varicella zoster virus and simple, very easy chicken. So very simple, very. Third, human herpes virus 3 is my human herpes virus. Remember the mnemonic? Simple, very easy chicken. So V is the third guy. Human herpes 3 causes multiple crops of rashes, each with fever, but sparing the palms and soles, right? Multiple crops of fever are present, meaning kabhi young crops, old crops, all types of multiple crops of rashes are there. And the rashes, they appear with fever. Every time there is a fever, there is a rash which comes, okay? But it does not involve the palms and soles. So it spares palms and soles. Alright, incubation period is 14 to 16 days. Probably the only one that you need to remember out of all. Most common complication of chickenpox. Who will tell me the most common complication of chickenpox? Secondary bacterial infection है ना, उसके ऊपर क्या हो जाए, ऊपर से secondary bacterial infection हो सकता है. Okay, very sorry about the space, there is very less space. Most fatal complication कौन सा है? Most fatal. Okay, let's do one thing. Let's write it on another page. Okay, let me put an asterisk here. And let's take a page and let's write it. I have also pushed myself out of the screen for this. Okay. So, we have complications of chickenpox because this is important. Complications of chickenpox. क्या complication है? We have the most common complication, which is the secondary bacterial infection. Secondary bacterial infection of the lesions. Then we have the most fatal complication, which is the most deadly. It is the pneumonia, the Hesch's pneumonia, which is the pneumonia due to the virus itself. Yes, then we have most common CNS complication. CNS, most common CNS complication is there. What is the most common CNS complication? Encephalitis. Encephalitis. वैसे भी you see in all of these viral exanthem diseases of childhood, encephalitis is the most common complication. Then, what if a person with chickenpox ends up taking aspirin? What happens when you take aspirin plus VZV happens? You have the Rise syndrome. Rise syndrome. Done? Done. Okay. Excellent. Now let's come on to mycology. Are we ready? Yes. Correct. You're saying all correct answers. Muffin rash, PDA microcephaly, slap cheek, aplastic crisis. Excellent. है ना? अच्छा. ये क्या दिख रहा है? Can I zoom in? No. Seems I can't zoom. Anyways, you see the hypopigmented rashes. Over here you see the hyperpigmented rash. They're both scaly in nature. So when you see the hypopigmented scaly lesion, and over here you see the hyperpigmented scaly lesion. Hyperpigmented scaly lesion. Okay. Hypopigmented is what? Tinea versicolor. Tinea versicolor. Who will cause tinea versicolor? Malassezia furfur. Malassezia furfur. Who will cause hyperpigmented? So it is black, na? Whenever it is black, it is Tinea nigra. Tinea nigra. And I have always told you, whenever you see nigra or black, you have to use the word Hortiae. So who will cause Tinea nigra? Hortiae wernickeae. Wernickeae. We will just use this Hortiae word somewhere else also. Whenever there is black, okay, let's see only. This was skin. On skin, you might have a hypopigmented lesion, hyperpigmented lesion. What if about the hair? In hair, if you have black nodules or you have black piedra, so infection of hair is piedra. You might have black piedra or you might have white piedra. व्हाइट पिएड्रा है ना. सो ब्लैक आ गया यहाँ पे भी. सो जैसे ही ब्लैक आया तो हॉर्टिए वर्ड तो आना पड़ेगा. डिजीज क्या है? पिएड्रा. सो ब्लैक पिएड्रा इज कॉज्ड बाय पिएड्रिएया हॉर्टिएई. So you see the word Hortiae and you immediately know either nigra or black is going to be there. ठीक है. Who causes white piedra? We will see in a little bit. Let's go back to Malassezia furfur. Let's go back to Malassezia furfur. क्या दिख रहा है KOH पे? You can see on KOH you can see the spaghetti and meatball appearance. Spaghetti and meatball appearance. ठीक है. What do you see on SDA? This is an SDA with olive oil overlay because this guy loves the sebaceous side. That is why it causes seborrheic dermatitis. That is why it causes atopic dermatitis because this is also a super antigen. We have seen in the slides above, है ना? Yes, Piedria hortiae. You're correct. So hypopigment. अभी हम Malassezia की बात करें ना. KOH spaghetti and meatball. What type of an SDA I am using? I am using SDA with olive oil overlay. I have overlaid the SDA with olive oil. So this is showing me the fried egg colonies. Fried egg colonies. Okay. And what is another thing about it? This guy is urease positive. Urease positive. So tell me, what are the urease positive fungi? UP का deputy chief minister. UP का deputy CM. So you have urease positive DCM. Okay, urease positive DCM. So what is DCM? D is dermatophytes. So all dermatophytes are urease positive. C, cryptococcus. Cryptococcus is urease positive. M, Malassezia furfur is urease positive. That becomes your urease positive fungi. Okay. Alright. Are we good up till now? Now let's go back to the piedra, which was the white piedra. So जब भी बिजली चमकती है, the moment there is lightning, तो बिजली चमकी तो सब white हो जाता है sky में, है ना? So who will cause white piedra? Trichosporon bijili. Yes, Trichosporon bijili. So remember, बिजली जब चमकती है तो white होता है. Yes, Trichosporon bijili. Sporon, it's a yeast. Are we good? Are we ready to go? So now we have talked about skin lesions. So we have tinea versicolor, tinea nigra. We have talked about piedra. Now we need to talk about the dermatophytes. So whenever I say dermatophytes, there has to be a microconidia or there has to be a macroconidia. How do we remember the names of the dermatophytes? We say too much emotion. By this time, after 2 hours of lecture, me speaking continuously, you are all tired and you are feeling too much emotion. So let's do one thing. Let's try to classify the dermatophytes, right? So whenever you have a dermatophyte, the dermatophyte is going to cause infection in your skin, in your hair and in your nails. Okay, skin, hair and nails. And what is the classification? Too much emotion. You start feeling too much emotion by this point. So it is TME. Okay, TME. So TME. With T, you have trichophyton. Trichophyton. M is microsporum. Microsporum. E is epidermophyton. Epidermophyton. Yes. What is trichosporum going to cause infection? Tri in all three. Yes, tri is all three. So it is going to cause infection in skin plus hair plus nail. What about microsporum? Microsporum. So they will all cause infection in skin, no problem. Microsporum will cause in hair. One of my students gave the mnemonic Maharashtra, MH. So just remember MH. So microsporum will cause in hair. What about epidermophyton? Nail. ठीक है. So epidermophyton has no microconidia, no नहीं है nail, ऐसे भी याद कर सकते हैं. अब छोड़ो. अब now these dermatophytes, they have some microconidias and some macroconidias. Okay, microconidias and macroconidias. So let's look at these images as LPCB images. You see these round small structures, these small round structures, they are the microconidia. So if I write here microconidia, I can see that microconidia are abundant. There are lots of microconidia in this picture. What about here? 4, 5 I can see here, and in this I can see none. So I am saying that there are very few microconidia in this picture. And over here I can see absolutely no microconidia. So there are no microconidia. So how to remember? See, when you start reading microbiology, you remember lots and lots of micro. So when you start the classification of too much emotion in trichophyton, initially less emotion but बहुत सारी micro याद है. Then emotion starts increasing in microsporum, but microbiology भूल जाते हैं. Finally, epidermophyton, emotion is very high and all micro has vanished from life. We have forgotten all microbiology. So, no microconidia left in the end. No microconidia left. Okay, this is epidermophyton. What about macroconidia? What about macroconidia? You see, first they are like thin like pencil. So they are pencil shaped. Then they become a little big. So when you swell up a pencil, it becomes a spindle. So now the pencil has become a spindle. So they are spindle shaped. You swell up the pencil a lot more, it will become big like a club. So finally you have club shaped macroconidia. So as your emotion is increasing, your pencil is swelling up. So it's a pencil, then it's a spindle, then it's a club. So the difference between microconidia and macroconidia of the first one here is the trichophyton. This we are talking about trichophyton. This is trichophyton. This here is microsporum. And this here is your epidermophyton. There is only one important species of epidermophyton you need to remember. So just remember epidermophyton flocosum. Nothing else is needed. Flocosum. Okay, nothing else is needed. Then what about microsporum? You have to remember two guys. Okay, microsporum canis and microsporum gypsum. जिसमें canis is a little important. We will see. And in trichophyton, you remember trichophyton ruberum, right? So ruberum is going to be red. Yes. And you remember trichophyton mentagrophytes. So mentagrophyte क्या है? Mental है. So when you are mental, you are on loopy. When you are loopy, you spiral. So mentagrophytes has spiral hyphae. It is mental, it is loopy, it has the spiral hyphae. Yes, because it is mental, so it is loopy. And T. ruberum is also called as bird on fence appearance. Just imagine red colored birds sitting nicely on the fence. Now another reason why I have told you this these species is you have to remember two things now. You have to remember that there is a test that we perform to identify these dermatophytes. dermatophytes. Okay, so for dermatophyte identification, let's write here. There is a test called as the hair perforation test. So what do? I take a hair of a baby, a naive hair, a small baby's hair, and I put some moldy growth on it. So I have taken the hair and I have the moldy growth of my dermatophyte, and I put it on it, then I put it in a dark corner for a few days, let it rest nicely, and this ये जो तुम्हारी फंगस है ना, it is so बदतमीज, it is going to eat up the hair like dogs. So like the dogs eat up hair, so what is going to be the mnemonic for your fungus which can eat up the hair? Men can, है ना. Obviously when we are saying canis or dogs, we are saying men as well. So men can. So what are the two fungus? Trichophyton, mentagrophytes, the loopy pagal spiral one, and Microsporum canis. They are both going to be hair perforation test positive. Mentagrophytes and varicosum will show hair perforation test. ठीक है? Microsporum canis. So यहाँ पे अगर मैं लिखूँ hair perforation test, so Trichophyton mentagrophytes is going to be hair perforation test positive. Microsporum canis is going to be hair perforation test positive. Do you think rubrum will give? No, rubrum will be hair perforation negative. है ना? Men can. Then I will ask you, give me the examples of red pigment producing molds. Yes, red pigment producing molds. So you will tell me two guys, Trichophyton rubrum, है ना. Trichophyton rubrum, that is going to be a red pigment producer. Name itself calls it rubrum. और दूसरा क्या था जिसमें झाड़ू से मार मार के लाल कर दिया था हमने? Talaromyces marnefi. Talaromyces marnefi. Old name is Penicillium marnefi. Penicillium marnefi gives the broomstick appearance. Broomstick appearance on LPCB, है ना. So हमने क्या बोला था? ब्रूम से मार के लाल किया. Okay, broomstick appearance. All right, so you have the red pigment producing. Yes, Penicillium marnefi, brick red. Excellent. Brick red pigment producing. ओके, एक्सेलेंट. ठीक है, और कुछ रह गया हमारा? सो वी हैव अबंडेंट माइक्रोकोनिडिया, फ्यू माइक्रोकोनिडिया, नो माइक्रोबायोलॉजी, नो माइक्रोकोनिडिया. ओके, डन, डस्टेड. नाउ मैच द फॉलोइंग. Try to match the following. Tell me, what can you see in the first one? You can see the vesicle, you can see the khopri of the aspergillus, and you can see the head full of conidia. And you can see that it is looking like somebody has pulled the hair. सिर्फ upper 1/3 में ही conidia है. Pulled hair appearance. What am I seeing here? Pulled hair. किस में दिखती है pulled hair appearance? It is Aspergillus fumigatus, है ना? Aspergillus fumigatus. Fumigatus कैसा दिखता है? ग्रीन कलर का. यहां तो मुझे कुछ नहीं दिख रहा. कैन आई सी द थिन सेप्टेट डाइकोटमस ब्रांचिंग हाइफे एट एन एक्यूट एंगल? ऐसे हाइफे हैं, बराबर डिवाइड हो रहे हैं, डाइकोटमस एंगल और ब्रांचिंग, और ये क्या है? एक्यूट एंगल. सो आई कैन सी द थिन सेप्टेट ब्रांचिंग at acute angle and dichotomous branching. Hyphae. Okay, that is, you can see. So A is matching with 2. What about this? This is also Aspergillus, but this is the flavorful lime green. तो लाइम ग्रीन से हम याद करते हैं फ्लेवस. सो दिस इज Aspergillus flavus. Can you see Aspergillus flavor anywhere? Here, head full of hair, no pulled hair, head full of hair. You can see the 2/3 covered vesicle with the conidia. 2/3 covered vesicle with the conidia. So this is Aspergillus flavus. So B becomes 3. What is this? This is the brown colored niger. This is also brown colored niger. So this becomes 1, right? Head full of brown hair. Aspergillus niger. Head full of brown hair. Done? बहुत easy था. Next slide में देखो क्या है. ये क्या है? रेड पिगमेंट, ब्रिक रेड पिगमेंट प्रोडक्शन, ग्रीन कलर्ड फंगस, ये क्या है? झाड़ू, ब्रूमस्टिक अपीयरेंस. सो दिस इज द ब्रूमस्टिक और द पेंट ब्रश. झाड़ू से मार मार के लाल किया, ब्रूमस्टिक और द पेंट ब्रश. कैन यू कॉल इट Telleromyces marnefi? And this is the brick red pigment production. Brick red pigment production. Yes, of course, this is Telleromyces. This is Telleromyces marnefi, which is a dimorphic as well as an opportunistic fungus. है ना? Dimorphic है. So D matches with 5. What is this? Flower-like conidiations. I can see some florets and flower-like conidiations. Here what can I see? I can see the asteroid bodies or the splendor Hopley phenomenon. What is this? This is asteroid bodies, cigar-shaped asteroid bodies. And what can I see here? I can see the flower-like conidiations. तो flower कहाँ बनेंगे? Rose gardener's disease में, है ना? So this is sporotrichosis. This is sporotrichosis because you see the floret-like conidiation at LPCB. LPCB में दिख रहा है. और asteroid bodies कहाँ दिख रही है? At tissue. At tissue. So tissue मतलब at 37 degrees Celsius. LPCB means at 25 degrees Celsius. This is also a dimorphic fungus. And so E is 6. Why? Because this guy here is sporotrichosis. Sporothrix schenki. Sporothrix schenki. Then what is this? Umbrella type. This is molds. You can see the broad aseptate hyphae. Yes, can you appreciate the broad aseptate hyphae? This is your mucorale. Mucorale. Broad aseptate hyphae. And you can see here totally filled the tube. So tube filler or a lid lifter. Tube filler or lid lifter. Okay, so this is your mucorale. So F becomes 4. Mucorales. Done? Done. Easy, but we have been able to revise very, very nicely. Thorn prick. The moment you get the history of a thorn prick, what do? You know you have to talk about 3 important guys in a thorn prick. Are we ready? Are we with me? Up till now? Any issues up till now? I don't think so. So whenever they say thorn prick, they ask you 3 important things. Either it is going to be sporotrichosis. If it is sporotrichosis, it will be nodules along the lymph nodes. Yes, nodules along the lymph nodes, and this is called as the rose gardener's disease. Rose gardener's disease. Okay, or it will be Medulla mycosis. Medulla mycosis. What is Medulla mycosis? Madura foot. Madura foot. Caused by Madurella species. Madurella species. What is special? You will have discharging sinuses. And what? Black granules. Black granules from the discharging sinuses. Discharging sinuses with the black granules. Important. And thirdly, it can be chromoblastomycosis. The only 3 possibilities for getting the thorn prick. In fact, November 2025 INICET, they asked the question exactly like this. Exactly in this format, they asked the question. That thorn prick, which of the following does not cause? 3 out of the 4 options were sporotrichosis, Madura mycosis and chromoblastomycosis. So obviously the odd one out you had to pick. Here it will show varicose. Yes, varicose lesions. Hai na? So chromoblastomycosis, what is it showing me? See, chromo means colored, blasto means round and mycosis means fungal disease. So these are the brown colored round bodies. What do I call them? Copper penny bodies. Copper penny bodies, or they are also called as sclerotic bodies. Sclerotic bodies, or they are also called as medullary bodies. Medullary bodies. Okay, and they show which type of the disease? Verrucous plaques. Verrucous plaques. Got it? Obviously, there is going to be thorn prick or blunt trauma as the history given. Okay, now what is happening here? in systemic mycosis. What is systemic mycosis? Bloodstream infection because of molds. So systemic mycosis mostly happens with the dimorphic fungi. Why does it happen with the dimorphic fungi? Because mold cannot roam about in your blood ना, इतना जैसा bread mold होता है इतना heavy सा, only yeasts can roam about. So who can infect your bloodstream? Either Candida or Cryptococcus, these yeasts, or dimorphic fungi. Dimorphic have the ability to exist as both molds and yeast. तो जब तुम्हारे blood में घूम रही है तो it will behave like an yeast. जब हम उसे lab में grow करेंगे it will start behaving like a mold. So only dimorphic fungi can lead to systemic mycosis. Now there is a very easy way to remember the agents of systemic mycosis. Okay, let's see. Systemic mycosis. तो एक ना story सुनते हैं. Now let's back up, let's drink some water and let's listen to a story. Are we ready? Ready? Yes. Okay. Just imagine you went to a party. When you go to a party, who do you meet? You meet a hot boy. Yes, a hot boy. What will that hot boy do? That hot boy will call you darling. That hot boy will say, come to my cave. That hot boy is an American guy. He's from Ohio. His name is Gilchrist. Okay. That hot boy is a bad boy. What will the bad boy do? The bad boy will ghost you because he can party. Hot boys can party. Okay. If they can party, they are going to ghost you. They can party, they are going to party in the coolest place where parties happen. Where do the coolest parties happen? They happen in Las Vegas, which is a valley. It is a desert. Then party karte karte, where will he take you? He'll take you on a ship. He's a mariner, he's a sailor, he's a pilot, and he's going to take you to South America, that is Brazil. So hot boys can party is going to be a mnemonic, and this story is going to sum up the entire picture. So this hot boy is histoplasma. This hot boy is histoplasma. He is calling you darling. So this is darling disease. He is from Ohio and his name is Gilchrist. Okay. That is all histoplasma. Then he is a boy. He is a bad boy. Okay. So he is blastomyces. Blastomyces. He is a bad boy. He has the broad-based budding appearance, and this is the North American disease. Okay, where do the bad boys come from? From North America. Okay, he is a bad boy, so there is the bad antigen which is going to act. Then he can party. He can party. So it is coccidioides. Coccidioides. Where is he going to party? If he can party, he is going to party in Las Vegas, which is a desert. So desert rheumatism. Or it is a valley. You can also call it valley fever. Valley fever. What is the hot boy going to do? He is going to ghost you. So you see the ghost cells. Right? Then he is going to take the party to South America. What is this? Paracoccidioides. Paracoccidioides. And who is he? He is a sailor. He is a mariner. He is a pilot. This guy, of course, he's a hot boy, so he's all of these things. So all of these wheel appearance you will see. What else? Where has he taken you? He's taken you to South America. South America. South American disease. And if it is South America, where will he take you? Brazil. So Paracoccidioides brasilensis. Paracoccidioides brasilensis. Coccidioides immitis. Immitis. Blastomyces dermatidis, and histoplasma capsulatum, where capsulatum is a misnomer, right? Yeah, some students are laughing. So that is how you remember the entire story. You met a hot boy, he was a bad boy, he ghosted you, and then he took you to South America on his ship. Excellent. So you look at him. Who is he? This is the gradient staining, intracellular budding yeast cells with the gradient staining. What are you seeing? You are seeing intracellular budding yeast cells with gradient staining. What is the meaning of gradient staining? This is an yeast cell. It is more stained at one side and less stained on the other side, like a crescent shaped moon. This is gradient staining. And over here, when I made an LPCB, so this is tissue. This is tissue. So at 37 degrees Celsius, it is showing me the yeasts. And what is this? This is my LPCB at 25 degrees. This will show me the molds. Histohides and macrophages. Yes, it is intracellular inside the macrophages. है ना? At 35 degrees Celsius, it is showing me what? The tubular conidiations, the tubercles. So this is a conidiation and these tubercles are arising out of this. These tubercles are arising out of. So what is this? This is tubercle conidiation. Tubercles conidiation. Okay, excellent. So histoplasmosis, Darling disease, cave disease, Ohio disease, how does it happen? Bird droppings. So in the nitrogenous soil because of the bird droppings, and we have just seen that what is spread by birds. So we had given the mnemonic of histoplasma there. Okay. Then hot boy, now the bad boy has come. What can you see here? What can you see here? You can see the BBB. You can see the broad-based budding of the Blastomyces dermatidis. Yes, it is called as North American disease, the big bad boy, and it is by the BAD1 antigen. BAD1 antigen. Important to note, it causes disease in the immunocompetent people. Immunocompetent host में it causes the disease. नेक्स्ट कौन है? ये कौन है बीच बीच में? गोस्ट सेल्स. हु गोस्टेड यू? बैड बॉय गोस्टेड यू. These are the ghost cells, and over here you can see the spherules forming. What is this? This is Coccidioides immitis. Coccidioides immitis. Immitis, valley fever, desert rheumatism, all of that we have already written, not wasting your time. What is this? Mariner's wheel, Mickey Mouse, pilot wheel appearance, Mariner's wheel, pilot wheel, Mickey Mouse. South American disease, Paracoccidioides brasilensis. Any problem up till now? Lesion mean mimic SEC. Yes, correct. Everything. So this is what I wanted to tell you about systemic mycosis all in all, and we have also looked at the pictures. What is this? This is a gomorrhomethamine silver stain. Yes, isn't it? It is because it is black. This is histopath staining, HNE staining. What is this? This is also gomorrhomethamine silver stain. So these are all GMS stains because you look at the black colored fungi in this, right? Shall we move ahead? Let's see. Let's talk about toxoplasma. We've come on to parasites. Guys, are we ready to discuss the parasites? Toxoplasma gondii. Toxoplasma gondii. What does it cause? It causes the toxoplasmosis. What are the infective stage, diagnostic stage? So in how many stages does it appear? There is a oocyst. Okay, oocyst is secreted where? It is excreted in the cat feces. Then it sporulates in the soil. Yes, it sporulates in the soil and then it is ingested. So ingested is the feco-oral. So yes, of course, this can be the infective stage. Can I diagnose it in the stool? Yes, I can. So it can be the diagnostic stage. Let's look at the next stage. Next stage is called as the tachyzoid. Tachy means fast. So these are fast multiplying ones. So they are present in the blood. So I can see them in the peripheral smear. I can see them in the CSF of the patient. I can see them in the placenta. They cross there. They're present in the blood. Do you think they will become the mode of infection if I do a blood transfusion, if I do an organ transplantation, or if there is the cross-placental transfer, if it has to go from mother to child, cross-placental transfer, transfer. So if I take the peripheral smear, can I see them? I can see them. So are they diagnostic? They are diagnostic. Can they pass from one person to other? They can. So are they infective? They are infective. That is also infective stage. The third one is the bradyzoid. Brady means fast. Slow. Why has it become slow? Because it has been encapsulated inside the cyst, so inside the tissue. So this is also called as the tissue cyst stage. Tissue cyst stage. If I take biopsies and I do look, can I find them? Yes, I can find them. I can do the diagnosis. If I ingest the raw meat, of an animal in whom the bradyzoids are sitting, can I get the disease? Yes, I can get the disease. So all three stages in toxoplasmosis are infective and all three stages in toxoplasmosis are diagnostic, right? Who's the intermediate host? It is going, who's the definitive host? It is going to be cat, no problem. Plasmodium. What is the infective stage of Plasmodium? Sporozoite. The sporozoite enters my body, goes to the liver, becomes, merozoite, then comes out into my blood, keeps roaming. Okay. How does it enter? Through the circumsporozoite protein. The sporozoite enters inside my body. Then what is the diagnostic stage? How do I diagnose malaria? By looking at the peripheral blood smear. So all the erythrocytic stages are going to be my diagnostic stage. So my ring stage, my trophozoite stage, or my gametocyte stage. Yes, or my gametocyte stage. What are the important ones you have to remember? The banana shaped gametocyte of Plasmodium falciparum and the band shaped trophozoite of Plasmodium malariae. These are important. Then in the ring stage, you have multiple rings. Falciparum is fancy, so it has multiple rings. A coal stage, multiple dot rings, one ring with many diamonds. Yes, Vivax is a simple guy. He has got single ring stages in the ring stage of Plasmodium. Okay? Now, what important to remember in Plasmodium is the stippling. Stippling. How do we remember stippling of Plasmodium? We have a funny mnemonic for it. Just imagine you have gone to a zoo, and in that zoo you see that there is, my zoo has fancy, smart, very smart and fancy monkeys. There are very smart, fancy monkeys who are drinking orange juice. Okay, they are drinking, what are they drinking? Orange juice. Orange juice. This is your mnemonic for stippling. Okay, my zoo. So Plasmodium malariae has Zeeman dots. Very smart. They are very smart monkeys. So Plasmodium vivax has Schaffner's dots. They are fancy monkeys, right? So falciparum, Plasmodium falciparum has Morer's dots and orange juice. So it is Plasmodium ovale, which will show me the James dots. Right? So that is the stippling of malaria, right? Okay, thank you so much guys for the compliments. Now, let's talk about cysticercosis. Okay. Cysticercosis में क्या होगा? Cysticercosis is by Taenia solium. But Taenia solium can also cause intestinal teniasis, है ना? तो Taenia solium जब cysticercosis करेगा, so we are talking about neurocysticercosis. Mostly we are talking about neurocysticercosis. अब neurocysticercosis कैसे होता है? Through ingestion of what? Now who will tell me this? The person who tells me this is really prepared. Come on. Infective stage क्या है इसकी? And diagnostic stage क्या है इसकी? And what about? Then we will talk what about the intestinal teniasis. Then we will talk about intestinal teniasis. Intestinal teniasis में pork tapeworm, Taenia solium. ठीक है, इसका क्या इन्फेक्टिव स्टेज है और इसका क्या डायग्नोसिस स्टेज है? कम ऑन, ये बताओ. Anybody, quickly, fast, we are short of time. Cysticercosis. Whenever, so, कितने stages होते हैं टीनिया के? There is the adult lays the eggs में से निकलता है larva बड़े होके बनता है adult. तो अगर तुम्हारे स्टमक में adult बैठा है तो वो eggs को ले करेगा, ठीक है? That is intestinal teniasis. Adult sitting in the intestine laying eggs in your feces. So, अगर आपके स्टमक में एडल्ट को बैठना है तो आपने क्या इंजेस्ट किया होना चाहिए? स्टेज बिफोर दैट, इमेजिन यू इंजेस्ट द लार्वा. हाउ विल यू इंजेस्ट द लार्वा? लार्वा सिट्स इन स्केलेटल मसल. सो यू एट द पोर्क के मसल्स में देयर वाज लार्वा ऑफ टीनिया सोलियम सिटिंग. व्हाट इज द लार्वा ऑफ टीनिया सोलियम कॉल्ड एज? सिस्टिसर्कस लार्वा. Suppose you ate the meat and with the meat you ate the Cysticercus larva. Cysticercus larva. What Cysticercus? Cysticercus cellulose. Now you have ingested the larva in your intestine. Larva will become adult. So that adult will cause intestinal teniasis. What will that adult lay? Eggs. So what is your diagnostic stage? Eggs. What is your infective stage? Cysticercus larva. What about neurocysticercosis or cysticercosis? Ultimately, what is sitting in your brain? The larva is sitting in your brain. Yes, who is sitting in your brain? The larva. So what is the diagnostic stage? You see the larva on radiology. The shiny lesions on radiology. The larva cysticercus cellulose you are seeing. What is the infective stage then? How has the larva reached your brain? Just imagine that you ate the eggs along with the salads that you were eating, the vegetarian food that you were eating, or because of your own poor hygiene, because of auto-infection, you ended up eating the eggs of the parasite. Those eggs hatched in your body, larva came out. Now this larva migrated to your brain and there it caused the neurocysticercosis. So always remember, for cysticercosis, eggs is the infective stage and larva is the diagnostic stage. For intestinal teniasis, larva is the infective stage and eggs become your diagnostic stage. Then you have Taenia saginata. Taenia saginata is beef tapeworm. Same thing repeated, just that the worm has changed and the mode of infection is now beef. Same. What is the infective stage? You have eaten the larva. What is the larva of this? Cysticercus bovis. What is the diagnostic stage? The eggs of Taenia become your diagnostic stage. What is the feature of eggs of Taenia? They are all going to be bile stained eggs. Bile stained eggs with the hexacanth embryo. Hexacanth embryo, no problem there. Alright. Saginata bovis larva. Peripheral striations, yes, radial striations. This is the egg with the radial striations. Correctly said. Very nice. है ना? चलो. Hydatid cyst. Hydatid cyst disease is caused by the dog tapeworm. Who is the definitive host? Dog. डॉग टेप वर्म है तो ह्यूमन इसमें भी इंटरमीडिएट होस्ट है. दिस इज इकाइनोकोकस ग्रैन्युलोसस. अभी देखा था हमने थोड़ी देर पहले. इकाइनोकोकस ग्रैन्युलोसस कॉजेज द हाइडेटेड सिस्ट. इसकी इन्फेक्टिव स्टेज क्या है? सो डॉग टेप वर्म. सो डॉग इज योर डेफिनेटिव होस्ट. तो ह्यूमन है क्या? व्हाट इज ह्यूमन इन दिस? Human is actually intermediate host. Yes, intermediate host. Is human an accidental host? हाँ, गलती से आया ह्यूमन नहीं चाहिए था साइकिल में, डॉग से काम चल रहा था. ह्यूमन इज एन एक्सीडेंटल होस्ट. अब ह्यूमन में आ गया तो क्या ह्यूमन को आगे कोई खाएगा? कोई नहीं खाएगा. ह्यूमन बिकम्स द एंड होस्ट और द डेड एंड होस्ट. ह्यूमन बिकम्स द डेड एंड होस्ट ऑल्सो. ठीक है. इन्फेक्टिव स्टेज में क्या होगा? इन्फेक्टिव स्टेज कैसे इन्फेक्ट होंगे हम हाइडेटेड सिस्ट से? यू ईट द एग्स. स्पेसिफिकली यू एट द एम्ब्रियोनेटेड एग्स. हाउ डिड यू ईट? डॉग की फीसेस में एग्स वर कमिंग आउट. डॉग के ऊपर एग्स लगे हुए थे. बेबीज दे प्ले विद डॉग्स, डोंट वॉश देयर हैंड्स, ईट. व्हाट डू दे एंड अप ईटिंग? दे एंड अप ईटिंग द एग्स व्हिच वर स्टक इन द फर ऑफ द डॉग्स. What is the diagnostic stage then? This guy, hydatid cyst. You will look at the cysts which are present inside the body. So hydatid cyst is your diagnostic stage for this guy. Okay? आगे आते हैं. हुकवर्म कौन थे? दो. एक खून पीने वाला नॉर्थ अमेरिकन किलर निकेटर अमेरिकानस इज योर नॉर्थ अमेरिकन किलर. एंड एंकाइलोस्टोमा डुओडेनेल is the other guy who's a hookworm. What do? They eat up all your blood. So they are going to lead to iron deficiency anemias. If you see this buzzword in the question, you know they're talking about hookworms. And B12 deficiency is caused by whom? Chat box. Come on, open question. B12 deficiency is caused by whom? So iron deficiency anemia causing parasites. Dysentery causing trichuris and hookworms. B12 deficiency anemia causing parasites. Let me know in the chat box. Come on. Okay. Both are non-bile stained, that is okay. I am asking you, tell me about B12 deficiency anemia. Ouch. Okay. Infective stage क्या होगी of the hookworm? The hookworms and strongyloids, they are different. They enter by skin penetration. Skin penetration. Skin penetration. By the infective means the filariform larva. How do they enter? They enter through the filariform larvae. Okay. And what is the diagnostic stage then? Either the eggs they come out, and what do the eggs look like? Non-bile stained with the blastomeres. Non-bile stained. Neha. Neha is your mnemonic. Non-bile stained. Nicator, Enterobius, and H. nana, and your ankylostoma. They are both all non-bile stained eggs. Or you can also see the rhabditiform larva. Rhabditiform larva. Okay. Iron deficiency anemia caused by Trichuris trichiura because it causes dysentery, or it is caused by hookworm. Hookworm. Right. In other note, the question that I was asking you, B12 deficiency, Diphyllobothrium latum, fish tapeworm, largest tapeworm, Diphyllobothrium latum. Okay, finished. Important. Yes, Neha, rightly said. Next क्या है? Liver fluke. What is liver fluke? Called as Fasciola hepatica. It is a trematode. Fasciola hepatica. It is a ट्रेमाटोड. ठीक है, जहां पे भी एफ का साउंड आता है, चाहे फैशियोलॉप्सिस बुस्की, चाहे फैशियोला हिपैटिका. What is the infective stage? It is ingestion of fresh aquatic plant. So what are we eating along with? So for all the trematodes, what is my mnemonic? Myceli red cat mews. My Miracidium, Cili, Sporocyst, red, Redia, cat, Cercaria, and meows, Metacercaria. So what are we ingesting? Just like all trematodes, we are ingesting the Metacercaria larva. मेटासर्केरिया लार्वा है ना. मेटासर्केरिया लार्वा कहाँ पे बैठा हुआ है? फ्रेश aquatic plant. Fresh aquatic plant. So F for fasciola, F for fresh aquatic plant. So for all trematodes, the first intermediate host is always snail. And what is the second intermediate host? That is going to vary depending upon the organism we are talking about. Then what is the diagnostic stage? Operculated eggs. All trematodes are going to have dhakkan wala eggs, operculated eggs. Who else is going to have an operculated egg? The dhakkan of the cestode family. He is also going to have the D for dhakkan, operculated eggs. Okay, he is also going to have them. So diagnostic stage is operculated egg, and adults can also be seen in the liver, right? Or it can be diagnosed clinically. A simple clinical diagnosis. So who have operculated eggs? It is all trematodes and Diphyllobothrium latum. Okay, all trematodes and Diphyllobothrium latum, they both have operculated or dhakkan wala eggs. Let's look at another trematode, Clonorchis, Clonorfish, Opisthofish. So over here the Metacercaria is coming on the fish. This is also Metacercaria, but now on the fish. Metacercaria on the fish. Diagnostic stage, it is operculated eggs again. Same, operculated eggs. ठीक है. नेक्स्ट कौन है हमारा? नेक्स्ट इज शिस्टोसोमास. शिस्टोसोम्स आर ऑल्सो ट्रेमाटोड्स, बट दे आर द एक्सेप्शन किंग्स ऑफ द फैमिली. सो इफ वी राइट शिस्टोसोम्स लाइक दिस, लेट्स राइट शिस्टोसोम्स. Schistosomes. All exceptions also start with S. So SC means skin penetration. All trematodes were by ingestion. These are by skin penetration. All trematodes were by Metacercaria larva. This is by Cercaria larva. So who is coming inside the skin? The Cercaria larva. So what is the infective stage for schistosoma? It is skin penetration by Cercaria larva. Yes, skin penetration by the Cercaria larva. Cercaria larva. Then S2. S2 means what? Two sexes. two sexes. So all trematodes are monoecious. These guys are dioecious. And SO means what? Snail only. So this is the only trematode where there is only one intermediate host. There is no second intermediate host. Okay. Then S, where is the female going to live? Female is going to live inside the male. So male is going to have a slit-like body. So while all trematodes are fluke-like or leaf-like body, cystosomes are slit-like body. And what is the other S or the last S? They don't have operculated eggs. They have spined eggs. Eggs with spine in them. So they have spine or non-operculated eggs. No dhakkan on the eggs. Yes, spine or non-operculated eggs. These are all the exceptions of the schistosomes family. Yes, in one place only we have been able to classify this. So when I say schistosoma, hematobium, you see the terminal spine. In schistosoma, hematobium. What does it cause? Bladder carcinoma. Then you have tedhimancy, lateral spine in schistosoma, Mansoni, tedhimancy. Then you have cute as a button. You have knob or button, and you have small round eggs. So Japanese people are cute as a button, right? So you have schistosoma japonicum. This is what the eggs of these guys look like. Schistosoma japonicum. Excellent. Let's move ahead. What can you see over here? You can see some acid-fast balls. You can see some acid-fast coccidian parasites. So these are coccidian parasites. Cryptococcus. Crypto means hidden. So this guy will not autofluoresce. So in Cryptococcus there is no autofluorescence. In all others there is autofluorescence, right? These are round and acid fast, and these are small, around 4 to 5 micrometers. Right? What are these? These are also acid fast, but these are big. This is cyclospora, just double, 8 to 10 micrometers, and they are non-uniformly acid fast. You see, this is taken acid fast in, this has not taken, this has not taken. So this is non-uniformly acid fast. And who is this? This is completely oval. This is cysto. Cysto means cyst, and iso means same. Spora. Cystoisospora. This is oval. This is big. Iska और double कर दो. 10 to 15 micrometers in breadth and 20 to 30 micrometers in length. Okay, that is the size. Cryptococcus, this is also called as naked sporozoites. Why? Because this is one cyst, and inside this there are the 4 sporozoites. So total there are 4 sporozoites which are naked. Naked means there is no double bond. These are naked sporozoites, right? So because of this nakedness, this can lead to auto-infection. And this guy goes and sits at the top border of the epithelium, making what we call as the parasitophorus vacuole. Parasitophorus vacuole. These all are important in HIV positive diarrheas. Okay, these are all important for causing HIV positive diarrheas. Right? What about cyclospora? Cyclospora is like this, and then there are 2 cysts inside. And inside that there are the 4 sporozoites. 4 sporozoites in the double walls. What happens here? This is the oval one. Then there are 2 cysts inside and 4 in each. So there are total 8 sporozoites here. Alright. Okay. Let's look. Coccidian parasites. We have just seen. What is the infective stage? Sporulated oocyst. Okay, the oocyst which we are eating has to be sporulated. What is diagnostic? This thing coming out in the stool, it can be either sporulated or non-sporulated. These guys which are shedding out, the naked sporozoites, are already sporulated. That is why autoinfection can also happen in them. So what is the diagnostic stage? In whom the sporulated oocysts come out? It is the Cryptosporidium. So sporulated oocyst is the diagnostic stage for cryptosporidium. Cryptosporidium, and that is the reason that only this guy can lead to autoinfection. Or unsporulated oocyst can also come out. Unsporulated oocyst is coming out, then it is taking its sweet time to develop in the soil, then becomes sporulated, then cause infection. So infective stage is always sporulated. Diagnostic can be both sporulated or unsporulated. Unsporulated is going to be in the case of cyclo and cystoiso. Okay, in that case it is going to be the unsporulated oocyst. Okay, I hope you remember the mnemonic for autoinfection: chest, cryptosporidium, H. nana, Enterobius verbicularis, Strongyloids stercoralis, Trichuris trichiura, and Tinea. Okay, please pause the video and write down if you have not written. I have just said it out. Then Giardia. Giardia looks like what? Giardia looks like this with the exostyle and the nuclei, the 4 eyes. Yes, and it also has a cyst. A cyst has a central exostyle and you have the nuclei. Yes, and it is a small cyst. So what is the infective stage of Giardia? It is the cyst. We end up eating the cyst. What is the diagnostic stage? It is the trophozoite. Giardia is also a protestant parasite. All protozoa make trophozoites and cysts. What is the size of the cyst? Around 8 to 10 micrometers. What is the motility shown by the trophozoite? It shows the classical falling leaf motility. So depending upon how fresh the sample is, you can see either cyst or you can see the, yes, chest, correct, as tennis racket. Okay, there's high and bi happening. Cryptosporidium, nitazoxanide, other two are cotrimoxazole, H2SO4. Okay, yes. Thanks for reminding me actually. Correct. Oh, you guys have been writing spellings. Okay. Right. So, you are right. What is the modification of Zn stain used here? We have used the modified Zn stain. Modified Zn stain. And what is the modification used? It is the coccidian parasite. So, it is the cold method. So that is also called as the Quinones method. And what is the percentage of H2SO4 that we use? We use 0.5 to 0.25% H2SO4 for this Zn staining. Correct. Thanks for pointing it out. Giardia, falling leaf motility we have seen. Yes, it's the Tanis racket formation. Great. Entamoeba. Which entamoeba we are talking about? We are talking about entamoeba histolytica. Name itself says it is going to cause amoebic liver abscess. It is also going to cause intestinal amoebiasis. What it causes is intestinal amoebiasis? Dysentery. It causes dysentery in intestinal amoebiasis, right? What is the infective stage? This mature guy with the 4 nuclei. So you have the mature quadrinucleate cyst is the only infective stage. All other stages do not cause infections directly. If they go inside, they are going to burst out and collapse. So what are the diagnostic stage? It is the cyst stage. Any. There can be the uninucleate, binucleate, quadrinucleate. Any stages can be the cyst stages, the diagnostic stage. And of course, you can also see the trophozoite. What is special about the trophozoite? This guy has ingested RBCs inside it, right? So it will form the pseudopodia with the help of which it will move, and it has the ingested RBCs. Okay, excellent. What about this? Naegleria fowleri. It is a free-living amoeba. It is present either in the trophozoite or the cyst stage. So it happens because of swimming in brackish waters, and it leads to the very fatal primary amoebic encephalitis. What is the diagnostic infective stage? Trophozoite. What have you inhaled? You have inhaled the trophozoite, which crosses the cribriform plate and reaches your brain. It will take time. Trophozoite stage is amoeboid. There is the amoeboid stage and there is the flagellate stage. So it exists in 2 stages. Either of them can infect us. Finally, what is the diagnostic stage? It is inside the CSF we can see the trophozoites. Does it also form the cyst? Yes, it also forms the cyst stage. What about acanthamoeba? Acanthamoeba is another brother of Naegleria. Acanthamoeba is another brother of Naegleria. This is a free-living amoeba. This too, Acanthamoeba leads to granulomatous amoebic encephalitis and keratoconjunctivitis. So you remember the swimmers conjunctivitis, contact lens users. There are two important guys, Pseudomonas and Acanthamoeba for the contact lens keratitis. Contact lens keratitis, right? Very, very important. What is the infective stage? This also has the trophozoite. So this iska trophozoite simple amoeboid trophozoite hota hai. There is no flagellate form here. And what is the diagnostic stage? Again, we can see the trophozoite, or in case we have taken the corneal biopsy. In the corneal biopsy, we can see the cyst. And the cyst you can see with the undulating membrane. The undulating double-walled cyst. Yes, double-walled cyst. Excellent. ऑलराइट, कॉन्टैक्ट लेंस इंसेफेलाइटिस, सही बोल रहे हो, बिल्कुल सही बोल रहे हो, है ना. चलो मैच करो जरा. इंटरमीडिएट होस्ट शिस्टोसोम्स. शिस्टोसोम्स के क्या थे? एक ही इंटरमीडिएट होस्ट था ना, स्नेल ओनली, स्नेल ओनली. सो दिस इज 3. एकाइनोकोकस granulosus, dog tapeworm. Human is the intermediate host. Yes, human is the intermediate host. Diphyllobothrium latum, ढक्कन क्या है? Trying very hard to be like a trematode. So this is the only cestode with 2 intermediate hosts. So option 2. Tinea solium क्या है? Human is the definitive host. Yeah, it has no intermediate host. बताओ. Tinea solium, human is the definitive host. Causes intestinal teniasis, neurocysticercosis. H nana. Nana says no. No means no intermediate host. This guy has no intermediate host, even though this is a cestode. So this matches with 4, right? So you remember the intermediate hosts. All cestodes have one intermediate host, except Diphyllobothrium latum and H nana. Diphyllo says 2. Di means 2. So it has 2 intermediate hosts. Nana says no. It has no intermediate host. Trematodes all have 2, except schistosomes. The exception king, snail only. Okay? These are the only things about intermediate host. Very, very quickly I have said it out. I hope you got it. So human as an intermediate host. What is the mnemonic? So in your life you thought that it would have been better if you had gone with your best plan, which was the plan C, to become an engineer. Then you would not have such mind-boggling marathons happening to you. So the best plan in life is the plan C. So you have Babesia microti, the tick-borne bloodstream infection. Who's the best friend of Babesia? Plan से याद करो, Plasmodium. Malaria, human is the intermediate host. Babesiosis, human is the intermediate host. कौन देखा था हमने? Dog, Echinococcus granulosus, dog tapeworm, आज ही मिले हैं इससे, है ना, dog tapeworm. So dog से क्या याद रखें? Cat से क्या याद आया? टॉक्सोप्लाज्मा हो गया cat, और dog और cat दोनों से याद करेंगे क्या? Sarcosistis lindemanae. Okay, this is both dogs and cats. Then finally C is what? C is cysticercosis, which is neurocysticercosis. So the same Tinea solium may, human can be definitive host if we are talking about intestinal teniasis. Human can be the definitive host. But if we are talking about neurocysticercosis, then human becomes the intermediate host in the same Tinea solium guy. Okay, best plan C. All right. Who are these? Very, very easy to identify. These are, of course, microfilarial worms. These are the microfilaria of the lymphatic filariasis causing filarial worms. What can you see? You can see that the cephalic sheath is present. इसमें भी sheath है. Okay, और इसमें भी sheath है. Okay, in all 3 the sheath is present. What can you see here? No nuclei. This guy is bankrupt. Who is he? Wuchereria bancrofti. Leads to Wuchererian filariasis. What does it cause? Wuchererian filariasis. Yes, Wuchereria. Who is the vector? Most important, Culex mosquito, followed by Aedes mosquito, followed by Anopheles mosquito. ये कौन है? 2 nuclei means Brugia malei. इंडिया में कौन है? Brugia malei. Causes Brugian filariasis. Brugian filariasis. More common in India. India में Brugian. Then vector Mansonia species. Mansonia species followed by Anopheles. Anopheles mosquito. Who's the third guy with the sheath and a line of nuclei? Line of nuclei in the tail tip. This is Loa loa. Loa loa. Loa loa. Causes loiasis. Loiasis. African eyeworm. This is spread by the deer fly. Deer fly. Also called as African eyeworm. Alright, let's look at other guy. It has no sheath, tail end, no nuclei. This guy is totally zero. This guy is zero. Onchocerca. Onchocerca volvulus. What does it cause? It causes onchocerciasis. Onchocerciasis. What are the features? We will just see. Onchocerciasis by the black fly. Black fly. How to remember onchocerciasis? Over here you will see this microfilaria not in the peripheral blood smear, but you will see it in the skin sample. In the skin sample you will see the microfilarial worm. Okay. How do you remember? Just remember O. O or 0. So he is 0. So 0 means no nuclei and it means no sheath. No sheath. Yes. 0. 0. Can I also draw it like this? Then it gives me the leopard skin. Leopard skin. Yeah. Can I also draw it like this? This is showing me I can't see anything. So this is river blindness. It causes river blindness. Yes. What else can I do? Can I also, sorry. Can I also, sorry guys. River blindness, leopard skin. Can I also make it like this? Now this is looking like a hanging groin. So it leads to hanging groin sign. Drug of choice is ivermectin. For all others, the drug of choice was DEC. Yes, for all others, drug of choice was DEC. DEC. For this guy, it is ivermectin. Okay, ivermectin. अच्छा, next क्या है? 24-year-old male resident Bihar. Bihar बोलते ही you know what we are talking about. Prolonged low grade fever, progressive weakness, weight loss. स्प्लीनोमेगैली है बहुत ज्यादा, हाइपर पिगमेंटेशन है. व्हाट आर वी टॉकिंग अबाउट? काला आजार, राइट. देन देयर इज नो मलेरियल पैरासाइट. तो हमने क्या लिया? बोन मैरो इज द सैंपल ऑफ चॉइस ऑलवेज. देयर इज मैक्रोफेजेस कंटेनिंग ओवल ऑर्गेनिज्म विद अ न्यूक्लियस एंड अ रॉड शेप्ड काइनेटोप्लास्ट. न्यूक्लियस काइनेटोप्लास्ट. व्हाट आर दीज? LD bodies, Leishman-Donovan bodies. LD bodies shown in which of the following most appropriate confirmatory test for this condition, guys? Aldehyde test, Napier's test, yes, it is correct. Or you could have written Chopra's antimony test also. They are correct, but this is not confirmation. Most appropriate confirmation test. Yes, this is a test performed, but not most appropriate. RK39 rapid immunography. Yes, of course. This you have to know. Rapid kinesing test. Okay, Buffy code smear for Donovan. Donovan is not correct. Yes, if they had written QBC, then QBC is not only performed for malaria, it is, oh yes, B is the correct answer. QBC is also performed for microfilaria. So QBC is the Buffy code. Montenegro. Leishman skin test. This is a delayed type hypersensitivity test. This is to tell me the prognosis of the disease. Prognosis. If this is positive, patient has good CMI, means the prognosis is going to be good. So what rule forms the Donovan bodies? Donovan bodies was in, come on chat box, Donovan bodies was in a genitourinary ulcer disease called as donovanosis with the safety pin appearance producing Haemophilus ducrei. So these are not Donovan bodies, these are LD bodies, Leishman-Donovan bodies. All right, what is the infective stage for Kala Azar? Quickly. Infective stage. Diagnostic stage. NNN, Novi McNeil Nicole culture, which stage? प्रोमास्टिगोट इज द इन्फेक्टिव स्टेज, मच्छर के अंदर इट इज प्रोमास्टिगोट, कल्चर में भी क्या बन जाता है, प्रोमास्टिगोट, प्रो मीन्स आगे, मास्टिगोट मीन्स फ्लैजिला, इसके एंटीरियर एंड पे फ्लैजिला होता है। डायग्नोस्टिक स्टेज क्या है, ए मास्टिगोट, अब कोई मास्टिगोट नहीं चाहिए, कोई फ्लैजिला नहीं चाहिए, क्यों, हम घुस के बैठे हुए हैं हमारे सेल्स के अंदर, मैक्रोफाजेस के अंदर ए मास्टिगोट घुस के बैठा हुआ है। एनएनएन मीडियम में प्रोमास्टिगोट स्टेज, विच अदर मीडियम, श्नाइडर्स ड्रोसोफिला लिक्विड कल्चर मीडियम कैन बी यूज्ड। काला आजार का कॉम्प्लिकेशन ऑफ काला आजार ऑन antimony treatment and sunlight after 20 years person will get post kala azar dermal leishmaniasis. PKDL is a long term complication of kala azar which is visceral leishmaniasis, ठीक है, which is the sandfly phlebotomous. sandfly भी देखेंगे अभी थोड़ी देर में। 32-year-old male worker recently returned from rural Uganda, कहाँ चला गया भाई, अफ्रीका, intermittent fever, severe headache, extreme daytime sleepiness, okay, sleeping sickness, irritable, disoriented, enlarged cervical lymph nodes on examination. क्या है भाई ये पेरिफेरल स्मीयर में दिखे, flagellated with the undulating membrane. Obviously we are talking about trypanosomiasis, ठीक है। trypanosomiasis दो तरह की होती है, अमेरिकन स्लीपिंग सिकनेस, अफ्रीकन स्लीपिंग सिकनेस। इस प्यारे से निमोनिक से हम सब देख लेंगे। यहाँ पे कौन है, टॉम क्रूज को क्या हो गया, प्यार, सो ही स्टार्टेड किसिंग दिस रेड हेयर्ड गर्ल whose name was Romana. This is all, and then he went away in his Mercedes-Benz C-Class. इस निमोनिक में सारा का सारा क्या हमारा कवर हो गया, अमेरिकन स्लीपिंग सिकनेस। कौन करेगा अमेरिकन स्लीपिंग सिकनेस, ट्राइपेनोसोमा क्रूजी। दिस इज caused by the red-haired reduvid bug, also called as kissing bug, also called as triatomine bug. Romana, this disease shows me the Romana's sign, the periorbital edema which you can see in this person. Benzimidazole is your treatment of choice. And C, it causes the skin infestation, the skin manifestation of this is called as Chagas disease. Done. अब अफ्रीकन स्लीपिंग सिकनेस कौन है ये, ब्रूसली, ट्राइपैनोसोमा ब्रूसी। व्हाट डज ब्रूसली ड्राइव, ही ड्राइव्स अ जी वैगन, and what is this, winter bottom sign, so he drives the G wagon in winters. So you have the winter bottom sign. Now there are two types of African sleeping sicknesses. Okay, there are two types of African sleeping sicknesses. There is the West African sleeping sickness and the East African sleeping sickness. How do I remember? Obviously both of them are Trypanosoma brucei. Okay, what was he riding? He was riding the G wagon. So West African is Trypanosoma brucei gambiense. Trypanosoma brucei gambiense. And Trypanosoma brucei rhodonense becomes your East African sleeping sickness. Sunrise in East ऐसे भी याद कर सकते हो आप, है ना। ब्रूसली राइड्स द जी वैगन इन विंटर, ऐसे भी याद कर सकते हो आप। विंटर बॉटम साइन भी होता है इसमें। व्हाट इज दिस स्प्रेड बाय अफ्रीकन स्लीपिंग सिकनेस, सी यू आर सेइंग ना स्लीपिंग सिकनेस, व्हाट आर यू सेइंग, स्लीपिंग सिकनेस, so it is spread by the sessafly or Glossina species, ठीक है। डन, डस्टेड, डन। बताओ रूमेटिक फीवर किससे होगा, गुयान-बारी सिंड्रोम, रीटर्स सिंड्रोम। व्हाट इज रीटर्स सिंड्रोम, कांट पी, दैट इज यूरेथ्राइटिस, कांट सी, conjunctivitis, can't climb a tree, arthritis, right? Multiple sclerosis, mixed cryoglobulinemia, arthritis. HCV क्या होता है, HCV means chronic, HCV means cirrhosis, HCV means hepatocellular carcinoma, and HCV also means cryoglobulinemia. Cryoglobulinemia, right? So mixed cryoglobulinemia is one HCV. GBS, ज्ञान बारे सिंड्रोम, अभी हुआ था महाराष्ट्र में, किससे? क्लेमाइडिया, सॉरी नॉट क्लेमाइडिया, campylobacteriosis. So B becomes 6. B6, Campylo GBS 4C. 4C, you are saying 4C, chlamydia is Reiter syndrome. Excellent. 4 is going to be C. Correct. और बोलते जाओ. D5, multiple sclerosis, EBV. Correct. Multiple sclerosis is EBV. So finally we are left with shawarmas of Mumbai. Yes. B6 हो गया. रीटर सिंड्रोम नहीं बताया किसी ने, यहां बता दिया. 4C हो गया. Rheumatic fever and arthritis is remaining. Rheumatic fever and arthritis. Come on. Group A streptococcus क्या करता है बाद में? What does group A streptococcus cause? Strep pyogenes. It causes rheumatic fever. So A becomes 2. What is left? 1, 2, 3, 4, 5 and 6. 1, 2, 3. 3 is remaining. So Borrelia. Borrelia is causing arthritis. ये Borrelia कैसे arthritis कर रहा है? Borrelia Bergdorferi. BB, Justin Bieber. BB से याद करो Justin Bieber. Justin Bieber को क्या हुआ था? Lyme disease. Lyme disease में क्या होता है? Chronic arthritis. So F is 3. Okay? A2 भी आ गया, F3 नहीं आ रहा था, because Borrelia Burgdorferi, you have to remember. Borrelia is not just recurrentous and relapsing fever, it is also Lyme disease. चलो, diphtheria toxin, what is the mechanism of action? Diphtheria क्या करता है? Pseudomembranous tonsillitis. What is the mechanism of action? It inhibition of elongation factor 2. What is the organism? Corynebacterium diphtheria. What is the disease? Diphtheria. What is the complication of diphtheria? Bull's neck, myocarditis. Yes, bull's neck, myocarditis. How do we detect the toxin? We detect it by the LX gel precipitation reaction. LX gel precipitation. Tetanus toxin causes tetanus. It is a spasmodic toxin. So it inhibits the negative inhibitory gamma glycine. Inhibits the GABA glycine. Clostridium tetani, spore former. What are the complications? Opisthotonus. What is the complication? Rhizus sardonicus. The grin sign, Rhizus sardonicus. And respiratory arrest, and the person can die. What is the mode? So for all others, mode of detection of toxin is PCRs or ELISAs. Simply detect the gene or simply detect the antigen of the toxin. Botulinum toxin, Clostridium botulinum. What is it going to cause? Canned food related. What is it going to inhibit? It is going to inhibit the acetylcholine receptor. It produces a neurotoxin leading to flaccid paralysis. So what is going to happen? Floppy baby syndrome. Why? Because of ingestion of honey. This is used for giving Botox as relaxant of the muscles, leading to vanishing of the honey in newborn. Correct. Vanishing of the wrinkles. So this leads to botulism. Okay. Pseudomonas exotoxin A. Same mechanism of action as that of your diphtheria toxin. Inhibition of elongation factor 2. Ultimately, what will this lead to? This will lead to inhibition of protein synthesis, right? So over here it is spastic paralysis. In tetanus it is spastic paralysis, and in botulinum it is flaccid paralysis, right? What about the organism of Pseudomonas? It is Pseudomonas aeruginosa only. No points for guessing that. What is the disease and complications? Pseudomonas क्या करता है? Malignant otitis externa, है ना? Malignant otitis externa, external ear infection. और क्या करता है? Eyes में contact lens keratitis, biofilms बनाता है ना? Contact lens keratitis, very important, and it makes the biofilms. It can cause a whole array of diseases which it can produce. Okay, next देखो क्या है? Pertussis toxin. Pertussis क्या करेगा? Whooping cough. What will it cause? It will cause the whooping cough, right? कहाँ से ऑर्गेनिज्म क्या है? बॉर्डेटेला पर्ट्यूसिस. Bordetella pertussis. What is the mechanism of action of pertussis toxin, guys? Same as E. coli. Increased camp by cholera toxin. Cholera toxin inhibits the zonula occludens ZOT, and it is also increased camp by binding to the GM1 ganglioside receptors. GM1 ganglioside receptors. Organism कौन है? Vibrio cholerae. Vibrio cholerae, no points for guessing, causes cholera. What is the cause of death in cholera? It is severe dehydration only, not because this is not the invasive disease. Okay, cholera does not cause any invasive disease. There is only toxin which is produced, so because of which there is lots and loss of water and leading to severe dehydration and ultimately shock and shutting down of the organs. Shiga toxin क्या है? Very very similar to the verocytotoxin of E. coli. So enterohemorrhagic E. coli के verocytotoxin की जैसा, हाँ TCBS बिल्कुल सही कह रहे हो. It inhibits the 60S ribosome subunit. 60S ribosome subunit को inhibit करने से क्या होगा? Inhibition of protein synthesis is gone over here also, right? What is the organism? E. coli, Shigella for that, or enterohemorrhagic E. coli for the O157H7 strain that can also produce the Shiga-like toxin, right? What will it cause? Dysentery, blood in stool is caused. Labile toxin, stable toxin, दोनों किसके हैं? E. coli के टॉक्सिन है ना? एंटेरोटॉक्सिजेनिक E. coli के टॉक्सिन हैं दोनों. एंटेरोटॉक्सिजेनिक E. coli के टॉक्सिन है, ठीक है ना? क्या LT क्या करेगा? इंक्रीज्ड कैंप. किससे बाइंड करेगा? GM1 गैंग्लियोसाइड रिसेप्टर्स. ST क्या करेगा? इंक्रीज्ड CGMP. दोनों ही किससे? E. coli. दोनों में क्या? वाटरी डायरिया. ETEC क्या करते हैं? ट्रैवलर्स डायरिया. T फॉर ETEC, T फॉर ट्रैवलर्स डायरिया. No problem. Revised. HUS. Come on, tell me. HUS. HUS is hemolytic uremic syndrome caused by O157H7 E. coli. What is the selective media for this E. coli? Sorbitol McConkie agar. So SMA. Colony morphology कैसी होती है इस पे? Yellow colonies are given on sorbitol McConkie agar by the E. coli O157H7. Hepatorenal syndrome is hepatorenal hemorrhagic syndrome. Wheels disease. कौन है एजेंट? Leptospira. This is another name for leptospirosis. Selective medium क्या है इसका? Ellingson, McAuliffe, Johnson, Harris. EMGH medium. Korthoff's medium, which is the solid agar, and Fletcher's medium. Colony morphology, ring is formed in the liquid medium. Let's look also. What is this? This is the SMA with the E. coli, the yellow colonies. What is this? This is the ring in the EMJH formed by the Leptospira. Let's talk about others first. Water can perineum, Waterhouse Friderichsen syndrome. Depending upon male or female, they are both caused by Neisseria gonorrhoeae or gonococcus. Neisseria gonorrhoeae, gonococcus. What is the medium used? Thayer-Martin medium, or very fancy New York City medium. Excellent. Shiny non-hemolytic colonies. What is Thayer-Martin? Thayer-Martin is nothing. Thayer-Martin is overglorified chocolate agar. So whenever you see this chocolate agar kind of thing with non-hemolytic colonies, you know you are looking at gonococcus right now on Thayer-Martin medium. ये क्या दिख रहा है? कुछ ब्लड अगार है जिसपे ब्लैक कॉलोनीज है. ब्लैक कॉलोनीज ऑन KTBA. व्हाट इज KTBA? पोटेशियम टेल्यूराइड ब्लड अगार, जिसमें पोटेशियम टेल्यूराइड टर्न्स टू टेल्यूरियम, जिसकी वजह से वो ब्लैक हो जाता है. तो सूडोमेम्ब्रेनस टॉन्सिलाइटिस कौन कराता है? कोराइनी बैक्टीरियम डिप्थीरी, modified NY medium. Okay, nice. Very nice. Nice answers coming in. कोराइनी बैक्टीरियम डिप्थीरी क्या है? कोराइनी बैक्टीरियम डिप्थीरी. इसके 3 स्ट्रेन्स होते हैं, माइटिस ग्रेविस बायोटाइप्स एंड इंटरमीडियस, इंटरमीडियस। क्या कौन सा अगार है? KTBA, पोटेशियम टेल्यूराइड ब्लड अगार, जिसपे ब्लैक कलर की कॉलोनीज दिख रही थी। ब्लैक कॉलोनीज कैन बी सीन ऑन KTBA अगार। दूसरा क्या है? टिन्सडेल मीडियम, टिन्सडेल मीडियम। तीसरा क्या है? LSS, लॉफलर सीरम स्लोप, in which you can see the metachromatic granules, on which you can see the metachromatic granules when you do the Albert staining. Yes, Albert staining. Next, जल्दी बताओ। लीजनेयर्स डिजीज, लीजनेला, लीजिए लीजिए लीजनेला। कहां से लीजिए? ठंडे पानी से एंड AC वेंट से। लीजियनेला मतलब वाटर कूलर्स एंड AC वेंट्स। लीजनेला नीमोफिला, लीजिए लीजिए लीजनेला लीजिए, ठीक है। न्यूमोफिला। कौन है इसका सेलेक्टिव मीडिया गाइस? कम ऑन, व्हाट्स द सेलेक्टिव मीडिया? बर्फर्ड चारकोल ईस्ट एक्सट्रैक्ट, BCYE। And what do you see? You see the shiny colonies. Shiny कैसी? Cut glass, like you have cut some glass. Let's look at it. Black color का अगार ये दिख रहा है, मस्त ब्लैक कलर का अगार with the shiny cut glass colonies, BCYE। ब्लैक तो मैम ये भी है, इसके ऊपर मुझे ऐसी लग रही है जैसे मर्क्यूरी ड्रॉप्स लगी हैं। मर्क्यूरी ड्रॉप्स एंड रीगन लोवी, रीगन लोवी मीडियम। ये किस में होता है बताओ जल्दी? वुपिंग कफ, पर्ट्यूसिस। कहाँ गया? वुपिंग कफ। बॉर्डेटेला, बॉर्डेटेला, पर्ट्यूसिस। क्या है अगार? रीगन लोवी। ये भी ब्लैक कलर का है, इसमें दिख रही हैं मर्क्यूरी ड्रॉप्स और टीयर ड्रॉप कॉलोनीज। रो दिया, पढ़ पढ़ के रो दिया है हमने अब तक, राइट। टीयर ड्रॉप कॉलोनीज। अब ये क्या है? कैप, कम्युनिटी एक्वायर्ड निमोनिया। कौन करेगा? एटिपिकल वाले लोग करेंगे, यानी कि माइकोप्लाज्मा निमोनी, माइकोप्लाज्मा निमोनी। इस दुनिया में कोई ही ऐसा होगा जिसको नहीं पता होगा कि माइकोप्लाज्मा निमोनी मुझे PPLO अगार के ऊपर, प्लूरो निमोनिया लाइक ऑर्गेनिज्म अगार के ऊपर फ्राइड एग कॉलोनीज देता है, जो कि माइक्रोस्कोपिक होती हैं, अराउंड 300 माइक्रोमीटर साइज की फिजिकल कॉलोनीज होती हैं। इट्स अ वेरी 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 स्मॉल बैक्टीरिया, राइट। अक्यूट बैक्टीरियल मेनिंजाइटिस 3 इंपॉर्टेंट भाई कर सकते हैं। स्ट्रेप्टोकोकस निमोनी, मैंने हमेशा बोला है मेनिंजाइटिस कहाँ होता है? खोपड़ी में। खोपड़ी का मतलब कैप्सूल, वो बैक्टीरिया जिनके पास कैप्सूल है। स्ट्रेप निमोनी, H इन्फ्लुएंजी और निजीरिया का एक भाई निजीरिया मेनिंजाइटिडिस। सो वाइल निजीरिया गोनोरी हैज नो कैप्सूल, meningitis has a capsule. What is the media I use? स्ट्रेप निमोनी को किस पे उगा लूं मैं? इसको पसंद है चॉकलेट अगार। H इन्फ्लुएंजी को भी चॉकलेट अगार पसंद है, लेवेंथाल मीडियम भी हम इसके लिए यूज कर सकते हैं, ठीक है। और निजेरिया मेनिंजाइटिस के लिए तो तुम्हें पता ही है, चॉकलेट अगार या फिर थेयर मार्टिन मीडियम, ठीक है। एनी स्पेशल टाइप ऑफ कॉलोनीज यू सी? स्ट्रेप निमोनी, चॉकलेट अगार, एनीबडी रिमेंबर्स द ड्राफ्ट्समैन कॉलोनीज और द Craftsman colonies or the carom coin appearance, carom coin appearance. Now verbal diarrhea is incoming. Come on, tell me what are all the things you see in H influenza, satellitism. What do you see in strep pneumoniae? Optochin sensitivity, draftsman colonies, Quellung reaction, inulin fermentation, bile solubility. Pause it and write it down in your notes. Okay, all of this you see in strep pneumoniae. Let's look at these carom coins. Let's look at these carom coins. यहाँ पे तो नहीं है, ये रहा कैरम कॉइन और ड्राफ्ट्समैन कॉलोनीज। कौन है ये? न्यूमोकोकस, स्ट्रेप निमोनी, कैरम कॉइन कॉलोनीज। चलो वापस चलते हैं। ये क्या हो गया? छोड़ो। ऑलराइट, लंपी जॉ सिंड्रोम, लंपी जॉ सिंड्रोम, सन रे अपीयरेंस। हमने पढ़ा था हमारा भाई सूरज, सूरज को हो गया था एक्टिनोमाइकोसिस। एक्टिनोमाइकोसिस इज द डिजीज कॉज्ड बाय द फंगस एक्टिनोमाइसिस, एक्टिनोमाइसिस, एक्टिनोमाइसिस इजराइली, एक्टिनोमाइसिस इजराइली इज मोस्ट इंपॉर्टेंट, राइट। अब एक्टिनोमाइसिस इजराइली के लिए हम यूज क्या करेंगे, ब्लड अगार करेंगे या कैल्शियम सप्लीमेंटेड। क्लासिकली इसमें क्या दिखती है, मोलार टूथ अपीयरेंस ऑफ द कॉलोनीज, मोलार टूथ कॉलोनीज, दैट इज व्हाट वी आर टॉकिंग अबाउट। मोलार टूथ ये दिख रहे हैं बिल्कुल जैसे दांत रखे हैं, मोलार टूथ कॉलोनीज इज सीन इन दिस गाय। देन बार्नवर्थ डिजीज क्या है, लाइम डिजीज का ही अलग नाम है, लाइम डिजीज। लाइम डिजीज किससे, बीबी, बर्गडॉफ बोरेलिया बर्गडॉफेरी, ये तो हमने देखा ही था। और पॉप स्टार लाइक जस्टिन बीबर, क्या होगा मीडियम का नाम, बार्बर स्टोनर, है ना, ऑब्वियसली बार्बर स्टोनर केली मीडियम, ठीक है, क्योंकि पॉप स्टार की डिजीज है लाइम डिजीज, तो बार्बर स्टोनर केली मीडियम ही होना पड़ेगा इसको। वी हैव जस्ट सीन द मॉर्फोलॉजीज ऑफ ऑल ऑफ दीज गाइज ऑल्सो, right? So quickly let's match the following. एरिदीमा इन्फेक्शियोसम, अभी देखा था हमने, एरिदीमा इन्फेक्शियोसम किससे होती थी, पार्वोवायरस B19, ठीक है। एरिदीमा नोडोसम बता दो, चलो बता जल्दी। एरिसिपेलास, एरिसिपेलास तो पता ही होगा, ग्रुप ए स्ट्रेप्टोकोकस, सो ई बिकम्स 1। नाउ बैसिलरी एंजियोमेटोसिस, बार्टोनेला, यस बार्टोनेला इज बैसिलरी एंजियोमेटोसिस। अब ये कौन बताएगा मुझे, एरिदीमा माइग्रेंस एंड एरिदीमा नोडोसम। Nodosum. Nodosum will happen in lepri as a part of the lepra reaction too. So D becomes 3. Erythema migrans is again Borrelia. Borrelia Burgdorferi Lyme disease. Lyme disease में भी तुम्हारी टारगेट साइन जैसा एरिदीमा माइग्रेंस बन रहा था। तो 2 4 5 3 1, very easy. Come on, let's talk about the ticks and the lice and the mites. Very quickly, let's finish it off. Ixodes tick. Derma center is also a tick. Lice and rat flea. I have always tell you, whenever you hear the word lice, you mark whatever has written epidemic in front of it, right? So lice is going to be epidemic typhus, no doubts here. Tick. So we have Borrelia, endemic typhus and anaplasma. Remember this mnemonic of ours. We had the Rickettsial railway station in which we had 2 trains, the typhus train and the spotted fever express. Spotted fever express pe chadha Rocky. Rocky is causing Rocky Mountain spotted fever, right? What was our mnemonic for it? Rick mountain tick. Rick mountain tick, right? So Rick mountain tick means Rocky Mountain spotted fever is tick borne. उसी के साइड में चढ़ा हमारा Indian prince. If he is Indian prince, he is going to be a crown prince. So Konori is crown, right? So Indian tick typhus. Both of these are spotted fever express. Both of them are tick transmitted. ये क्या है, typhus strain. ये चढ़ा soldier. Soldier was pro-war. So a pro-war soldier is Rickettsia provasica. इसके सिर में क्या है, lice. Whenever you hear the word lice, you say epidemic, ठीक है। ये क्या है, एक tough rat. He is a tough rat जिसके ऊपर इतने सारे rat fleas हैं, still he is going home. So tough rat is Rickettsia typhi causing endemic typhus. Life becomes easy for me, right? So endemic typhus is the tough rat with the rat flea. So D becomes 2 here. What about derma center tick? Derma center tick, it is which, Borrelia or anaplasma? Come on, Borrelia Burgdorferi, Lyme disease. हमने क्या पढ़ा था, Justin Bieber was bit by Ixodes tick. So A becomes 1. And finally, derma center tick is anaplasma, Anaplasmataceae family. All right. Who is he? Identify. Who is he? Anopheles or Aedes. Yes, the tiger mosquito, Aedes aegypti. Aedes aegypti with the tiger mosquito. ये कौन है, ये क्यूलेक्स है, क्यूलेक्स। ये कौन है, ये है एनोफिलीज। मैंने बोला हमेशा जापानीज आर क्यूट, क्यूलेक्स मतलब क्यूट, क्यूट मतलब जापानीज इनकेफेलाइटिस। व्हाट विल क्यूलेक्स कॉज, इट विल कॉज जापानीज इनकेफेलाइटिस। अब जापानीज इनकेफेलाइटिस का से मिलता जुलता भाई कौन था, वेस्ट नाइल इंसेफेलाइटिस, है ना, वेस्ट नाइल इंसेफेलाइटिस। और फाइलेरियासिस कौन सा, वुशरीरियन फाइलेरियासिस। एनोफिलिस, एनोफिलिस से क्या होगा, एनोफिलिस से क्या होगा, मलेरिया और फाइलेरियासिस, फाइलेरियासिस, है ना। एडीज इजिप्टाई सबको पता है, डेंगी, डेंगी के भाई फ्लैविविरिडी, येलो वेस्ट किड इज डीजे, जे यहां पे आ गया, वेस्ट नाइल यहां पे आ गया। और कौन, येलो फीवर जिसको हम आने नहीं दे रहे, जीका वायरस और डेंगू का भाई चिकनगुनिया, चिकनगुनिया, ये सब हो जाएंगे एडीज इजिप्टाई से। सो जस्ट समराइजिंग ऑल द मस्कीटोज। अब ये कौन है, खून पीने वाली, फ्लीबोटोमस, जो लोग तुम्हारे ब्लड सैंपल्स ड्रॉ करते हैं लैब्स में, उनको फ्लीबॉटमिस्ट कहा जाता है, क्योंकि वो खून चूसते हैं। सिमिलरली जो सैंड फ्लाई खून चूसती है उसको क्या बोला हमने? फ्लीबॉटोमस अर्जेंटाइपस। फ्लीबॉटोमस अर्जेंटाइपस क्या करती है ये? ओल्ड वर्ल्ड लीशमेनियासिस। क्या? ओल्ड वर्ल्ड लीशमेनियासिस। ओके, एक्सीलेंट। दिस इज अ सैंड फ्लाई, दिस इज अ सैंड फ्लाई। अब एक और बात याद रखेंगे हम, जस्ट रिमेंबर सिल्वर बुलेट लाइंग इन सैंड। ये सैंड है और ये क्या है? ये एक सिल्वर बुलेट है। So silver bullet lying in sand. What is this virus? Chandipura virus. So Chandipura virus is also sand fly transmitted. So sand fly से old world leishmaniasis and Chandipura virus, which is a type of a viral hemorrhagic fever. ठीक है, viral hemorrhagic fever. Chandipura belongs to rhabdoviridae, कोई जरूरत नहीं। ये कौन आया? ये है हमारा hard tick. This is the hard tick. What is hard tick going to cause? Rocky Mountain Spotted Fever, Indian Tick Typhus, yes, and Lyme disease by Ixodic tick. ये कौन है? और टिक से एक और क्या याद रखें? लिखते हैं टिक, के से याद हो गया KFD एंड सी से याद हो गया क्रीमन कॉन्गो हेमोरेजिक फीवर। Right, so these 5 diseases are by the ticks: Crimean Congo hemorrhagic fever and Kaisonur forest disease. बाकी RMSF, ITT और Lyme disease तो एक साथ ही चलते हैं। ये कौन है जिसके मुंह पे ऐसे कटिंग प्लेट्स लगे हुए हैं, सॉफ्ट सा है? दिस इज माइट। माइट जब बच्चा है, बेबी है, वी कॉल इट चिगर, एंड चिगर से होता है चिगरोसिस। चिगरोसिस इज ऑल्सो कॉल्ड एज स्क्रब टाइफस, है ना? स्क्रब टाइफस विच वॉज द ऑक्स के ऑफ द प्रोटियस, व्हील फिलिक्स रिएक्शन, हेट्रोफाइल एंटीजन, है ना? जस्ट वर्बल डायरिया हैपनिंग, इट्स ओके। माइट से और क्या होता है? स्केबीज, स्केबीज विच इज बाय सार्कोप्टिस स्केबियाई, सार्कोप्टिस स्केबियाई। ठीक है, और क्या नेक्स्ट? लाउस। मैंने हमेशा बोला जब भी देखो लाउस हमेशा बोलो क्या? एपिडेमिक। तो एपिडेमिक टाइफस, एपिडेमिक टाइफस हमने देख लिया, रिकेट्सिया प्रोवाजिकाए, द प्रो वॉर सोल्जर। दूसरा क्या? एपिडेमिक रिलैप्सिंग फीवर, बुरीलिया रेकरेंटिस। ठीक है, और ट्रेंच फीवर, ट्रेंचेस जहां पे बहुत ज्यादा ओवर क्राउडिंग होती है, बार्टोनेला क्विंटाना, बार्टोनेला क्विंटाना। रिलैप्सिंग फीवर कौन? बोरीलिया रेकरेंटिस, रेकरेंटिस। ये क्या? रिकेट्सिया प्रोवाजिकाए, प्रोवाजिकाए। इजी इजी। ये कौन है? रैट फ्ली। रैट फ्ली क्या करेगी? रैट से होने वाली प्लेग, ब्यूबोनिक प्लेग, ब्यूबोनिक प्लेग। और क्या करेगी? एंडेमिक टाइफस, द रफ टफ रैट, एंडेमिक टाइफस। ठीक है, ब्यूबोनिक प्लेग किससे? यर्सीनिया पेस्टिस। एंडेमिक टाइफस किससे? रिकेट्सिया टाइफी, टफ रैट। रैट फ्ली का नाम क्या होता है? व्हाट इज द नेम ऑफ द रैट फ्ली? एनीबडी? नोबडी? Xenopsiella chiopis, Xenopsiella चियोपिस। तभी हम चियोपिस इंडेक्स देखते हैं ना प्लेग में, ब्यूबोनिक प्लेग में, राइट? वी फिनिश्ड इट, सक्सेसफुली फिनिश्ड इट विद इन टाइम। ऑल राइट गाइज, थैंक यू सो मच, ऑल द बेस्ट। देर इज plenty which is left, you can check out the last year's marathon as well, but this was enough. Yes, ghee broth, stalactite growth in the ghee broth is Yersinia pestis, chiopis index, plague. Excellent, excellent. I love it when the verbal diarrhea happens with you guys also, है ना? So much is not able to cover, but most important ones which help you recapitulate other things. So that was the purpose of this marathon. As I am writing whatever, you guys are saying more and more answers in the chat box. So all of everything is getting actively recalled by you guys, right? So that was all in this session. Thank you so much for joining. So very soon we are going to have the next faculty and you're going to have the next session. So thank you so much, all the best, and I will see you very soon in the recall and very soon after the exam. It will go very well. Thanks guys.
Hello, everyone. Very good afternoon. I hope you all are having a good day. So, we begin this marathon session with a lovely subject and a very important subject for your INICET, that is microbiology. And as far as INICET is concerned, if you are someone who has already given once this INI exam, then you know that this exam is very previous year question and previous year topic heavy, right? One or two questions they are going to give, which is new research based, but most of the questions are repeat questions. INI people usually have a pattern with them, right? Hi, Dharmesh. Good afternoon. Okay, so as far as pediatrics is concerned, we are not going to waste any further time. As this is a marathon, we are going to literally run through pediatrics and cover most of the commonly asked questions in INICT. And I'm sure if you're well versed with these topics, you're going to score almost 100%. We have to keep a gap for new questions, but most of the topics and the concepts that are tested in this exam are going to be discussed today. So we are going to take this session from 2:30 p.m., that is 2:30 p.m. to 5 p.m. And in this duration, my target, my goal is to let you know, to let you give an idea how to make that last leap of revision as far as pediatrics is concerned. Right? So without further ado, let's begin. So the first question, general pediatrics, we are going to start. Right? We all know growth and development is very important for your all exams, right? Be it INI, be it NEET PG, be it FMGE, and amongst this growth and development, this growth curves, also known as human growth curves, also known as Scammon curves. These are also known as Scammon curves. Very important, right? So I have yes, scam and curves, Dhanush. These curves, I have four curves in front of me. Let's look at each of them. Look at the curve A. In this curve A, I can see that in the initial 2 years of life, the growth is rapid, very rapid. 90% of the growth is happening in this phase only. And after that, there is little to no growth. What is this? This is the curve for neural growth, neural growth. Remember Horlicks की ad में क्या बोलते हैं कि बच्चे के दिमाग का विकास पहले 2 सालों में सबसे तेज होता है, वो सही बोलते हैं। 2 साल में सबसे ज्यादा बच्चे का न्यूरल ग्रोथ होता है, उसके बाद लिटिल टू नो होता है ना। सो दिस इज न्यूरल ग्रोथ कर्व, न्यूरल ग्रोथ कर्व, राइट। नाउ लेट्स लुक एट कर्व बी। कर्व बी में क्या हो रहा है? इनिशियली बहुत रैपिड ग्रोथ हो रही है, फिर उसके बाद अब प्लाट्यू जैसा है, फिर उसके बाद दोबारा से 10 to 12 years ऑफ एज पे दोबारा से रैपिड ग्रोथ हो रही है। ये क्या है? दिस इज ऑल्सो नोन एज सिग्मोइड शेप कर्व, सिग्मोइड शेप और एस शेप कर्व। ये सिग्मोइड शेप्ड और एस शेप्ड कर्व किसके लिए है? मेरी बॉडीली ग्रोथ के लिए है। सो दिस रिप्रेजेंट्स माय सोमैटिक ग्रोथ, सोमैटिक ग्रोथ, right? S for sigmoid, S for S-shaped, S for somatic, right? Then we have C. Look at the C. What is happening over here? Little to no growth in the initial 10 to 12 years, and then there is rapid growth. What is this? जैसे 10 to 12 years हुए, puberty hit की, sexual growth curve. This is sexual growth curve or gonadal growth, you can say, राइट। एंड लास्टली कर्व डी, जिसमें आपको दिख रहा है 4 to 8 years के बीच में एक पीक है। दिस इज फॉर लिम्फॉइड ग्रोथ, दिस इज फॉर लिम्फॉइड ग्रोथ। सो वेरी इंपॉर्टेंट फोर स्कैमन कर्व्स। Yes, Dhanush is right. First is for the neural growth, second is for the somatic, then for the gonads, and then for the lymphoid growth. Dharmesh is also correct, right? So these Scammon curves or human growth curves are very important. Don't forget to revise before the exams. So now let's start with the normal growth pattern. So when we are talking about the normal growth pattern, you should know what is the average birth weight in India, what happens just after birth, how the weight grows thereafter, because once you know the normal things, then only you can tell the abnormal things. So let's start with the normal things first, right? What is the average birth weight in India? Can you tell me what is the average birth weight of a term baby in India? What is that? Can you tell me? What is the average birth weight of a term child in India? It's 3 kgs. And you are right, Dhanush, there is physiological weight loss. Just after birth, the baby does not gain weight. In fact, the baby loses weight. How much the baby loses? The baby loses up to 10% of the body weight, and this happens in the first 7 days of life. This happens in all the babies. That's why it is known as physiological weight loss, right? And this weight that is lost is further regained by day 10. So this is regained by day 10 of life, by day 10 of life, right? Now, how does the weight increases after birth? So let's say the birth weight of the child was X, it doubles. That becomes, it becomes 2x at 5 months of age. Then it becomes 3x at 1 year of age, 4x at 4 years of age. Then it becomes 5x at 3 years of age, 6x at 5 years, and then 7x at 7 years of age. So if someone asks you, when does the weight double? 5 months. When does the weight triples? 1 year. When does the weight quadruples? 2 years, right? So it is a ready reckoner for you. So, and if someone around you also asks if their baby's weight is fine or not, just ask them their birth weight and you can have a rough idea whether they're going towards a good trajectory or not, right? This was about weight. Now let's talk about the length. Average birth weight was 3 kg. What about the average length? Average length of a term baby at birth is 50 centimeter. And how does this grows? Remember, from birth till 1 year of life, there is gain in 25 centimeter. So by the end of 1 year, baby's length would be 75 centimeter. Then again, in the next 1 year, there is gain of 10 to 15 centimeters. So by the end of 2 years, baby's length is going to be 85 to 90 centimeters, right? Right. And by the end of 3 years, what would be the height? What would be the height? It said that after 2 years of age, there is increase in 6 centimeters per year till puberty. After puberty, after that puberty, males can grow up till 10 centimeters per year and females grow up till 8 centimeters per year, right? So by the end of 3 years, what would be the resultant length? It would be around 96 centimeter or 97 centimeters, right? So now I have some questions for you. Weight doubles by, we discussed weight doubles by how many months? Yes, weight doubles by 5 months and triples by 1 year. Similarly, they ask, when does the height double? Remember, the height doubles by 4 years of age and triples by 12 years of age. Another favorite question of INI people, a child is half of the adult height by what time? So what is the average adult height? Average adult height, we take it as 160 centimeters. So half of that would be around 80 centimeter. We discussed that by the end of 2 years, it's 85 to 90. So around 1.5 to 2 years, the child would be half of the adult height. So the good answer over here is going to be 18 months. 18 months to 24 months. The child is going to be half of the adult height, right? Now after weight and length, now let's move to another favorite topic, that is head circumference. So again, what is the average head circumference for term child at birth? It's somewhere around 33 to 35 centimeter. And how does it grows? A very unique pattern. In the first 3 months of life, how does it grows? It grows 2 centimeters per month. 2 centimeters per month. Then in the next 3 months, that is 3 months of the age till 6 months of the age, it's 1 centimeters per month. And thereafter from 6 months to 12 months, it's 0.5 centimeters per month. 0.5 centimeters per month. So let's see how much the head circumference will grow. 2 centimeter per month means 2 into 3, that means 6 centimeter. Right? So in the first 3 months, 6 centimeters gain would be there. Then in the next 3 months, 1 into 3, that is 3 centimeters. And thereafter 0.5 into 6, that is again 3 centimeters. So 6 plus 3 plus 3, that is total 12 centimeter gain would be there in the first year of life, right? If I ask you at birth, what percentage of the head size is that, how much percentage of the head size does the child have when we compare to the adult head circumference? So remember, at birth, baby's head size is actually 60% of the adult head circumference, and it becomes 90% by 2 years of age. 2 years of age, right? Neural growth, remember, by 2 years of age, it's going to be 90% growth happening, right? So by 2 years, this is achieved. Okay, so this is all about the head circumference. Now, a very small topic, periods of growth. Here we are going to discuss some of the entities. In pediatrics, no neonate is similar to an infant. No infant is similar to a toddler, right? So we have different age group and different names for different age groups. We start with antenatal period. That means before the birth of the child, when the child is in the womb of the mother. So when there is fertilization of the ovum and zygote is formed, we label that product of conception as fertilized ovum or zygote till 2 weeks, right? From 0 to 2 weeks, it is called fertilized ovum. Then after 2 weeks, that is 3 weeks to 8 weeks onwards, it's embryo. Then 9 weeks onwards, 9 weeks till birth, it is fetus. So how to remember? Remember E for embryo, E for 8, right? And for fetus, if I make a mirror image of F, it becomes like this, and I join it and become a 9, right? So embryo 8 and fetus 9. Then we have a period, perinatal period, that includes some part of the antenatal period and some part of the postnatal period. So perinatal period starts from 22 weeks of gestation till 7 days of life. 7 days of life, right? So this is perinatal period. Now let's start with the postnatal period, that is just after the birth. So just after the birth, the baby is called a neonate. This neonatal period is from 0 days to 28 days of life. Early neonatal period is the first 7 days, and thereafter we have late neonatal period. Till 1 year of age, this child is going to be called as infant. So if I say every neonate is an infant, that's true. But if I say every infant is a neonate, that's false, right? So in between period of a neonate and the umbrella term of infant is known as post-neonate. So post-neonate starts from 28 days of life till 1 year of age. That's post-neonate. Then comes a toddler. What is a toddler? 1 to 3 years of age. T for 3, T for toddler, right? Then comes preschool, 3 to 6 years. Then school going, 6 to 12 years. Remember, S for school, S for 6. So this is school going child. Now then starts postnatal adolescent phase. This adolescent period is from 10 years to 20 years. 10 to 20. It is further divided into early adolescent, mid adolescent, and late adolescent. Early is 10 to 13, mid is 14 to 16, and then late is 17 to 20 years of age, right? So this is all about the periods of growth. Periods of growth. No doubt till now, right? Dhanush, Dharmesh, both are amazingly answering each and every question. Very good. I hope you're on the right track of the revision. Now let's start with the assessment of growth. So when we talk about assessment of growth, we have certain factors that are dependent on the age and certain factors that are independent on the age. The things that we have discussed till now, weight, length, head circumference, all of these are age dependent. Why they're age dependent? If I say 3 kg of a newborn, that sounds right? But if I say 3 kg of a 1-year-old child, that doesn't sound right? So this weight factor is an age dependent factor. Same goes for length, same goes for head circumference. So the 3 parameters, age dependent parameters that we have discussed are weight, we have length, we have head circumference, right? Now we have certain age independent factors too. What are these age independent factors? We have 2 age independent factors. We have mid upper arm circumference and skin fold thickness. Here age won't be a factor. Age won't be a factor. Now let's understand these. Firstly, we are going to see some of the instruments for the age dependent factors, and then we will move on to age independent factors. So weight we have already discussed. We have 2 instruments over here. See this one instrument, we have certain a scale like thing and a jhola in which the child is lying down. What is this device known as that we use to measure weight? Remember this S-shaped hook over this jhola? This S-shaped hook tell me that this is S spring, right? This is spring balance. So remember, this is spring balance. And then we have this scale where the child will lie down. This is known as beam balance. Beam balance. This can be electronic also. This can be this manual type also, right? Ancient type also, not electronic, non-electronic beam balance also, right? Now let's move to length and height. What is the difference? Look at this image. Here, I'm measuring the length of the baby by making the child lie down using a device whose head end is fixed and foot end is mobile. I will move this foot hand to rest on the foot of the baby and take the length. On the other hand, look at this device. I'm making this girl stand straight and then noticing the height, recording the height. So what is device A? What is device B? By device A, I am measuring the length, and this device is known as infantometer. Infantometer. The name says infant, so you might think that I am going to use this for an infant that is less than 1 year of age, but no, we take length till 2 years of age. 2 years of age. Why 2 years of age? By 1 year the child can stand. I can take the height through the stadiometer, but why I am using this infantometer till 2 years? Because there is physiological bowing of legs. Physiological bowing of legs till 2 years. So if there is physiological bowing and I make the child stand like this, the height recorded would be less than the actual height. Here I'm actually, when I'm making the child lie down, I'm actually straightening the legs and taking the length, right? So remember, till 2 years of age, you're going to take length by using this device that is infantometer. And after that, after 2 years of age, you're going to take height by this device which is known as stadiometer. Stadiometer, right? So with that, now let's move to age-independent parameters like mid-upper arm circumference. So why mid-upper arm circumference is an age-independent parameter? What happens in the age group 6 months to 5 years of age? In this age group, what happens? Initially, the child is very chubby, has a lot of fat. And after that, after 6 months of age, the child now will start moving, crawling here and there. So eventually this fat gets reduced and muscle increases. So if I take the mid upper arm circumference, how do I measure this mid upper arm circumference? I take these two bony points, I take the acromion process above and olecranon process below. I draw a line joining these two, and at that midpoint I'm going to measure the mid upper arm circumference. So initially it is made up of mainly fat, and then fat reduces and muscle increases. So overall circumference will stay somewhat constant in this age group, right? That's why it is an age independent parameter. So 6 months to 5 years, I can employ this mid upper arm circumference. To measure this for community screening of this malnutrition, we use this concept, right? Can you see this colorful tape? What is this colorful tape called? This colorful tape is known as Shakir's tape. We use this for community screening of malnutrition. How do we use this? Can you see in this colorful tape, we have a red zone, we have a yellow zone, we have a green zone. So what is this red zone? This red zone is less than 11.5 centimeters. Then we have yellow zone 11.5 to 12.5 centimeters. And then we have green zone that is above 12.5 centimeters, right? So we have this mid upper arm circumference. So this red zone, which is less than 11.5, denotes that my child is severely malnourished. This tells me that the child has malnutrition, and above 12.5 means the child has normal nutrition. Right, so this is all about mid upper arm circumference. Now let's move to the next age independent parameter, that is skin fold thickness. So again, skin fold thickness is an age dependent thing. In skin fold thickness, we are actually grabbing a part of the skin using this device. What is this device called? This is calipers. Which caliper? Harpenden caliper. So we use this caliper to measure the skin fold thickness. We are actually pinching the skin within this device, right? We can take areas like triceps area, your subscapular area, your interscapular region. In all of these regions, you can make a pinch of the skin and measure the thickness. The most common site that we use is triceps. And apart from that, other sites that we can use include scapular region, infrascapular region, interscapular region, And if it is less than 6 mm, the child has severe malnutrition. And above 10 mm, it is considered as normal nutrition. Normal nutrition. Right? Apart from this completes my age-independent parameters. Now let's move to the next segment, that is puberty and adolescence. So what is puberty and what is adolescence? Adolescence we have just discussed, right? Adolescent is nothing but an age group. Age group 10 to 20 years of age. What is puberty? Puberty means appearance of secondary sexual characters. Appearance of secondary sexual characters. So what the examiner asks here? The examiner asks what is the sequence of puberty? How the sexual changes are going to appear? So remember, in both males and females, the mnemonic to remember the sequence of puberty is TPM. In females, T, thelarche, that is appearance of the breast tissue, thelarche. Then we have P for pubarche, appearance of the pubic hair, and lastly we have menarche, onset of menses. Right? Similarly for males, how the males TPM is said? T is here testicular enlargement, right? So firstly there is testicular enlargement. Then we have penile enlargement, that is penile growth. And pubarche, and thereafter there is manly. What do I mean by manly? That means the child will show all the manly features like beard appearance, deepening of the voice, all of those manly things. Okay, so this was all about the sequence of puberty. Now when we are discussing puberty, a very important grading here is important. What is that important grading? My Tanner staging. Before Tanner staging, let's discuss some of the MCQs that they ask. So first MCQ, they ask what is the biochemical evidence, first biochemical evidence of puberty. Remember, the first biochemical evidence of puberty is GnRH pulse. GnRH pulse. But if they ask what is the best marker of puberty, then you have two options, LH and FSH. Prefer LH over FSH, right? Then comes the staging for assessment of puberty that we are going to discuss. What's that? That is sexual maturity rating or Tanner staging. Sexual maturity rating or Tanner staging. Then they ask, when does the growth spurt happen in females and when does this growth spurt happen in males? So remember, in females, this growth spurt is going to happen earlier. When earlier? In this SMR stage 3. In SMR stage 3, female growth spurt is going to happen. And what about males? In males, it happens in stage 4. Stage 4. Right? Now, one by one, now let's discuss this SMR staging. Firstly, we are going to discuss the male staging. Very easy to remember. Look at this. So remember, stage 1 and 5 is very easy to remember. 1 is going to be a pre-adolescent stage or pre-pubertal stage, and 5 is going to be adult-like. Right? So first is my pre-pubertal appearance. Pre-pubertal in case of male means the testicular volume would be less than 4 ml. Once it reaches the 4 ml, we can say that the secondary sexual characters, the growth has started, growth has begun. So here the testicular volume is going to be less than 4 ml. 4 ml. Right? Then comes stage 2. In stage 2 now there will be increase in the testicular volume. It becomes 4 ml, above 4 ml. And also, can you notice over here, very sparse pubic hairs have started to come. So here you notice sparse pubic hair. Then in stage 3, now the pubic hair, can you see, are coarsening. So there is coarsening and curling of the pubic hair. And also there is increase in the penile length. Thereafter, we all know in stage 4, growth spurt is going to happen and there is increase in the penile girth now. And with staging, you can definitely appreciate that the pubic hair are coarsening, curling. Here in stage 4, everything is like almost like the adult. But the only difference is that now in stage 5, these pubic hair will be present on the medial thigh as well, right? So remember, in stage 5, now the pubic hair would be on the medial thigh. And what is the volume, testicular volume, adult volume? That is 25 ml. So these are the important points, important MCQ points that they ask in the exam. Okay, so stage 1 is going to be prepubertal, testicular volume less than 4. Stage 5 is going to be adult, where the pubic hair are present on the medial thigh as well. Testicular volume is going to be 25 ml. Another question that they ask is, when does scrotal reddening happens and when does scrotal darkening happens? So remember, scrotal reddening happens in stage 2. So in stage 2, you will see scrotal reddening. And what would you see in stage 4? In stage 4, you see scrotal darkening. Scrotal darkening, right? So this is all about the Tanner's changing in males. Now let's go to females. Look at this. Here we are seeing two things. Firstly, the appearance of the breast, and second is the pubic hair. So first and 5 would be similar. First is prepubertal. What about 5? 5 would be adult-like, where you see pubic hair on the medial thigh as well. Now let's talk about stage 2. Can you see in stage 2? Now the breast bud has appeared. So there is appearance of breast bud and very sparse pubic hair. Very sparse pubic hair. In stage 3, we all know growth spurt happens. And again, the pubic hair becomes coarsen and start to curl. In stage 4, everything is like adult, except there is no hair on the medial thigh, not on medial thigh. And in stage 4, there is formation of secondary mount. Secondary mount, right? Secondary mount. So this is all about the Tanner staging in females. We all know growth spurt is going to happen in stage 3. So with that, we finish this segment of growth. Now let's move to a very important section, that is short stature assessment. I told you in the beginning that you need to know the normal things, then only you can tell what is abnormal. So you know what is the normal height, what is the how the height normally grows. So when do you say a child has short stature? We say a child has short stature when the height for age and sex of that child is less than -2 standard deviation or less than third centile. Less than third centile. We all know that the population exists in a bell-shaped curve, right? Some people are going to be short, some people are going to be tall, majority are in the middle. But how do you know that this is a normal variant of the short stature or there is some pathology happening? For that, we calculate this target height. I am short because my parents are short. My genetic potential is such that I am going to stay short. So how do we calculate a target height when a child comes to us? We ask about the mother's height and the father's height. So you calculate this target height, you take the mother's height and the father's height and average it out. If it is a boy child, then you add 6.5 centimeter. If it is a girl child, then you subtract 6.5 centimeter. This is going to give you target height or mid-parental height. This is one of the questions that the examiner is going to throw at you. He is going to give you a scenario of short stature or give you mother's height and father's height and ask you to calculate the target height. Right, so these two important concepts, you should know what is the definition of short stature and what is target height, right? With that, now let's discuss short stature. Firstly, short stature could be either physiological or pathological. Physiological, one thing I have discussed, that I am short because my parents are short. This is familial short stature, that the genetic potential of the child is such that the child is going to be short. So this is familial short stature. Parents will also be short over here. Here the baby's bone growth will collaborate with the actual age of the child. The second entity in the physiological short stature is CDGP. What is CDGP? Constitutional delay in growth and puberty. What do you mean by that? Here what happens, the parents are of normal height but the child is short. When you ask the history of the parents about their puberty, they will say that we were also short in the beginning, but later on in class 9th or 10th, they gained their height and now their final height is within the normal range. So this is nothing but constitution delay in growth and puberty, where you are going to encounter a history of delayed puberty in parents, right? So let's differentiate these two causes of physiological short stature. Firstly, history wise, in constitutional delay in growth and puberty, you are going to have a history of delayed puberty, whereas in familial short stature, you're going to have a history of short stature in parents, short parents. What about the final height? I told you final height in cases of constitutional delay in growth and puberty is of the normal range. This child is not going to be to stay short for long, right? He will eventually catch up the height. But in familial short stature, the final height will also be short. What about puberty over here? The puberty over here is delayed. But on the other hand, in familial short stature, puberty here would be at the normal time. What about the height age? What is height age? Height age means, I told you what is the normal length at the birth. It's nothing but 50 centimeter. At 1 year, it's going to be 75. So 50% of the population, I know average is going to be 75 centimeter at 1 year. So if I randomly tell you that 75 centimeter is the length of the child, what do you think is going to be the age? You will say, ma'am, it is going to be around 1 year of age, right? So height age is nothing but the age at which that particular height is at 50th centile. So here, because the child is short in both of these scenarios, height age is going to be less only. Height age is less. So in both of the scenarios, height age would be less. What about the bone age? Here in constitutional delay in growth and puberty, puberty is delayed, so bone age would also be less. But in familial short stature, bone age is going to be normal. What about the relationship between chronological age, bone age, and height age? See, bone age and height age both are decreased over here. Here, bone age is normal, but height age is less. So compare it to the chronological age. What is chronological age? What is chronological age? Chronological age means the actual age of the child, actual age of the child. So here, bone age is equal to the height age, and both of these are less than chronological age. On the other hand, I know bone age is going to be normal. So here, height age is less than chronological age, but it is equal to the bone age, right? So this is how you're going to make a relationship between these two. Remember, bone age is going to be normal and familial short stature. Right? So with that, we complete the concept of physiological short stature. Now let's move to the disproportionate side of the short stature, pathological short stature. So when we talk about pathological short stature, it could be either proportionate or disproportionate. Proportionate means the child is proportionately small, proportionately short. And disproportionate means either the trunk is short or the limbs are short. For that, we need to assess this ratio. This ratio is very important, upper segment, lower segment ratio. How do we measure this ratio? So we make the child stand like this. Height from the top of the head till pubic symphysis. This is upper segment. And what is lower segment? If you subtract the entire height of this child from the upper segment, the remaining is going to be lower segment. And we take this ratio, upper segment, lower segment ratio. So normal upper segment, lower segment ratio at birth is 1.7 is to 1. 1.7 is to 1. Right? The growth is in the cephalocaudal direction. So initially my upper segment is big and lower segment is short. But as the child grows in the cephalocaudal direction, my lower segment is going to increase. If the lower segment is going to increase, my ratio is going to decrease as I age. Right? So by 3 years of age, it becomes 1.3 is to 1. By 6 years of age, it becomes 1.1 is to 1. By 10 years, it becomes 1 is to 1. And after 10 years, the adult ratio, that is 0.9 is to 1. Or I can also write it as 1 is to 1.1 is achieved. So this normal ratio you should know. If this ratio is not normal, the child has disproportionate short stature. disproportionate short stature, right? So let's see what are the causes of this disproportionate short stature. Firstly, if in a scenario you see that the upper segment lower segment ratio is increased, what do I mean by that? Let's say take an example of a 3-year-old child. I know at 3 years what should be the upper segment lower segment ratio. It should be 1.3:1. If I say this ratio is 1.6:1, that means this ratio is increased for that particular age. That means my upper segment is big, my lower segment is short. What type of dwarfism is this? This is short limb dwarfism. Short limb dwarfism. And whenever I say short limb dwarfism, think of these three conditions. What are these three conditions? First is rickets. Second is your achondroplasia. And third is congenital hypothyroidism. Congenital hypothyroidism. If I wake you up in the sleep and I ask you what are the three causes of the short limb dwarfism or increased upper segment lower segment ratio, you should be able to answer me, right? You have three causes: rickets, achondroplasia, and congenital hypothyroidism. Now let's discuss this decreased upper segment lower segment ratio. Decreased upper segment lower segment means my upper segment is short, lower segment is normal. So short upper segment is mainly my trunk. So this is short trunk dwarfism. And we all know what is our trunk made up of. Our trunk is made up of mainly spine and the ribcage. So any problem with the spine, my trunk's growth is going to be affected. So all the conditions where my spine is affected, for example, if the child has some vertebral anomalies, TB spine, that is spot spine, storage disorders like mucopolysaccharidosis, right, and spondyloepiphyseal dysplasia. In all of these conditions, you are going to appreciate short trunk dwarfism. So with that, we cover this important topic of short stature. Now let's move to development section, that is developmental milestones. So remember, in developmental milestone, what is important? These individual terms are indeed important, but very important is that you remember the sequence, so that when you are in the exam, by just ruling out that this thing happens at this age, this thing happens at this age, so the answer should be in between, you should know that sequence is very important, right? So remember, in these developmental milestones, mug up the sequence first. Sequence is important. Right, sequence is important. So let's start with the gross motor milestones first. What is gross motor? That means my gross bodily movement: sitting, standing, holding neck, running, walking. All of these are my gross motor milestones. So firstly, this gross motor milestone, the very first gross motor milestones is the neck control or the head control. This is achieved by 3 months of age. Then after head comes the trunk. So child now tries to sit. So firstly, the child will be sitting with the support, that is in the tripod position, at 6 months of age. Then the child will be sitting without support at 8 months of age. Right? Then the child will try to stand. So the child will stand with the support at 9 months of age. After standing, now the child would be creeping, crawling. So remember, at 8 months, the child starts crawling, and by 10 months, the child will be creeping. And also at 10 months of age, the child will be holding the furniture and walking. So this is known as cruising. Cruising while holding the furniture. Cruising while holding the furniture. At 12 months, that means at the first birthday of the child will be now standing independently and cutting the cake. That means by the age of 12 months, the child will stand without support. But what about walking? When the child has to receive gifts or return gifts, how will the child go? Will the child walk independently? No. The child will now walk with one hand held. So walking with support will be there. Walking with support. Walking without support will happen at 15 months. So by 15 months, the child will be walking without support, walking without support. And at 18 months, what will happen? When we turn 18, what do? We are adults now. We can run here and there, run away from our homes, right? Similarly, at 18 months of age, the child also runs. So the child will be running at 18 months of age and will be exploring here and there. What will the child will explore? The child will explore the drawers of that home, exploring drawers. By 2 years of age, now the child can jump and climb the furniture, jumping, climb the furniture. By 3 years of age, the child will be driving a 3-tire cycle. What's that? Tricycle. So by 3 years of age, the child will be riding a tricycle. By 4 years of age, the child will be hopping, skipping, but on one foot. Hopping, skipping on one foot. And after that, at 5 years of age, the child will be hopping, skipping on both the feet. Right? Like adults, like we do, skipping on both the feet. Right, so this was about the major gross motor milestones. When we talk about gross motor milestones, one important milestone is stairs milestones, right? Going up the stairs, down the stairs. So what happens initially that when the child goes up and down the stairs, the child will be keeping both the feet on one step. Both feet on one step, then both feet on one step. Similarly, when the child is coming down, both feet on one step. Thereafter, the child learns how to keep one foot on each step and go up and down the stairs like we do, right? So firstly, the child learns to do this upstairs and then learns to do this downstairs. Going up is easier than coming down. So remember, firstly, the child goes upstairs and downstairs, both feet on one step. This happens by 2 years of age. Then the child will learn how to keep 1 foot per step. This first the child will learn to go upstairs because that's easier. That happens by 3 years of age, and thereafter downstairs by 4 years of age. Right, so this was about important gross motor milestone. Now let's understand the fine motor milestones. So when we talk about fine motor milestone, what are these milestones? These milestones are the fine movements of my hand, right? Holding objects, making drawings, doing everything from my hands. These are the fine movements, right? So fine movements, initially what happens? The child learns to grasp toys, rattles. So remember, by 3 months, the child's palmar grasp will begin to lose and the child will try to grasp toys and rattle. And by 4 months, the child will be taking those objects into his mouth. So grasping of toys, rattle, and mouthing will happen around this age group. Then by 5 months of age, the child has a bidextrous grasp. Bidextrous grasp, that means the child will be using both the hands to hold the object. And then by 6 months, the child is going to have a unidextrous grasp. Unidextrous grasp. By 9 months, now the child will have pincer grasp. So what is pincer grasp? Child will be holding the object like this, within the thumb and the other fingers. This is pincer grasp. Initially, this is immature pincer grasp. And by 12 months, it becomes mature when the child will be just holding the object like this, right? Okay sign between the thumb and the index finger. This is the mature pincer grasp. So remember, whenever you see this okay like sign like this is mature pincer grasp. This is going to be immature. This is going to be immature grasp. This is immature grasp. When accessory fingers are used, this is immature grasp. Right? So now the child has learned how to hold this pencil. Mature pencil grasp has come. So by 15 months, the child will now be drawing things from this pencil. By 15 months, the child starts to scribble. Thereafter, the child will draw various things. Firstly, 2 years of age. 2 years of the age, child will join these 2 points and make a line. Right? So by 2 years of age, the child will be able to draw this line. By 3 years of age, the child draws this circle. By 4 years of age, the child will draw this plus sign or cross sign. 4.5 years, the child will draw this rectangle. 5 years of age, the child will draw a triangle. Then 7 years, the child will draw a kite-like or diamond-like structure. 9 years, a cylinder. And then a cube at 11 years of age. Right? So this is all about the various drawings or the fine motor skills that are going to happen. When we talk about fine motor, Then a very important fine motor skill we use every day. What is that? Dressing, undressing, right? Dressing, undressing is a fine motor skill that we use every day. So when does the child learns to dress and undress? Firstly, understand what is easier, dressing or undressing. We all like undressing, right? So undressing is easier. Firstly, the child learns how to undress himself and then dress himself. Same goes for buttoning. Firstly, the child learns how to unbutton and then button. Okay, so firstly the child would be undressing with help, then sooner or later the child will learn how to undress and dress without help, right? So remember, firstly comes undressing with help. This undressing with help comes at 2 years of age. Then comes dressing with help, 3 years of age. Then the child will undress without help at 5 years of age. Then comes buttoning, unbuttoning. Firstly, the child will learn unbuttoning at 3 years of age and then buttoning at 4 years of age. Right? So this is all about the dressing skills. With that, we complete the fine motor milestones. Now let's move to the social milestones. So what are social milestones? How the child interact with others, right? First interaction is with the mother, of course, then with the family members, then the child goes outside, the child is going to play with other peers. So how is this relationship going to happen? So firstly, the very first social milestone that the child gives a social smile. So the child will look around and smile, right? Smile in response to interaction, a social interaction. So the social smile, the first smile that parents notice is at 2 months of age. Then by 3 months, the child realizes that my mother is really important, right? She feeds me, she cares for me. So this 3 months of age, the child will develop a special mother regard. Mother regard, the child will appreciate the mother. Then comes 6 months where the child has mirror play. Now, what happens in mirror play? The child looks himself in the mirror and appreciate himself, right? So, firstly, appreciating the mother, then appreciating himself mirror play. So now the child knows who is my mother and who am I, right? So both the things the child appreciates. So after the 6 months, the child will now develop a stranger anxiety. Child will develop stranger anxiety. Right, child knows now who is my mother, who am I, anyone else who comes from outside is a stranger, and the child has stranger anxiety. Right, and by 9 months a very important milestone happens. This milestone is object permanence. Object permanence. What is object permanence? So before 9 months, what happens? If you show a toy to the child will play with it. But if you hide it somewhere, the child won't look for it, right? Because whatever is in the vision is only the universe of this child. The child does not have any object permanence. But once this 9-month milestone has happened, then the child realizes if it is not in my vision, does not mean it is not in my universe. It is still in my universe. The child will look for this object here and there, right? So this object permanence will happen. And when this object permanence happens, the child needs to wave bye-bye to some things, right? So here the child learns to wave bye-bye. Bye-bye. Right? If you're in a long distance relationship, this milestone is a very sad milestone, right? You know that your partner is still in the universe but not in front of you, and you unfortunately have to say bye-bye every time they leave, right? So remember, this 9-month milestone is a very important milestone, object permanence. After 12 months, after this 9 months, now all the drama is going to happen. All the drama, I mean, acting will happen. So by 12 months, now the child will be acting. That means the child will do imitation, mimicry, all of these things is going to happen. So by 12 months, the child is going to act, imitate, right? By 18 months, the child will be doing domestic mimicry. That means the child will notice day-to-day functioning that is happening. Maybe your maid or you sweeping the floor, the child will mimic that. Cooking food, the child will mimic that. So domestic mimicry happens at 18 months. By 3 years of age, the child will now be able to tell 3 things. What are the 3 things that the child will be able to tell you? His age, sex, and name. Age, sex, and name. Right? And also by this 3 years, handedness develops. That means the child will be preferentially using one hand. That means either right hand or left hand, right-handed or left-handed. And by 4 years, now the child develops right to left discrimination. Right to left discrimination. That means by the end of 4 years, the child will be able to tell me, this is my right shoe, this is my left shoe, this is right, this is left, right? So this is all about the important social milestones. Important social milestones are also this interaction thing that the child will do now outside. Right, so we discussed that the child developed stranger anxiety around 7 months. What happens by 15 months? 15 months, the child appreciates parents a lot. Right? Firstly, the child will hug the parents. By 18 months, the child will now kiss the parents. Right? You're hugging, kissing the parents, developing such a cute bond, right? So now, if the parent is going outside for work, the child will develop intense separation anxiety. So remember, by 18 months, there is going to be separation anxiety. Now, this child cannot stay anxious for long, has to go outside and make new friends. So, by 2 to 2.5 years of age, the child will go out and do parallel play. What is this parallel play? Remember your first day of MBBS, what was happening? You were all sitting in the lecture theatre or maybe dissection hall, and your friend was sitting next to you. You didn't know that this is going to be your friend, and you both were doing your own thing initially, very hesitant to talk to each other, right? So, maybe you were on your phone or you were looking at your notes or your books, and he was looking at his own books and notes. So you both were sitting together but doing things separately, right? So this is parallel play. So two children will be sitting together but they will be playing with themselves only. So this is parallel play. And thereafter, as they grow, by end of 3 years, they are now going to share thoughts, share toys and play together. This is known as cooperative play. So by 3 years, they are going to have cooperative play. And by 4 years they will have a whole gang and do gang bang. They will do role play, right? Role play, ghar ghar jo khelte na, so group play, role play they are going to do. So this was all about interaction and playing. With that, now let's move to the last category of the milestones, that is language milestone, where the child will use his vocal abilities to connect, to express himself. So first milestone that we notice is at 2 months of age, the child will produce cooing sounds. So cooing would be there. By 4 months, the child will be laughing out loud. By 6 months, now the child will speak out monosyllables like ma, ba, da. And by 9 months, the child will join these monosyllables and make mama, papa, dada, right? So bisyllables. By 1 year of age, now the child will say his first real word. What do I mean by first real word? So if you ask your mother or your father, what was my first word, mama, papa, they will tell you either it was mama, papa, dada, anything like that, right? But this is not your first real word. You were just joining two monosyllables and making mama, papa, dada sounds. First real word means the word that the child uses and knows the meaning of. That comes at first year of life. That means 12 years. By 15 months, now the vocabulary is of 4 to 6 words. By 18 months, it becomes 8 to 10 words. By the end of 2 years, the child will be using 2 word sentences like give food, give water, pani do, khana do, like 2 word sentences, bahar chalo, like this, right? So by the end of 2 years, remember 2 word sentences the child would be using, 2 word sentences. And when we talk about the vocabulary would be around 50 to 100 words. And now the vocabulary would increase and increase. By 3 years, the child would now be using pronouns like मैं, तुम, आप, I, me, you, all of these pronouns the child will be using. I, me, you. And by 5 years, by 4 years, what is going to happen? 4 years, remember the child will be telling you stories, poems. And how does you start the story? Once upon a time. That means the child knows that past tense is something, right? So child will also be able to tell you about past tense. That means द चाइल्ड विल टेल कि कल ये हुआ था, वो सारी चीज़ें वो बता पाएंगे. एंड देन कम्स 5 years of age. इन 5 years द चाइल्ड विल नाउ विल टेलिंग यू फ्यूचर टेंस कि हम कल जाएंगे, कल करेंगे. एंड विल ऑल्सो आस्क मीनिंग ऑफ वर्ड्स, मीनिंग ऑफ वर्ड्स. Will also ask meaning of words. So with that, we cover the 4 major domains: gross motor, fine motor, social and language milestone. Now let's talk about the special milestone, that is vision milestone. In this vision milestone, what we are checking? We are checking the visual field of this child, tracking movement of the child. So what do? We take a ring, a red dangling ring, and we keep this red dangling ring 8 to 10 inches away from the child. Right? A newborn can follow this till 45 degrees. 45 degrees, a newborn can follow it. By 1 month, this child can follow up till 90 degree. And by the end of 3 months, the child will be following it 180 degree. Right? Then the examiner asks, when does the binocular vision develop? So this binocular vision is established by 4 months of age. 4 months of age. With that, we cover very important segment of growth and development. Now let's move to the next segment, that is micronutrients in health. So I know this is an overlap topic. You must have studied this in biochemistry also, but here we need it a special mention because yet again a very important topic for your INICET exam. So in micronutrients, let's start with vitamins. So we all know what are vitamins? What are vitamins? Vitamins are micronutrients. That means we need little amount of these in our diet. And if we don't get these micronutrients, a variety of deficiency syndromes happen. So we have 2 types of vitamins, water soluble and fat soluble. What are the water soluble vitamins? We all know B and C, B complex and vitamin C. And what are the fat soluble vitamins? Vitamin A, vitamin D, vitamin E, vitamin K. Right, so one by one we'll discuss each of these and whatever deficiency these vitamins causes. So firstly, let's start with water soluble. We are covering the B complex vitamins first. So in B complex, the first vitamin that we are going to discuss is B1. The other name of B1 we all know is thiamine. What does thiamine deficiency causes? It causes beriberi. It could be dry beriberi or wet beriberi. Dry beriberi, as the name suggests, it affects my nerves, and wet beriberi is going to affect my heart, cardiac tissue. So the child is going to have cardiac failure. And in alcoholics specifically, this vitamin deficiency is noted. And the syndrome, the constellation of symptoms in those alcoholics because of this thiamine deficiency is termed as Wernicke-Korsakoff syndrome. And the mnemonic to remember the triad that is seen in this Wernicke-Korsakoff syndrome is GOA. What is G? G is for global confusion, ophthalmoplasia, and ataxia. So this is Wernicke-Korsakoff syndrome. Now let's move to B2. What is the other name of B2? It's riboflavin, and ribo means red. Remember ribo, red, right? So what happens in riboflavin deficiency? You will see red vessels coming out. So can you see this cornea over here? There is this neovascularization, blood vessels are coming into the cornea. So this is neovascularization of the cornea. Also you see reddening around this angle of mouth, angular ciliitis, glossitis, magenta colored tongue. These all are features that you're going to appreciate in cases of riboflavin deficiency. So remember these markers: magenta colored tongue, neovascularization of cornea. After that, we have B3. B3, other name we all know is niacin. What does niacin deficiency causes? It causes pellagra, right? Pellagra, 3D or 4D diseases. So this pellagra, what is this 3D? 3D, what is this 3D? Diarrhea, dementia, and dermatitis, right? So diarrhea, dementia, and dermatitis. Sometimes a 4th D, that is death, is also included. This is a typical sign that you see in this niacin deficiency or pellagra. What can you notice over here? Can you notice this hyperpigmentation in the skin exposed areas? What is this? This is known as Casal necklace. Casal necklace. This you see in cases of pellagra, right? niacin deficiency. After B3, there's nothing like B4. We have B5. What is the other name of B5? The other name of B5 is pantothenic acid. Pantothenic acid, right? And how do you wear your pants? Through your foot, right? So pantothenic acid deficiency causes burning foot syndrome. Burning foot syndrome. As the name suggests, burning foot, you see burning sensations in the foot. So that's why it is also known as nutritional melalgia. Nutritional melalgia. Then we have B6. B6, the name is pyridoxine. And what does the pyridoxine deficiency causes? The pyridoxine deficiency causes anemia, as well as a very important cause of neonatal seizures. Neonatal seizures, remember, though it is rare, but when you are seeing the algorithm of neonatal seizures, you have tried first line drugs, second line drugs, third line drugs, and the child is still not responding. Then we give this trial of pyridoxine, because this pyridoxine deficiency can also cause neonatal seizures. Then we have B7. What is the name of B7? Biotin. And what does this biotin deficiency causes? It causes alopecia, dermatitis, right? If you are a skincare, hair care enthusiast, especially the girls out there, then biotin supplements is something which is very well advertised, right? So remember, this biotin deficiency causes alopecia, dermatitis. That's why the skincare and the hair care enthusiasts take this biotin supplements, right? After B7, we don't have B8, we have B9. What is B9? B9 is folic acid. Folic acid. Folic acid. Right? Folic acid. So what does this folic acid deficiency causes? Firstly, remember, all the pregnant ladies, they should be taking their folic acid supplementation. Why? Because this folic acid deficiency is very well known to cause neural tube defects in the fetus. So first is neural tube defects, and after birth, megaloblastic anemia. Megaloblastic anemia. Then we have B12, that is cobalamin, and we all know it again causes megaloblastic anemia. What else B12 deficiency is known to cause? SACD. What is SACD? Subacute combined degeneration. Subacute combined degeneration. Right? So this is all about the B complex vitamins and the deficiency that they cause. Now let's talk about vitamin C. Vitamin C. What is vitamin C? Ascorbic acid. And what is the disease that you see with the deficiency of vitamin C? It's scurvy. Scurvy. Right? It's scurvy. So why the scurvy is happening? Vitamin C is less. Vitamin C is deficient in the diet. Vitamin C is very important in the collagen synthesis step. Very important in the collagen synthesis step. It is important for the hydroxylation of proline and lysine, right? So proline and lysine, very important for the hydroxylation. And when this vitamin C would be deficient, my collagen synthesis would be affected. So this collagen is everywhere, right? It is a connective tissue in my blood vessels, in my joints, in my skin. So what is going to happen? There's going to be bleed because of the fragile blood vessels. So gum bleeding would be seen. In skin, you will see the corkscrew hair and the perifollicular redness. In joints, you see this classical subperiosteal hemorrhage and classical signs. This is the x-ray that the examiner is going to throw at you. Look at this x-ray. What are the signs that you can appreciate? This is the periosteum. Beneath the periosteum, can you see this area, black area? This is nothing but bleed. So there is subperiosteal hemorrhage. Look at the joint. The epiphysis is ring-like, right? Circular. This is known as ring epiphysis or Wimberger's ring. Then you have this line is known as provisional line of calcification or white line of Frankel. Just beneath this white line of Frankel, there is a black zone. This is known as scorbutic zone or Tremorfeld zone. And at the edge of this white line, these spurs are known as Pelton's spur. So what all signs are you appreciating over here? Subperiosteal hemorrhage, epiphyseal ring or ring epiphysis, Wimberger ring, white line of Frankel, Pelton's spur, and scorbutic zone or Tremorfeld zone. And look at the cortex. The cortex is so thin. The bone cortex is very thin. This is known as pencil thin cortex. Pencil thin cortex. Right? So all of these signs you appreciate in cases of scurvy. Now with that, now let's finally move to the fat soluble vitamins. And fat soluble, firstly, let's talk about vitamin A. So what does this vitamin A deficiency causes? This vitamin A deficiency causes night blindness, right? And skin, this is appreciated as dryness of the skin, phrinoderma, toad-like skin. And in eyes, you see night blindness. They ask what is the first symptom and what is the first sign that you appreciate. Remember, the symptom is something that the patient presents to you with, and sign is something that you pick. So what is the first symptom? What is the first complaint from the patient end? It is going to be night blindness, that I am not able to see at night, night blindness. And what is the sign that you are going to pick? The first sign that you are going to pick is the dryness of the conjunctiva, right? So conjunctival xerosis is the first sign. Now let's look at the classification. Firstly, we have XN, that is the night blindness. We have X1. In X1, we have X1A and B. X1A is your conjunctival xerosis. B is appearance of this bite-dot spot. Can you look at this picture? Can you see this heap of epithelial tissue over here? What is this? Bite-dot spot. Then we have X2. In X2, now cornea will begin to dry. So there is corneal xerosis. Then we have X3. In X3 now, this dried cornea will ulcerate. So you will have corneal ulceration. If this corneal ulceration involves less than 1/3 of the surface area, then it is X3A. And in X3B, it will be more than 1/3 of the surface area, right? Then we have XS and XF. XS denotes scarring and F indicates your fundal changes, So this WHO classification is very important. This you must have studied in opthal also, right? So very important, staging WHO classification of vitamin A deficiency. And what is the first symptom? What is the first sign? This image of bite-dot spot. Right? With that, now let's move to the next fat soluble vitamin, that is vitamin D. And we all know, what does this vitamin deficiency in growing bones cause? It causes rickets. And what about the grown bones like you and me? If we have vitamin D deficiency, what is going to happen? Osteomalacia is going to happen, right? In rickets, they ask the clinical features. What is the first sign that you appreciate in vitamin D deficiency or rickets? The first sign is craniotabes. Right? Ping pong skull, very soft skull, ping pong skull. Then comes the chest. You see a pigeon-shaped chest, right? Pectus carinatum, pigeon-shaped chest. Also, you see beading. What is that? Rachitic rosary. Painless round beading around the costochondral junctions. This is rachitic rosary. In lower limbs, you see bowing of legs. If you see that the knees touch each other, this is known as genu val gum. Remember, gum means something that is sticking, right? So if the knees are sticking, knees are joining in the middle, this is valgum. And when you see that the knees are falling apart, when you drink a lot of rum, you lie down on the floor or the bed like that, right? So if the knees are apart, this is genu varum, right? So remember, rum, knees apart, gum, knees together, right? And what if both knees go in one direction, either left or right? It looks like as if my knees are swept by wind, right? So this is known as wind-swept deformity. You can see any one of the following in the lower limbs. Right? Bowing of legs, it could be genu valgum, genu varum, or wind-swept deformity. Right? Knock knees. Now let's look at the X-ray feature. We discussed about the X-ray in cases of scurvy. Now let's look at the X-ray features in cases of rickets. Look at the bone right here. Can you see the hollowing, the cupping? This is known as cupping. And also there is widening around the joint area, which is known as splaying. And if you closely look at the end, there is fraying, right? So these three features, cupping, splaying, fraying. And also you can see the bones don't look that white, right? They look very hollow. This is decrease in the density of the bone. Right? So these are the features, X-ray features of rickets. Now let's look at the lab investigations that you will see over here. Right, so what is happening in rickets? My vitamin D is deficient. So why vitamin D is important for us? Because vitamin D is the only thing that helps me in the absorption of calcium and phosphate and helps in this deposition of calcium and phosphate into my bones. So it is helping in the mineralization of bones. So what will happen if vitamin D is deficient? What will happen to my calcium? My calcium would be low, my phosphate will be low. And what happens when the calcium is low in my body? Then there are small glands around my thyroid, parathyroid glands. These parathyroid gland sense the low calcium by calcium sensing receptors and secrete parathyroid hormone. So in this case, parathyroid hormone will increase, right? Parathyroid hormone, what does this parathyroid hormone will do? This parathyroid hormone will try to increase the calcium in my body. How? By causing resorption from the bones. So this ALP would increase and there will be resorption of this calcium levels. So what are the lab investigation picture? Calcium could be low or normal, phosphate would be less, parathyroid hormone will increase, ALP will increase. They ask what is the most characteristic feature out of this. So remember, increase in the ALP is a very important marker over here. Right, now let's talk about the treatment, how you're going to treat this rickets. There are two regimens, high dose regimen and low dose regimen. Which one do you follow? You prefer low dose regimen, right? What is the high dose regimen? In high dose regimen, we give 60,000 international units per week for 10 weeks. So total 6 lakh international units, right? So high doses 60,000 international units per week. So we take either vitamin D shots or capsules of 60,000 IU and we take that for 10 weeks. So we say that every Sunday, we tell the parent every Sunday you give this one shot of vitamin D or one capsule of vitamin D and give that for 10 weeks. So total dose is going to be 6 lakh IU. And what about low dose regimen? Low dose regimen, every day you give vitamin D. 6,000 IU, every day we give this vitamin D. And this we give for 3 months. After completion of this regimen, we check the response to the treatment. What is the first response to the treatment? Response to the treatment would be appearance of white line of Frankel at the joint. White line of Frankel at the joint. This denotes that calcification has started, right? Deposition of calcium has started. So white line of Frankel is the first response to the treatment. With that, we cover vitamin D, rickets. Now let's move to vitamin E, tocopherol, right? So this vitamin E deficiency, what does this vitamin E deficiency causes? With the deficiency of vitamin E, you are going to see some of the hematological features, some of the neurological features. So what is the hematological feature that you notice in vitamin E deficiency? Hemolytic anemias, right? So here you're going to appreciate hemolytic anemia. And what about the neurological feature? Spinocerebellar ataxia. Spinocerebellar ataxia. Spinocerebellar ataxia. Right, then comes vitamin K. Vitamin K, very important vitamin, also one of the vitamins that is deficient in breast milk. So if we do not give this vitamin K injection at birth, what the neonate is going to have? The neonate is going to have hemorrhagic disease of newborn, right? Bleeding tendencies would be there. With that, we complete important vitamins and their deficiencies. Now let's move to the very important topic of nutrition. Before going to nutrition, you should know what is malnutrition. Whenever we say malnutrition, it includes both the terms, undernutrition as well as overnutrition, right? But in India, overnutrition is still not a big problem. Undernutrition is. So whenever we are talking about malnutrition, think undernutrition only, right? So let's one by one look at these parameters and interpret the nutritional status of the child. Simple thumb rule is anything beyond plus minus 2 standard deviation is going to be abnormal. And anything beyond plus minus 3 standard deviation is going to be severely abnormal. Right? So for example, we studied short stature. Short stature means stunting. So if the child's height for the particular age is less than -2, then it is stunting. But when it becomes less than -3, it becomes severe stunting. Right? So one by one, let's look into the parameters. Firstly, look at the first parameter, weight for height -2 to -3. That means this child's weight for that particular height, for his height, is abnormal. This is wasting, that the muscle or the weight is less for the particular height, wasting. This is going to be wasting or severe wasting. In between -2 to -3, this is wasting. If it is less than -3, it is going to be severe wasting. On the opposite end, if the child's weight is higher for the height, +2 to +3, this is going to be overweight child, overweight. And after +3, it is obese. Same goes for height for age. In between -2 to -3, this is stunting. When it is less than -3, it is going to be severe stunting. Right? So this is how you interpret these different criteria and tell the nutritional status of this child. Right? Now let's move to a very important concept of acute and chronic malnutrition. So what is acute malnutrition? So imagine a child, he got sick and had stopped eating food, right? So for a good whole week, the child did not eat much. So the child obviously is going to lose some weight during this course. But once he starts eating, he will be back to normal. During this phase, the child's weight dropped down. But what do you think? What would have happened to his height? Would his height be also affected from the 7 days of illness? No. Right? So this is what is acute malnutrition. The child's weight will drop, but height will stay normal. On the other hand, in chronic malnutrition, what happens? If the child is depleted of food for a long period of time, right? Maybe due to a prolonged illness, maybe due to some poverty issues, socioeconomic issues. If the child is deprived of nutrition, what will happen? Now, not only the weight but also the height would be affected. So remember, what will happen to the weight for age in acute malnutrition? It will be less. What about height? It will be normal. If you take the ratio weight for height, weight is decreased, height is normal. So weight for height will be decreased. Now what about chronic malnutrition? Weight for age obviously would be less. Height for age again would be less. But what about the ratio? Weight is also decreased, height is also decreased. So weight for height may be normal over here, may be normal over here. So with this table comes 3 important MCQs. Which of these factor is common to both acute and chronic malnutrition? You can appreciate which factor is common. Weight for age is common. Then they ask which factor is the best marker for acute malnutrition. Weight for height is the best marker for acute malnutrition. Then they ask which marker is better for chronic malnutrition. It is the height for age, right? So remember these 3 MCQs. Common to both acute and chronic malnutrition is weight for age. Best marker for acute is weight for height, and best marker for chronic malnutrition is height for age. Height for age, right? With that, now let's move to the severe forms of malnutrition, right? This is the extreme forms or severe forms of malnutrition. Look at this child A, right? Very cachectic child, skin and bones baby, skin and bones baby. Very cachectic looking. It looks like as if this child has no fat. On the other hand, this child B, lying down listlessly, very lethargic, looks very chubby. This is known as fat sugar baby. Both of these are chronically malnourished children. Look at the child A. He's very alert looking. If I offer food to this child, the child will very eagerly eat all that food. If a normal child of his age group eats 1 chapati, he is going to eat 4 chapatis, 5 chapatis, very voracious appetite. On the other hand, this child B, you can see he is lying down listlessly on the bed. If I offer food to this child won't have food, his appetite is lost. What do you think out of these is going to have a better prognosis? Obviously, a child is going to have better prognosis. He has preserved appetite. He can eat food and gain muscle, gain fat. But on the other hand, child B is not having food. His appetite is lost. He has swelled up, right? So poor prognosis in cases of child B. Child A is my marasmus child who has very well adapted to this malnutrition. On the other hand, B is your kwashiorkor child, which is maladapted to the situation. Right? With that, now let's differentiate both of these. Firstly, marasmus. In marasmus, you saw how was the baby? Very cacexy looking. So wasting is appreciable, right? Wasting is very much visible. You have visible severe wasting. And what about the kwashiorkor baby? The kwashiorkor baby was also wasted, but because of the edema, that wasting was masked. So here, the wasting is masked. What about the general appearance and the status? So, child A was very alert, sitting, है ना? So here, the general status and appearance, the child would be very alert looking. And how the appearance is? Skin and bones baby. On the other hand, kwashiorkor baby was very lethargic, very dull, fat sugar baby. Marasmus child had good appetite, in fact, voracious appetite. But the kwashiorkor baby has lost appetite, very low appetite. Age of onset marasmus can happen anytime, but kwashiorkor usually happens after 1 year of age when suddenly the child consumes carb-rich and very low protein diets. Right? Prognosis here is going to be good. Here the prognosis is going to be poor. Right? Then we have certain skin and hair changes that we can see in both, but they are more pronounced in cases of kwashiorkor. What are these skin and hair changes? We have flaky paint dermatosis, where there is peeling of skin as if the paint is flaking off the wall, chipping off the wall. Flaking paint dermatosis. You see flag sign in the hair, hypopigmentation and hyperpigmentation. And lastly, what do you see? You see crazy pavement appearance of the skin. Crazy pavement appearance. Crazy pavement appearance. You can see the skin and hair changes in both. Remember, here they are going to be more. Kwashiorkor, they are going to be more. And lastly, liver. What about liver? You see fatty liver in cases of kwashiorkor and shrunken liver in cases of marasmus. Right, so this was about your extreme forms of malnutrition, marasmus and kwashiorkor. Now let's move to a very important topic, SAM. Definition of the SAM is something that you cannot miss out on. So what is the definition of the criteria for SAM? Remember, anyone, you need anyone, I need anyone. Only one thing to label it as SAM. First, weight for height less than -3 standard deviation. That is, there should be severe wasting. Severe wasting. Second is MUAC. Obviously, you will apply this MUAC in 6 months to 5 years of age. MUAC less than 11.5 centimeter. And lastly, there will be nutritional edema. Nutritional edema. Right? So this is SAM, definition or criteria of the SAM. With that, we cover the general pediatrics module. Now let's move to genetics. So in genetics, we are going to discuss some of the important genetic syndromes that the examiners are fond of asking. One of these is the most common encountered chromosomal anomaly, that is Down syndrome. So we all know what is Down syndrome. Down syndrome is trisomy 21. We have one additional 21st chromosome. What are the classical features of this Down syndrome? This picture tell you. What is this baby? What is this baby? This baby has typical down faces. What do I mean by down faces? Down faces are essentially flat faces, as if someone has flattened out the face, right? And also look at the eyes. The eyes are slanting like this. Eyes are slanting like this. That is, the outer canthi of the eye is going up as compared to the inner canthi. This is known as mongoloid slant. Mongoloid slant. Also, you have an eye fold. This is known as an epicanthal fold. Nose is flattened, mouth is open, protruding tongue is there. Also, you can see thickened, redundant skin over the back of the neck, very small ear, flat facial profile. All of these are features of Down syndrome. Also look at this hand. Can you see a single horizontal crease? This is known as simian crease. Similarly, if you look at the foot of this child, what do you appreciate? That the great toe is separated like this from other toes, as if there has to be a sandal in between. So this is known as sandal gap. This is sandal gap. Right? So these are some of the phenotypic things that you are going to notice in Down syndrome. What about other features? The cardiovascular features, neurological features. These Down syndrome patients are prone to some cardiac anomalies, some of the renal anomalies, some of the gut anomalies. So let us look at these anomalies system-wise, right? So there are certain associations. When we talk about the cardiovascular system, then the most commonly encounter congenital heart defects here is going to be endocardial cushion defects. Endocardial cushion defects. In GIT, you have annular pancreas, duodenal atresia, annular pancreas. Hematological, they are more prone to leukemias. What type of leukemias? ALL more than AML. And amongst AML, it is the M7 that you appreciate. Endocrine, they have congenital hypothyroidism predisposition. In skeleton, there are chances of atlanto axial dislocations. That's why screening by neck x-ray is very important, especially after the age of 4 years. Atlanto axial dislocations. And when we talk about neurological problems, then most of these children are mentally retarded, right? Their mental retardation is actually universal in cases of Down syndrome. Okay, so you can see how many problems are there associated with Down syndrome. So it is very important that you do antenatal screening and get an informed decision from the parents whether they want to continue this pregnancy or not, right? So it's very important that you go for antenatal screening. Some screening we do in the first trimester, some screening we do in the second trimester. We are going to look at the first trimester screening. In first trimester screening, you can do some blood test and also you can go for ultrasound. In ultrasound, what do I look for? In ultrasound, I look for nuchal thickness or nuchal translucency. If it is more than 3 mm, it is indicative of Down syndrome, right? And also we do a blood test in the mother, that is known as dual test. Two things I am going to check. I am going to check beta HCG and PAP-A. Beta HCG and PAP-A. If beta HCG is high, PAP-A is low, this is again indicative towards Down syndrome. Now let's move to the second trimester. In second trimester, we do triple test and quadruple test. In triple test, 3 markers again we are going to do: beta HCG, unconjugated estriol, and alpha fetoprotein. Remember, high again beta HCG and low of unconjugated estriol and alpha fetoprotein. In quadruple test, we do all the things of the triple test and add inhibin also. Which marker increases or decreases in Down syndrome? A very simple mnemonic to remember. Remember beta HCG and inhibin. H for high, I for increase. So these two only increase, rest all fall. Rest all fall. Right? So very simple mnemonic for Down syndrome. So this is all about your Down syndrome and important markers, antenatal screening things, what are the associations. Now let's move to yet another important syndrome, that is Turner syndrome. So Turner syndrome is often confused with Noonan syndrome, which is also labeled as male Turner. But remember, Noonan is not male Turner, it's definitely a different set of disorder. What happens in Turner syndrome? We have monosomy XO. That means I have one chromosome less, XO. But what happens in Noonan syndrome? It is a genetic problem, a gene problem, right? PTPN11 gene problem. So here the chromosome number is fine. Some genetic problem is there. So definitely both of the disorders are different. One is the problem of chromosome number and other is a genetic problem, right? Here, because we don't have any Y chromosome, we have only one X chromosome, these are going to be girls only. But here the chromosome number is fine. This can be boy also, this can be a girl also. If you talk about IQ, then the Turner syndrome girls have normal IQ, but their gonads are dysfunction. They have streak ovaries, amenorrhea, streak ovaries, right? So I remember it that way, that they are intelligent but they cannot have sex, right? They can have sex obviously, but they cannot reproduce. So normal intelligent but cannot reproduce. On the other hand, Noonan syndrome, they have low IQ, but their gonads are normal. Their gonads are normal, right? No gonadal issue per se. Then comes the associations, cardiac associations and renal associations. We all know left-sided brain is very important for our intelligence, right? Left-sided has mainly your reasoning and everything. Right is for creativity. So here they have normal intelligence, left-sided. So they have left-sided heart anomalies. What is on our left side? We have left atrium, left ventricle, aorta. So they have bicuspid aortic valve and coarctation of aorta. On the other hand, here you are going to have right-sided problem. On the right side, you have right atrium, right ventricle, pulmonary artery, right? So you have pulmonary stenosis over here. So this is all about Turner and Noonan. Now let's briefly go through the features of the Turner syndrome. Short stature is going to be there, low hairline, droopy eyelids, low set ears, typical shield, widely spaced nipples, also known as shield chest. You have congenital heart diseases like we discussed, bicuspid aortic valve, coarctation of aorta. Renal anomalies are going to be there, streak ovaries are there, going to be there, very increased carrying angle, elbow deformities seen, brown spots, some nevi, knee deformities. All of these things are associated with Turner syndrome. Now let's move to yet another microdeletion syndrome, that is Digeorge syndrome. In Digeorge syndrome, we all know which chromosome is affected. Chromosome 22q11, right? Here the third and the fourth pharyngeal defect is there. Third and the fourth pharyngeal arch defect. This third and fourth pharyngeal arch is very important for the development of thymus, for the development of my heart, for the development of my palate and everything. So what are the features you are going to appreciate over here? The mnemonic is catch 22. C is for the cardiac anomalies. The most common cardiac anomaly that you see over here is, most common cardiac anomaly that you see in Digeorge syndrome is conotruncal defects like TOF, right? So conotruncal defects. Then A is for abnormal facies. T is for thymus. There is going to be thymic aplasia. C is for cleft palate, cleft lip, cleft palate. This H is for hypocalcemia. And this 22 is nothing but your 22 deletion, 22q11 deletion, microdeletion syndrome, right? Okay. So with that, we cover the genetic section. Now let's move to neonatology. Neonatology, very important segment for INICET, especially the neonatal resuscitation protocol. So don't forget to revise that before your exams, right? So firstly, we are going to discuss some of the respiratory disorders that we encounter in cases of neonates. So broad thumb rule, if it is a preterm, most common respiratory disorder that you're going to see is respiratory distress syndrome or hyaline membrane disease. If it is a term baby born via cesarean section, the most common respiratory distress syndrome that you're going to see over here is transient tachypnea of newborn. And in post-term babies or babies who have faced some sort of in utero stress, they're going to have this meconium aspiration syndrome. So firstly, let's start with respiratory distress syndrome or hyaline membrane disease. It is caused, what is the cause over here? It happens in preterms because of the deficiency of mature surfactant. Because of the deficiency of mature surfactant, what happens? My alveoli will collapse because there is increase in the surface tension. So how will the X-ray appear? The X-ray will appear as white out lung or ground glass appearance. Right? And secondly, you will appreciate air bronchogram over here, air bronchogram. How can we prevent this? We can prevent this by giving antenatal steroids to the mother, and the treatment here is CPAP therapy. If CPAP therapy fails, then we give surfactant. What is the technique by which surfactant is given? The technique is INSURE technique. INSURE technique. What is this? Intubate, give surfactant, and extubate. INSURE technique. Right? Then comes the second entity, that is transient tachypnea of newborn. I told you why this happens. This happens in a term baby who is born by a cesarean section. So normally when the delivery happens through the vagina, what happens? My baby slides through a very narrow vaginal canal. Normally in utero, my lungs are filled up with fluid, and during this vaginal delivery, what happens? This fluid is expelled out through mouth and nose, and the lungs are clear. But when we take out the baby through a cesarean section procedure, then the baby is not going through that narrow vaginal canal. Some of the fluid gets retained into the lungs, right? So because of this retention of fluid in the lung, the baby has this transient tachypnea. How do you see that on X-ray? There will be infiltration. This infiltration of this fluid in the lungs gives a sunburst appearance. And also you can see this fluid in the interlobar fissure. So fluid in the interlobar fissure. What is the treatment over here? This is a transient condition. Sooner or later this fluid will be resorbed. So here just supportive treatment is to be given. This is a transient condition, not more than 72 hours this is going to be there. Then we have meconium aspiration syndrome. What does this meconium do? This meconium can block my alveoli. This meconium can block the airway leading to the alveoli, completely block one airway. And what will happen to that segment? There will be collapse, segmental collapse. Also, this can be a partial blockage leading to a ball valve effect and hyperinflation on one side. And pneumonitis picture you will see. So these are the features. Again, supportive treatment is given over here. So now let's look at these x-rays and one by one tell me which one is what. So image A, what can you see? White lung, white out lung, ground glass appearance, and also can you see the big airways delineating out? This is bronchogram, air bronchogram. This is the typical x-ray of RDS, respiratory distress syndrome, or hyaline membrane disease. Look at the image B. White arrow is pointing towards fluid that is accumulated in the horizontal fissure. This is transient tachypnea of newborn. Also, can you see the infiltrations around the hilum? This is sunburst appearance. Then we have the last picture where I can see one segment has collapsed and overall hyperinflated picture is there. This is meconium aspiration syndrome. Right? Dharmesh is correct. RDS, TTNB, and MAS. ABC. Right? Now let's move to the scores. Important scores. INI people very favorite this score, Silverman-Anderson score. They will give you a newborn, and every time they give the same question, the answer to that question is 8. But definitely you should know the scoring system, because someday they might give some different question here and there, and the answer might change. So let's understand these respiratory distress score. So what this score is going to tell me? This score is going to tell me how much is the level of distress. More the score, more the distress. So it will help me to tell whether this child is in severe distress, mild distress, moderate distress. Should I ventilate this child? Should I intubate this child? Should I give CPAP therapy? Should I just wait and watch? Right? So more the score, severe the distress. So we have two scores in newborn for respiratory distress: Silverman-Anderson score and Downey score. Silverman-Anderson score, it is a big name. This big name is for a small child. That means this score I am going to use for preterms. So for preterms, we apply the Silverman-Anderson score. 5 components: upper chest retractions, lower chest retractions, xiphoid retractions, nasal flaring, and grunting. Lower chest and xiphoid retractions and nasal flaring, these three are very easy to remember. I told you that more the score, that means worse the distress. So if the lower chest retractions, xiphoid retractions, and nasal retractions are very mild, no nasal flaring, then it is 0. Sorry. If there is no chest retractions, no xiphoid retractions, no nasal flaring, it's 0. If it is mild retractions, mild flaring, 1. If it is moderate to severe, 2. Right? Then comes grunting. What is grunting? Expiration against the partially closed glottis. So at the time of expiration, a weird sound you are going to hear. Right? This grunting, if you are not hearing any grunting, 0. If this grunting is audible only via stethoscope, that means you cannot hear grunting per se, but once you place the stethoscope over the chest of the child, you are going to appreciate grunt. So if grunting is audible with stethoscope, this is 1. And if you don't need stethoscope, you are able to hear the grunt with your naked ear. Even mother can hear that the child is grunting. This is 2. So if the grunting is audible with naked ear, it's 2. Lastly comes upper chest retractions. Why I left this in the end? Understand when the child is breathing. If you look at the child, a newborn lying down breathing, the breathing is synchronous. The chest will move and the abdomen will follow. This is synchronous breathing. But if the child is in distress, the child will be breathing fast. So initially you will appreciate that there is certain lag on inspiration because of this fast breathing. And at one point when the distress becomes too severe, you will appreciate seesaw movement, seesaw breathing. So initially if there is synchronous breathing, 0. If there is a lag on inspiration, 1. If there is seesaw movement that you appreciate, 2. Right? So this is the Silverman-Anderson score. Now let's move to Downey score. I told you Silverman-Anderson score, big name for a small child, preterm child. Downey score is for a term child. Here the mnemonic to remember the components of Downey score is GRACE. In one of your exams, INICET exam, they asked which of these components is not a part of Downey score. And different things were given. And out of these, one was color, something like that. Right? So remember what are the components? G is for grunting. R is for respiratory rate. A is for air entry. C is for cyanosis. And R is for retractions. So grunting is similar to the Silverman-Anderson score. No grunting, 0. Audible with stethoscope, 1. Audible with naked ear, 2. Retractions is also same. No retractions, 0. Mild, 1. Moderate to severe, 2. Then comes respiratory rate. The normal respiratory rate of a newborn is 40 to 60. So if the respiratory rate is less than 60, 0. If it is 60 to 80, 1. If it is above 80, it's 2. Right? Then comes air entry. If the air entry is normal, equal air entry on both the sides, 0. If the air entry is decreased, 1. If the air entry is absent, 2. Then comes cyanosis. If there is no cyanosis, 0. If there is cyanosis on room air and you apply oxygen and the cyanosis disappears, it's 1. So if there is cyanosis on room air, it's 1. If even after giving 30% oxygen, the cyanosis is still there, it's 2. So if there is cyanosis on 30% oxygen, it's 2. Right, so this is Downey score. Remember the components are very important. GRACE is the mnemonic: grunting, respiratory rate, air entry, cyanosis, and retractions. Okay, so now that we are talking about scores, let's talk about this other score. This score we all are given, the very first score that we are given after birth. right? What is the score? APGAR score. This was named after the Dr. APGAR who actually gave the score, but we are going to remember this APGAR score by its name only, right? Its name, APGAR, is its mnemonic. A is for appearance, P is pulse rate or heart rate, G is for grimace, A is activity, and what is R? R is the respiratory efforts. So firstly, appearance. If the baby is appearing blue to you, you give 0. If the baby appears, if the baby appears blue to you, you give 0. If the baby's extremities are blue, that means hands or feet are blue, but the trunk, the body is pink, we give it 1. If the baby is fully pink, you give it 2, right? So here, more the score, better the baby is. Then we have pulse rate, heart rate. If there is no heart rate, 0. If the heart rate is there but less than 100, 1. More than 100, 2. Then we have grimace response. How do we elicit grimace response? By suction. If there is no response, 0. If the grimace response is there, then 1. And if the baby sneezes or cough out, then 2. Activity wise, if the baby is limp, 0. If there is some flexion, 1. If good flexion, actively moving the limbs, 2. Then comes respiratory efforts. If no efforts, 0. If there is labored breathing, then 1. And if there is good cry, then 2. Right? So this is all about APGAR score. Yes, acrocyanosis is 1. Okay, so with that, now let's move to the next topic, that is neonatal sepsis. Neonatal sepsis. So when we talk about neonatal sepsis, what does the examiner ask first? What is the most common cause? Which organism, which bacteria is the most common cause of the sepsis? Remember, when we talk about India, the most common organism is Klebsiella. But worldwide, it's Streptococcus. Group B, Streptococcus. Group B, Streptococcus. Right? In neonatal sepsis, we have two categories of sepsis: early onset neonatal sepsis and late onset neonatal sepsis. So, what are the risk factors for both of these? When we are talking about neonates, initially we discussed in the periods of growth that we have something that is known as early neonate and late neonate. Neonate is someone who is in the first 28 days of life. Early neonate is first 7 days, and late neonate is after 7 days till 28 days. But this is early onset. So this is not, don't confuse it with first 7 days of life, right? Early onset means if the infection is contracted within 72 hours of life. Within 72 hours of life, this is early onset, and after 72 hours, it is late onset. So early onset sepsis means within the 72 hours only the child contracted some infection. So that means this infection is from the mother itself. That's why this child has developed such early infection. So the risk factors for such early infections would be if the mother had prolonged labor, if the mother had prolonged rupture of membranes, if the child had spontaneous prematurity, if there is some sort of birth asphyxia, if there were multiple unsterile PVs who were done, right? So let's list down the risk factors. Firstly, it's prolonged labor. So if the labor is prolonged for more than 24 hours, if there is prolonged rupture of membranes, if there is spontaneous prematurity, if there is birth asphyxia, right? If multiple PVs were done, it is said that a single unsterile PV and 3 or more sterile PVs. Lastly, if the mother has some features of chorioamniotis, maternal fever, all of these is going to be a risk factor. What about late onset sepsis? This late onset neonatal sepsis happened after 72 hours of life. So it must be community acquired or hospital acquired. So the reasons would be unclean hands, poor hygiene, right? If the poor cord care is there, if the mother is not breastfeeding, there is bottle feeding, right? All of these are signs of risk factors for late onset sepsis. Now, how you are going to investigate? It is an infection, sepsis. So, gold standard here is going to be blood culture only. So, gold standard is blood culture, but we know this blood culture take 3 to 5 days to appear. So, what you are going to do meanwhile? We are going to do the screen that is known as sepsis screen. So, the sepsis screen has certain components, and if two or more abnormal components are there, we say that the screen is positive, right? So for the sepsis screen, remember, I need two or more abnormal values. If two or more abnormal values are there, the screen is positive then. So first component is absolute neutrophil count. If it is less than 1800, then it is abnormal. Then comes IT ratio, that is immature to total neutrophil ratio. Right? If it is more than 20% or more than 0.2, it is considered abnormal. Then we have CRP more than 1 nanogram per ml. And lastly, we have micro ESR. If it is more than 15 per hour, it is abnormal. So I need two or more than two abnormal values out of these to label it as positive. Now let's understand the treatment. The treatment, because it is an infection, is going to be antibiotics only. We start with empirical antibiotics first. We give ampicillin plus gentamicin. If the child is not responding to these antibiotics for 48 hours, we move to third generation cephalosporins. And if the child has meningitis, then we start with the third generation cephalosporin only. We don't give this empirical ampi and genta. The question comes, how much duration of antibiotics you have to give. So remember, if the sepsis screen is positive, then you give it for 7 days. If blood culture is positive, 14 days. If there is meningitis, 21 days. So remember, 1 week, 2 week, 3 week. Right? So this is all about neonatal sepsis. Now we are talking about neonatal sepsis. One sepsis, gut sepsis, that is necrotizing enterocolitis, deserves a mention. In NEC, what is happening? The name itself says itis means inflammation. So there is inflammation of the colon and the small intestine, enterocolitis, right? Small intestine and the colon, and necrotizing inflammation is there, right? Necrosis is overall there. So we have staging, Bell staging, which the examiner is fond of asking. So what will happen over here? Firstly, I know there is inflammation of the gut. So what is the first feature that you're going to see? The child is going to be looking sick, lethargic. There will be feed intolerance, vomiting. There will be abdominal distension. There can be blood and stools. So stage 1 is divided into stage 1A and 1B depending upon this clinical feature only. In 1A, you will see occult blood and stools. Occult blood and stools. That means you won't see blood with the naked eye. You see that under the microscope. Under the microscope, you will notice these RBCs. And in stage 1B, you will see gross blood. That even mother will tell that in stools, doctor, there is blood. Here, no specific imaging feature is seen. Only the clinical features that the baby is sick, lethargic, there's going to be abdominal distension, feed intolerance, vomitings, right? And this blood and stools. Then comes stage 2. In stage 2, along with the features of stage 1, you will appreciate a classical triad. This is known as a metabolic triad. What do you see in this metabolic triad? In this metabolic triad, I will see metabolic acidosis. You will see hyponatremia and thrombocytopenia. That means low platelets. And now you will appreciate certain imaging features. On the basis of imaging features, the stage 2 is divided into stage 2A and B. In A, what do you see? In A, you see In A, you see that the air has gone and trapped in between the layers of the intestine. This is known as pneumatosis intestinalis. Pneumatosis intestinalis. And in stage 2B, you see air trapped in between the portal vein. So this is known as pneumatosis portalis. Now in stage 3, full-blown perforation has happened. So in clinical feature, you will notice peritonitis-like features, which is stage 3A. And then in 3B, you will see the imaging feature, that is pneumoperitoneum, air under the diaphragm. Right? So remember stage 1A, 1B on the basis of clinical feature, that is presence of blood in the stools. If occult blood, 1A. If gross blood, 1B. Stage 2 is divided into 2A and 2B on the basis of imaging feature, not clinical, imaging feature. Pneumatosis intestinalis, 2A, portalis, 2B. And stage 3, 3A is clinical feature, that is peritonitis, and stage 3B is pneumoperitoneum. Right? So this is about the Bell staging, very important for your exam. Now let's start with a big topic of neonatal resuscitation, right? If one topic you have to study before your exam, especially the INI exam, every year they are going to throw one question, at least one question out of this segment, right? They will never ever forget this, right? If one question has to come from pediatrics, it will be from neonatal resuscitation. That's how important this is. So neonatal resuscitation protocol. So remember, these babies are born with good heart and bad lungs. Good heart and bad lungs. What do I mean by good heart and bad lungs? That means whenever we are starting with resuscitation, the focus is going to be lungs because they are bad, right? So my cornerstone of NRP is going to be effective ventilation. The cornerstone of NRP is effective ventilation. If despite this effective ventilation, the baby is not responding, if despite this effective ventilation, the baby is not responding, then I will go towards heart. Then I will choose chest compressions or epinephrine, right? So this is the cornerstone. So one by one, let's understand what we are going to do. Firstly, we have pre-birth questions. What are the pre-birth questions? 4 pre-birth questions. Whenever a baby is going to arrive into this world, your OBGYN friend will call you that please come and receive this baby. You're going to ask these 4 pre-birth questions. The first pre-birth question, whether this is a term baby or a preterm baby. That means I'm going to ask the gestation age of this baby. Right? Secondly, I'm going to ask if there is any maternal risk factor. Maternal risk factor. That means if it is a high-risk delivery or not. Then I'm going to ask whether there is meconium stained liquor or not. That means I'm going to ask the color of the amniotic fluid. And lastly, the cord management plan. Cord management plan means whenever the baby is delivered, I will discuss pre-hand with the OBGY that I am going to do delayed cord clamping or not. Cord management indicates my delayed cord clamping, that is, we will delay the cord clamping in most of the scenarios. If resuscitation is needed, we won't delay, but if everything is normal, we will delay the cord clamping for 60 seconds. 60 seconds. So this is the 4 pre-birth questions. Earlier, single baby or multiple baby was also included, but now that question is not there. Right? So after this pre-birth questions, we will start equipment check, assign our team members and prepare for the birth. Post-birth, 3 questions are there. What are the 3 questions? Term, tone, cry. Whether this baby is a term baby, is the tone good? Did the baby cry well? If the answer to all of these questions is yes, then the baby needs routine care. If the answer to any of these questions is no, then I will go for initial steps of resuscitation. if the baby is not responding to this initial steps of resuscitation. That means even after the initial steps of resuscitation, if the heart rate of the baby is less than 100, then we start ventilation. Which was earlier called positive pressure ventilation. Now the recent update has come, and this positive pressure ventilation is replaced by the term ventilation, right? So we start ventilation. After 30 seconds of this effective ventilation, if the heart rate is still less than 60, then I'm going to start chest compressions along with this ventilation. 1 minute, that means 60 seconds of this chest compression and effective ventilation. If the baby's heart rate is still less than 60, you're going to give epinephrine. And even after repeating this dose of epinephrine, if the baby is not responding, then you rule out two conditions: pneumothorax and shock, blood loss, right? For pneumothorax, you do thoracotomy, and for blood loss, you give volume expanders like normal saline. So one by one, we are going to cover this concept only by these one-liners. So first one-liner, what is the most important step in neonatal resuscitation? I told you, babies are born with good heart and bad lungs. So what is the most important step? Most important step is effective ventilation. Then comes the golden 1 minute. What is the golden 1 minute? If the baby needs positive pressure ventilation, if the baby needs ventilation, it should be started within 60 seconds. This is the golden 1 minute, 60 seconds. Then comes the initial steps of resuscitation. If the baby term, tone, cry, if the answer to any of these questions is no, you start this initial steps of resuscitation. What are these? Position, airway, dry, stimulate, pads. What is the position? Sniffing position. Do we do routine suctioning? No, routine suctioning is not done. Even if meconium is present, you don't do routine suctioning. Suctioning is done only when needed. No routine suctioning. Then when do you start positive pressure ventilation? When do you start ventilation? If the heart rate is less than 100 after your initial steps of resuscitation. When do you start chest compressions? If the heart rate is less than 60 after 30 seconds of positive pressure ventilation. How do you start chest compressions? What are the compression to ventilation ratio? The compression to ventilation ratio is 3:1. That means you compress 1, 2, 3 and then give one breath. 1, 2, 3, breath. 1, 2, 3, breath. Right? What is the site of this chest compression? The site of this chest compression is lower 1/3 of the sternum. Just below the level of nipples. And how deep should be the compressions? Compressions again would be 1/3 of the AP diameter of the chest. The best indicator of the effective ventilation is increase in the heart rate. Remember, this is a very favorite mnemonic. Which of these is the most important indicator of the effective ventilation? If you are doing effective ventilation, heart rate should increase. Then comes the concentration of oxygen. For term babies, you should start with 21%. For preterm babies, 21 to 30%. If the baby has labored breathing but heart rate is more than 100, then you give CPAP and positive pressure ventilation if needed. When do you start epinephrine? If the heart rate is still less than 60 after 60 seconds of compression and ventilation. What is the preferred route of giving epinephrine? Preferred route is your intravenous route through umbilical venous catheter. The dose that we use is 0.2 ml per kg. 0.2 ml per kg. This is of the concentration 1:10,000 dilution. If no IV access is there, then you use intraosseous route or endotracheal route. The volume expander that we use is normal saline. What is the indication of volume expansion? If there is a suspected bleed happening, right? So by this, we have covered most of the question. Then comes a very old concept of meconium stained Laika babies. Earlier, these babies were divided into vigorous baby, non-vigorous baby. What is a vigorous baby? When the heart rate is more than 100, the activity is good. In these babies, nothing you have to do with routine care you have to do. Non-vigorous babies are those babies whose heart rate is less than 100 and activity is poor. In these babies, you have to follow your routine neonatal resuscitation protocol. No special thing has to be done. Remember, this delayed cord clamping is not done if the baby requires immediate resuscitation. If no resuscitation is required, then we delay the cord clamping for 60 seconds. Why we are delaying this cord clamping? Because this will allow some additional amount of blood to flow to the baby through the cord, and this decreases the chances of anemia in later life, right? Not right now, but in later life the chances of anemias would be less, right? With that, we end this NRP protocol, a very rapid fire thing. Now let's move on to a very important segment of congenital infections. Right, so congenital infections, 3 infections very important: toxoplasmosis, congenital rubella syndrome, and CMV infection. And all three things would be there. A triad like scenario would be there. Let's start with congenital toxoplasmosis. Firstly, you are going to see one eye sign. What is that eye sign? Chorioretinitis. Then you see certain calcification in the brain. What type of calcification? Diffuse calcification. And third is, yes, hydrocephalus. Head problem, hydrocephalus. Then comes congenital rubella syndrome. Firstly, you will see a heart problem, patent ductus arteriosus, PDA. Then an ear problem, sensory neural hearing loss. Then you will see eye problem, that is cataract. Similarly, for CMV, compare it to toxoplasma. Here also you will see C for chorioretinitis, M for microcephaly, small head, right? And V is for ventricle. So here I see ventricular calcification. Right, so compare it with toxoplasma. Chorioretinitis is common. In toxoplasma, diffuse calcification were there. Here you have periventricular calcification. In toxoplasmosis, big head hydrocephalus were there. In CMV, small head microcephaly is there. Right, so CMV is the mnemonic for congenital CMV infections. So with that, now let's move to important segment, GI system diarrheas. What is the most common cause of diarrhea in preterms? Is it bacterial or viral? It's bacterial or viral? It's viral, right? So viral diarrheas are more common as compared to bacterial diarrheas. That's why usually we don't give antibiotics for pediatric diarrheas, right? No antibiotic for the pediatric diarrheas. If I ask you what is the most common viral cause, it is the rotavirus. And what is the most common bacterial cause? The most common bacterial cause is E. coli. Which type of E. coli? Enterotoxicogenic E. coli. Right? And what about dysentery? What is dysentery? Loose stools with blood. The most common cause of dysentery is Shigella. So now let's talk about the management of diarrheas. As I told you, the viral diarrheas are more common in pediatric population as compared to the bacterial diarrheas. So here the management of diarrhea would be just managing the dehydration that is associated with this diarrhea and supplementation with zinc. So dehydration management and administration of zinc make the cornerstone of management of diarrhea. So let's understand the dehydration correction first. So whenever a child with diarrhea, loose stools, vomiting comes to you, firstly you assess the level of dehydration and then you manage accordingly. We have 3 levels of dehydration: no dehydration, some dehydration, and severe dehydration. No dehydration, the name itself tells that there is no dehydration. So that means this child would be an alert child, normal consciousness alert child. If you offer water to this child, this child has no dehydration. So this child will be drinking water normally, right? So normally gentle like the child, the thirst is normal, so the child will gently drink water. If you do skin pinch, the skin pinch recoil would be again normal, immediate recoil. Eyes would also look normal to you. Here I give plan A. Then comes some dehydration. Now some degree of dehydration has set in. So this child will be very irritable. If you offer water to this child will now very eagerly drink water. He will be very thirsty, right? So this child will eagerly drink water. Skin pinch here would be delayed, but less than 2 seconds. Eyes would look sunken. Oral mucosa would also be dry. Here I do plan B. Then comes lastly severe dehydration. Here the child is now severely dehydrated. So the child would look comatose, lethargic. If you offer water to this child, will this child be able to drink water? No, he's lying comatose, lethargic. So this child will be unable to drink water, unable to drink. Here the skin pinch is very delayed, more than 2 seconds. Eyes look very sunken, oral mucosa is very dry. Here I go for plan C. Right, so one by one, let's understand plan A, plan B, plan C. So plan A was for a child who had no dehydration, just losses through the stool or vomiting is happening, and the child does not have dehydration per se now. The child can very easily drink water through his mouth. So here, the rehydration solution that I am going to give is oral plan ORS, oral rehydration solution. How much? 10 ml per kg per loose stool. Right, so for every loose stool the mother has to give 10 ml per kg of fluid to drink. So if it is a 10 kg child, for every loose stool 100 ml of ORS has to be consumed. Then comes plan B. Plan B, the child has some dehydration. So though this child can take orally, but this child has some level of dehydration. So firstly, I have to rehydrate the child. Alongside, I have to replace the losses of the child. Right. So here again, the child can drink. So I will give oral ORS. Firstly, you have to rehydrate 75 ml per kg over the next 4 hours. Replacement of losses will stay the same. And again, in all of these, you have to ensure that maintenance fluids that we all require every day are covered. In plan C, when the child has severe dehydration, now the child cannot take orally, so I will give IV. The fluid of choice here is Ringer lactate. Rehydration here is given as 100 ml per kg. If it is a small child less than 1 year, then I give initial 30 ml per kg over 1 hour, rest 70 ml per kg over 5 hours. And if it is a big child above 1 year, then this can be given faster. Over 30 minutes I give this, and over 2.5 hours I give this. Other replacement of the losses and maintenance fluid stay the same. Let's understand the concept of maintenance fluid. So we all know that we need water. We need water. So you and me, everyone, we need some amount of water for our day-to-day activity, right? This is known as maintenance fluid. This is the bare minimum that you and me, everyone requires. How do we calculate this? By holiday cigar method. For the first 10 kgs, it is 100 ml per kg. For the next 10 kg, that is from 10 kg to 11 to 20 kg, it's 50 ml per kg. And thereafter, for every subsequent kg, it is 20 ml per kg. So if I say calculate the maintenance fluid requirement for a 22 kg bacha, how will you calculate? For the first 10 kg, it's 100 ml per kg, so that would be 1000 ml. Then for the next 10 kg, it's 50 ml per kg, so it would be 500 ml. And for subsequent 2 kgs, it would be 2 into 20, that is 40 ml. So total maintenance fluid requirement would be 1540 ml for a 22 kg child. Right? So Dharmesh, Dhanush, absolutely correct. Right? So now let's move to the next important topic, that is pneumonia. In pneumonias, we have IMNCI guidelines. Before we go into the guidelines, we need to get our basics straight, right? First basic, what is fast breathing? Fast breathing, we have cut off as compared to the age. So remember, less than 2 months, if the respiratory rate is more than 60 breaths per minute, it's fast. After that, more than 50. After 12 months, more than 40. So remember 60, 50, 40, 2 months, 2 to 12 months, after 12 months. This is the criteria for fast breathing. Then we have danger signs. If any of these danger signs is there, the child is going to have severe pneumonia, you need to refer this patient further. Right? So what are the danger signs? If there is stridor in a calm child, this is a danger sign. if there is severe respiratory distress, cyanosis, if the child is unable to drink water, if the child's consciousness is not normal, the child looks abnormally sleepy, if the child's saturation is less than 90%, if the child has convulsions or seizures, all of these are danger signs. So with that, let's move to our IMNCI guidelines. So IMNCI divides this pneumonia, upper respiratory distress features and everything into three categories: no pneumonia, pneumonia and severe pneumonia. No pneumonia means there is no pneumonia, just child has some upper respiratory infection. So here the child is going to have cold, cough-like features, URI-like features, right? No signs of respiratory distress, no retractions, no respiratory rate, no danger sign, right? So here nothing but supportive treatment has to be given. Then comes pneumonia. In pneumonia, either you are going to have fast breathing, that is increase in the respiratory rate, and or retractions, but no danger sign, no danger sign. So here though child has pneumonia but no danger sign is there, so you can give oral antibiotic to this child and treat the child on OPD basis and tell the mother or the caregiver about the danger sign and when to follow up back, right? So here you will give antibiotic, we give oral amoxicillin for 5 days and ask for follow up, explain danger signs. Then comes pneumonia, severe pneumonia. If any danger sign is there, it is severe pneumonia. Here you give one shot of antibiotic. Usually we give ampicillin, gentamicin and refer to the higher center. So you give IV ampicillin and gentamicin and refer to the higher center for further management. So this is all about IMNCI guidelines. With that, I want to discuss a very important topic for respiratory system, that is cystic fibrosis. This is very favorite of INI examiners, that's why we are going into detail of this particular topic. So we all know this is a genetic problem. What is the inheritance over here? The inheritance here is autosomal recessive. The gene is the CFTR gene that is defective. On which chromosome this gene is there? On chromosome number 7. The most common mutation is deletion of the phenylalanine at 508 position, right? And the protein defect is the CFTR protein only. This is a chloride channel. The common pathogens over here depend upon the age. In infancy and childhood, the common pathogen is the Staph aureus, and in adults it is Pseudomonas. Right, what are the features? You see pancreatic insufficiency, you see meconium ileus, rectal prolapse, liver involvement, male infertility. The gold standard test for diagnosis over here is the sweat chloride test, right? Sweat chloride test. What is the abnormal levels that you see over here? The abnormal levels are more than 60 millimole per liter in more than 2 separate tests. It is indicative. And obviously genetic testing detecting 2 CFTR mutations is yet another confirmatory test. There is no permanent cure yet. We do supportive therapies like Dornase alpha to liquefying the mucus, hypertonic salines, bronchodilators, antibiotics, pancreatic enzyme supplementation, and in pharma you'll be studying about the CFTR modulators, which is yet another important topic for your exam, right? Now certain one-liners that are associated with this cystic fibrosis, they ask about the earliest GI manifestation that you see. Remember, the earliest GI manifestation is meconium ileus. The earliest pulmonary pathogen is Staph aureus. Most characteristic is Pseudomonas, and remember the most deadly is Burkholderia capacia. Burkholderia capacia, right? With that, now let's just discuss some more one-liners and examiner's favorite question. So examiner usually asks some of the associations of congenital heart disease with genetic syndromes. In genetics, we discussed about Down syndrome, Turner syndrome, Noonan syndrome and their associated CHTs. This table encapsulates all the common commonly associated congenital heart disease in different genetic syndromes. So firstly, Down syndrome, we all know. What is the most common? It's endocardial cushion defects. Endocardial cushion defect. Then in Turner syndrome, you see left-sided heart defects like bicuspid aortic valve, coarctation of aorta. In Noonan, right-sided problems like pulmonary stenosis. In Edward and Patau syndrome, you see ventricular septal defect. What is Edward syndrome? It's trisomy 18, E for Edward, E for 18. And what is Patau? It is trisomy 13. In Dijaw syndrome, we see conotruncal defects like TOF. And then we have Williams syndromes where you see supravalvular aortic stenosis. Supravalvular aortic stenosis. Then in certain maternal conditions, again you see association of congenital heart diseases. For example, in gestational diabetes, the most common congenital heart disease that we see is ventricular septal defect. And the most specific one is transposition of great arteries. We have rubella, causing congenital rubella syndrome in the baby, and the common CHD we discussed is PDA. If the mother is an alcoholic, consumes alcohol throughout the pregnancy, the baby has chances of developing fetal alcohol syndrome. If the mother is taking phenytoin throughout the pregnancy, fetal Hydantoin syndrome. In both of these conditions, VSD is an important congenital heart disease that we encounter. If mother has bipolar disorder and is taking lithium, we see Epstein anomaly. If the mother has SLE and antero-antila antibodies, baby can have neonatal bradycardias, heart blocks, right? Heart blocks, neonatal bradycardia, right? With that, certain X-rays need a mention. So this first X-ray, what can you see? Figure of 8 appearance or snowman appearance. Where do you see this? We see this in TAPVC. Which TAPVC? Supracardiac TAPVC. Supracardiac TAPVC. Then we have this characteristic boat-shaped heart. Where do you see this? In tetralogy of Falley. Tetralogy of Falley. What are the 4 components of tetralogy of Falley? We have pulmonary stenosis, ventricular septal defects, overriding aorta, and right ventricular hypertrophy. Then we have this box-shaped heart. Where do you see this? If the mother is consuming lithium for the bipolar disorder, right? So Epstein anomaly, box-shaped heart. Then we have this egg on string appearance. Where do you see this? In transposition of great arteries, right? With that, we cover a very important segment of cardiovascular system. We will now fatafat wrap up the pediatric epilepsy syndromes and end the session. So in pediatric epilepsy syndrome, what is important? Age group is very important. The semiology or type of seizure that you are going to see is very important, and typical EEG finding. So West syndrome. The typical age group is before 1 year of age. You see characteristic triad. What is this triad? This triad will have one seizure finding, that is salam spells or infantile spasms. You have one developmental finding, that is developmental regression. And you have one EEG finding, that is hips arrhythmia. Very chaotic pattern on the EEG, hips arrhythmia. The treatment is nothing but ACTH and steroids. So remember, if you look at Ghai, they have given drug of choice as ACTH or steroids. But as you are going to give INI exam, please prefer steroids, because in AIIMS they give steroids. If this West syndrome is associated with tuberous sclerosis, the drug of choice changes to vigabatrin. Vigabatrin. Then we have Dravet syndrome. After 1 year, you see this, 1 to 2 years. Here, what do you see? Again, these 3 things. Seizure type, you see multiple seizures, different types of seizures, right? EEG finding will be typical. What is the EEG finding? An abnormal EEG study of these seizures. The drug of choice here is valproate. And the new drug over here is teripental. Teripental. Okay. Then we have Lennox-Gastaut syndrome. This you see after 2 years of age. Here you will again see multiple types of seizures, developmental regression, and the EEG pattern typical over here is 2 to 3 spikes and wave complex. Drug of choice again here is going to be valproate. We have juvenile myoclonic epilepsy. Next, Here, the age group is adolescent females. The typical pattern here would be myoclonic jerks, shock-like contractions. And how they are triggered? Triggered by sleep deprivation and alcohol consumption. How do you see that on EEG? 4 to 6 polyspike wave complex. Drug of choice again valproate. The other alternative that you can use is levetiracetam. Lastly, absence seizures. Where do you see the school going child? Brief episodes of vacant staring. Brief episodes of vacant staring. Here the typical pattern is 3 hearts, spike and wave complex. Drug of choice here is ethosuximide. If ethosuximide is not in the option, choose valproate. With that, finally at 5 p.m. We are able to end this session. I hope this was helpful for your revision part and also to highlight all the segments that are important from the pediatric section. All the best for your exam. I will definitely meet after the exam with the recall session and would definitely want you to go through all of these topics, especially the neonatal resuscitation protocol before your exam. All the best. Thank you.
Hello friends, welcome to the INI-CET Marathon series, marathon sessions. I hope I am audible and visible. Everything fine? Can we start? So we are calling it Predictor series, and why? Because कुछ टॉपिक्स जो कॉमनली पूछे जाते हैं, ऐसे टॉपिक्स लेके हम आपके पास आए हैं. ऑल राइट, आई होप आई एम ऑडिबल एंड विजिबल. तो ऐसे टॉपिक्स लेके हम आए हैं. मतलब मैंने MCQs तो लिए हुए हैं, इमेज बेस्ड क्वेश्चंस, लेकिन उसके अलावा भी मैंने और कोई चीजें ऐसी ली हुई है. ओके, या नाउ इट्स सेट. कैन यू सी मी? चलो, तो टॉपिक सिलेक्ट किए हैं इस तरीके से कि इस बार INI-CET में क्या क्या टॉपिक्स के ऊपर क्वेश्चन आ सकते हैं. बेस्ड ऑन दैट मैंने मतलब ये टॉपिक सिलेक्ट किए. सो विदाउट वेस्टिंग एनी फर्दर टाइम, लेट्स स्टार्ट आवर सेशन. आपको कुछ डाउट रहेगा तो आप पूछ सकते हो. फिलहाल तो मुझे कोई चैट नजर नहीं आ रहा, बट आई होप कि आप लोग टाइप कर रहे हो, लिख रहे हो. चलो, ओके. सो MCQs तो लिए ही है, उसके अलावा कुछ और टॉपिक्स के ऊपर भी मैंने कुछ कहते हैं ना बिट्स एंड पीसेस, क्योंकि ये टॉपिक्स के ऊपर क्वेश्चंस आने के चांसेस मुझे मैक्सिमम लगते थे, तो मैंने वो टॉपिक सिलेक्ट किए हैं हमने. और विल स्टार्ट नाउ. ऑल राइट, सो वी आर स्टार्टिंग विथ इमेज बेस्ड क्वेश्चंस. Okay, I hope you can see and hear properly. चलो, तो let's start. Starting with the first one, identify the condition indicated by the shaded area. आपको पता है कि ये डैरो यूनिट डायग्राम्स है. हेलो अर्श, वेलकम वेलकम. चलो, फाइनली किसी ने तो हेलो कहा. मैं वही देख रहा था कि चैट बॉक्स में कुछ नजर नहीं आ रहा. तो चलो, हेलो अर्श, हेलो मुलश्री सिंह जी, हेलो. यस यस यस, गुड इवनिंग गुड इवनिंग. चलो, गुड गुड, फाइनली गुड टू सी यू ऑल. तो अभी पहला क्वेश्चन है डैरो यूनिट डायग्राम्स के ऊपर. मेरे हिसाब से इस साल इसके ऊपर क्वेश्चन आने वाला है, आ सकता है. तो क्या लगता है आपको, ये कौन सी कंडीशन है? हेलो मयूर, वेलकम. क्या कंडीशन लग रही है आपको? शेडेड एरिया में वो कंडीशन दिखाई गई है. सो व्हाट इज दिस कंडीशन? यू कैन रीड द ऑप्शंस. इज इट SIADH, और इज इट डायबिटीज इंसिपिडस, और एडमिनिस्ट्रेशन ऑफ हाइपरटोनिक सलाइन, या तो फिर कॉलरा? सो व्हाट इज दिस कंडीशन? आपने डैरो यूनिट डायग्राम्स इसके पहले की होगी, आई एम श्योर. क्लिनिकल साइड में किया होगा, या फिर इवन फिजियोलॉजी में भी फर्स्ट प्रॉफ में बॉडी फ्लूइड कंपार्टमेंट्स. तो क्या लगता है आपको, ये क्या कंडीशन है? मतलब सबसे पहले आपको ना वो डायग्राम रीड करनी होती है. मेरे हिसाब से इस साल इसमें से एक क्वेश्चन आएगा ही आएगा, बॉडी फ्लूइड कंपार्टमेंट्स के डिस्टर्बेंसेस से. मतलब डायग्राम देके पूछा जा सकता है. तो ये कैसे रीड करते हैं? हॉरिजॉन्टल एक्सिस पे है अपना वॉल्यूम्स, आप देख सकते हो, ICF वॉल्यूम, ECF वॉल्यूम, और वर्टिकल एक्सिस पे है ऑस्मोलैरिटी ऑफ द बॉडी फ्लूइड्स. तो अगर डायग्राम दिया है, इसका मतलब आपको सिर्फ ECF साइड में देखना है। सबसे पहली बात, यू नो द रूल ना इसका, कि प्राइमरी चेंज जो होता है वो इट इज ऑलवेज इन द ऑन द ECF साइड। तो ECF साइड में क्या हुआ है, वॉल्यूम हैज इंक्रीज्ड, दैट मीन्स दिस इज ओवर हाइड्रेशन, एंड टोनिसिटी कम हो गई है ECF की, ECF टोनिसिटी हैज डिक्रीज्ड, मतलब ये हाइपोटोनिक ओवर हाइड्रेशन है। करेक्ट 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 करेक्ट। तो मयूर पाटिल जी ने और अर्श जी ने दोनों ही ने करेक्ट लिखा है कि ये हाइपोटोनिक ओवर हाइड्रेशन है। लेट्स जस्ट राइट इट हियर। ओके, हाइपोटोनिक ओवर हाइड्रेशन, इसका मतलब क्या, ओनली वाटर इज गेन्ड इनटू द बॉडी, वाटर ही वाटर कॉन्जर्व हुआ है। तो ये कब होता है, और आपने आंसर दे भी दिया है, दिस इज SIADH, इनएप्रोप्रिएट ADH सिक्रीशन हो रहा है, जिसकी वजह से वाटर रिटेन हो रहा है और इसलिए ओवर हाइड्रेशन। चलो एक काम करते हैं, बाकी 3 कंडीशंस में भी क्या होगा ये आप सोच के बता सकते हो। ओके, जैसे डायबिटीज इंसिपिडस, इसमें क्या होगा, व्हाट टाइप ऑफ कंडीशन विल बी डायबिटीज इंसिपिडस। सोचो, डायबिटीज इंसिपिडस मतलब ओनली वाटर लॉस, वाटर ही वाटर लॉस हो रहा है फ्रॉम द ECF। तो क्या होगा इफेक्ट, डिहाइड्रेशन, एंड क्योंकि सिर्फ वाटर चला गया फ्रॉम द ECF, इसलिए ECF विल बिकम हाइपरटोनिक एंड डिहाइड्रेटेड। सो अपना डायबिटीज इंसिपिडस विल बी हाइपरटोनिक डिहाइड्रेशन। ठीक है, एडमिनिस्ट्रेशन ऑफ हाइपरटोनिक सलाइन, ये सिंपल है, अगर आप हाइपरटोनिक सलाइन देते हो तो क्या होगा, हाइपरटोनिक सलाइन मतलब ओवर हाइड्रेशन होगा एंड द ECF विल बिकम हाइपरटोनिक, सो इट विल बी हाइपरटोनिक ओवर हाइड्रेशन, बराबर। एंड फाइनली कॉलरा, equal amount of sodium and water is lost in cholera, so it will be isotonic dehydration। और मेरा एक फेवरेट MCQ है, वो पूछता हूं, मे बी आपको कॉन्सेप्ट अगर पता हो तो आप इसका आंसर दे सकते हो, ऐसे ही हाथ के हाथ MCQ पूछ रहा हूं। ओके, तो सुनो, ए मैन वॉज ऑन अ वोयाज, एक मैन है, शिप में है, और वो गलती से ही ड्रिंक्स 1 लीटर ऑफ सी वाटर, 1 लीटर ऑफ सी वाटर, तो व्हाट विल हैपन टू हिम आफ्टर 1 आवर। सुन रहे हो सब लोग, एक ऐसे ही MCQ थ्रो कर रहा हूं, लेट्स सी क्या आप ये सोच सकते हो। मैन शिप पे है, वोयाज पे है, वो शिप होती ना क्रूज पे, और उसने गलती से 1 लीटर सी वाटर पी लिया, सी वाटर, तो उसका 1 घंटे के बाद उसकी कंडीशन क्या होगी, ये क्वेश्चन है। ओके, और ऑप्शंस है हाइपरटोनिक डिहाइड्रेशन, हाइपोटोनिक डिहाइड्रेशन, हाइपरटोनिक ओवरहाइड्रेशन और हाइपोटोनिक ओवरहाइड्रेशन। तो अर्श, मयूर, डॉक्टर मोलश्री सिंह, क्या आपको लगता है कभी सुना हुआ सा ये MCQ? मेरा फेवरेट MCQ है, थोड़ी सी कॉन्सेप्ट लगती है इसको। चलो हिंट देता हूं, फिर आप सोच के जवाब दे सकते हो। ऑस्मोलैरिटी ऑफ सी वाटर इज 2400 मिली ऑस्मोल्स पर लीटर। सी वाटर की ऑस्मोलैरिटी होती है 2400 मिली ऑस्मोल्स पर लीटर। तो क्या अब आप सोच के जवाब दे सकते हो? अरे वाह, क्या बात है, स्मूथ स्पेशलिस्ट जी ने भी आंसर दिया है। धनुष, यस, करेक्ट करेक्ट करेक्ट, हाइपरटोनिक डिहाइड्रेशन। मोलश्री सिंह जी ने हाइपोटोनिक लिखा है, लेकिन बाकी सब ठीक है, डिहाइड्रेशन करेक्ट। ये सिंपल था, सी वाटर की ऑस्मोलैरिटी होती है, जैसा मैंने कहा, 2400 मिली ऑस्मोल्स पर लीटर, 2400, और किडनी की मैक्सिमम कॉन्सेंट्रेटिंग एबिलिटी कितनी है? नोट डाउन करो, 1200 मिलियोस्मोल्स पर लीटर। किडनी जब यूरिन को कॉन्सेंट्रेट करती है तो मैक्सिमम कॉन्सेंट्रेटेड यूरिन कितनी जा सकती है? 1 लीटर में 1200 मिलियोस्मोल्स इतने सोल्यूट्स एक्सक्रीट हो सकते हैं मैक्सिमम। तो सोचो, इस बंदे ने 1 लीटर सी वाटर पिया, उसमें 2400 मिलियोस्मोल्स इतने सोल्यूट्स थे और सभी एक्सक्रीट करने थे, एक्सक्रीटेबल है। तो 2400 मिलियोस्मोल्स को एक्सक्रीट करने के लिए उसका यूरिन आउटपुट कितना होना पड़ेगा? 1 लीटर में 1200 ही एक्सक्रीट हो सकता है मैक्सिमली, तो 2400 एक्सक्रीट करने के लिए 2 लीटर आउटपुट लगेगा। तो उसने 1 लीटर पानी पिया और 2 लीटर एक्सक्रीशन, तो मतलब 1 एक्स्ट्रा लीटर जो है वो उसके बॉडी से चला गया, रिजल्टिंग इन डिहाइड्रेशन। सो यस, दैट्स राइट। 1200 एनसीईआरटी में भी दिया रहता था, करेक्ट करेक्ट, एनसीईआरटी में 1200 मिलियोस्मोल्स दिया था। सो दैट्स राइट। चलो अगले एमसीक्यू की तरफ बढ़ते हैं। ओके, नेक्स्ट एमसीक्यू, ये थोड़ा सा एक एक स्लाइड है बीच बीच में। ओके चलो, नेक्स्ट एमसीक्यू एलवीपीवी लूप के ऊपर अगला एमसीक्यू है। मेरे हिसाब से इस साल, दिस ईयर, एलवीपीवी लूप के ऊपर एमसीक्यू आने का चांस है, पूरा पूरा चांस है। सो एक एलवीपीवी लूप दिखाई गई है यहां पर और पूछा गया है कि आइडेंटिफाई द कंडीशन, and the options are, these are the options: aortic regurgitation या pericardial effusion या mitral stenosis या heart failure। अर्श जी कह रहे फर्स्ट ईयर स्टूडेंट है, कोई बात नहीं अर्श जी, आपका भी फायदा होगा सुन के ऑब्वियसली, क्योंकि फिजियोलॉजी है और अब तक आपने कर भी ली होगी। धनुष जी कह रहे हैं ए, एओर्टिक रिगर्जिटेशन। वेल यस, बिल्कुल सही जवाब है, एओर्टिक रिगर्जिटेशन। तो क्यों ये एओर्टिक रिगर्जिटेशन है और बाकी कंडीशंस में क्या होगा वो देखते हैं। सबसे पहली बात आपको इसमें ये देखनी थी कि ये देखो, आइसोवॉल्यूमिक कॉन्ट्रैक्शन। सबसे पहली हिंट इस डायग्राम में, सबसे पहली हिंट जो है इस डायग्राम में वो ये है कि आइसोवॉल्यूमिक कॉन्ट्रैक्शन और आइसोवॉल्यूमिक रिलैक्सेशन, इन दोनों में चेंज हुआ है। दैट मींस देयर इज नो ट्रू आइसोवॉल्यूमिक कॉन्ट्रैक्शन, देयर इज नो ट्रू आइसोवॉल्यूमिक रिलैक्सेशन, ऐसा इसमें बताया गया है। एंड दैट हैपन्स इन द कंडीशंस ऑफ रिगर्जिटेशन। तो सबसे पहले हमेशा ये कंडीशंस देखा करो, ये लाइन, आइसोवॉल्यूमिक कॉन्ट्रैक्शन, आइसोवॉल्यूमिक रिलैक्सेशन, और वो चेंज हुआ है, इसका मतलब दिस इज डेफिनेटली अ कंडीशन ऑफ रिगर्जिटेशन। तो उसके बाद आसान हो जाता है क्योंकि रिगर्जिटेशन वाली एक ही कंडीशन दी है, एओर्टिक रिगर्जिटेशन। सो द आंसर वाज एओर्टिक रिगर्जिटेशन। जस्ट एक्सप्लेन करता हूं, हाँ ये क्यों है एओर्टिक रिगर्जिटेशन। सुनो, थिंक अबाउट आइसोवॉल्यूमिक कॉन्ट्रैक्शन, ये लाइन है आइसोवॉल्यूमिक कॉन्ट्रैक्शन। उसके बाद दिस इज इजेक्शन, फेज 3 इज इजेक्शन एंड फेज 4 इज आइसोवॉल्यूमिक रिलैक्सेशन। फिर ये फेज 1 पीरियड ऑफ फिलिंग। अब देखो इसमें आइसोवॉल्यूमिक कॉन्ट्रैक्शन, देयर शुड बी अ स्ट्रेट लाइन, वॉल्यूम एंड डायस्टोलिक वॉल्यूम जो है वो उतना का उतना रहना चाहिए, 130 ml का 130 ml। नाउ थिंक अबाउट आइसोवॉल्यूमिक कॉन्ट्रैक्शन। हाँ धनुष जी ने लिख दिया ऑलरेडी जो मैंने कहा, नो ट्रू आइसोवॉल्यूमिक कॉन्ट्रैक्शन, नो ट्रू आइसोवॉल्यूमिक रिलैक्सेशन। सो यू नो द कंडीशन ऑलरेडी, लेकिन जस्ट लेट मी एक्सप्लेन क्विकली कि ये देखो आइसोवॉल्यूमिक कॉन्ट्रैक्शन क्या होता है उसमें, AV valves are closed, यानी mitral valve is closed, but aortic valve is yet to open. Aortic valve अभी ओपन नहीं हुई है। क्यों? क्योंकि लेफ्ट वेंट्रिकल का प्रेशर बढ़ तो रहा है, कॉन्ट्रैक्शन है ना, तो सिस्टोल है, मतलब प्रेशर तो बढ़ रहा है, लेकिन अभी एओर्टा के प्रेशर से ज्यादा नहीं हो सका है, इसलिए एओर्टिक वाल्व अभी ओपन नहीं हो सकी है। सो माइट्रल वाल्व बंद हो गई, लेकिन एओर्टिक वाल्व अभी ओपन नहीं हो सकी है, दोनों वाल्व्स बंद, एंड लेफ्ट वेंट्रिकल इज कॉन्ट्रैक्टिंग लाइक अ क्लोज्ड चैम्बर, दैट्स आइसोवॉल्यूमिक कॉन्ट्रैक्शन। लेकिन यहां क्या हो रहा है देखो, हाँ अच्छा, एओर्टिक वाल्व क्यों ओपन नहीं हो सकी अब तक? बिकॉज लेफ्ट वेंट्रिकल का प्रेशर अभी भी एओर्टा के प्रेशर से ज्यादा नहीं हुआ है। लेफ्ट वेंट्रिकल का प्रेशर अभी भी एओर्टा के प्रेशर से ज्यादा नहीं हुआ है। इसका मतलब एओर्टा का प्रेशर अभी भी लेफ्ट वेंट्रिकल के प्रेशर से ज्यादा है। आइसोवॉल्यूमिक कॉन्ट्रैक्शन मतलब अब अगर एओर्टिक वाल्व रिगर्जिटेंट है तो क्या होगा? एओर्टा का प्रेशर is still more than the left ventricular pressure and aortic valve regurgitant. So what will happen? Blood will go from aorta back into the left ventricle during isovolumic contraction. मैं अक्सर मेरे लेक्चर में बताता हूं ये बात कि इसको महसूस करो, इसको समझने की कोशिश करो। लेफ्ट वेंट्रिकल इज कॉन्ट्रैक्टिंग, isovolumic contraction, and left ventricle is contracting and yet receiving the blood, regurgitant blood back from the aorta, है ना? तो ये अप्रिशिएट करो। अरे कॉन्ट्रैक्शन करती है मतलब ब्लड इजेक्ट आउट होना चाहिए ना, वो करेगी वो, लेकिन उसके पहले क्योंकि एओर्टा का प्रेशर लेफ्ट वेंट्रिकल के प्रेशर से ज्यादा है तो ब्लड रिगर्जिटेट्स बैक, मतलब कि लेफ्ट वेंट्रिकल का प्रेशर वॉल्यूम, लेफ्ट वेंट्रिकल का वॉल्यूम बढ़ता है ड्यूरिंग आइसोवॉल्यूमिक कॉन्ट्रैक्शन, सो कॉल्ड आइसोवॉल्यूमिक कॉन्ट्रैक्शन। तो ये लेफ्ट वेंट्रिकल का वॉल्यूम बढ़ते हुए हम देख रहे हैं यहां पर, द लाइन इज गोइंग लाइक दैट, एओर्टिक रिगर्जिटेशन। ओके, गॉट इट, थैंक यू। स्मूथ स्पेशलिस्ट जी कह रहे हैं गॉट इट। एक और बात पूछता हूं फिर आगे बढ़ते हैं। ओके, व्हाट डू यू थिंक विल हैपन इन पेरिकार्डियल एफ्यूजन? कॉन्स्ट्रिक्टिव पेरिकार्डाइटिस और पेरिकार्डियल एफ्यूजन, बताता हूं। See, here, constrictive pericarditis या pericardial effusion, these are the conditions of negative lusitropy. These are the conditions of negative lusitropy. I hope you have heard this word lusitropy. ल्यूसिट्रॉपी वर्ड सुना है आपने? जैसे आइनोट्रोपी होता है, हाँ, वन लाइन गो अप, ऐसा अर्श जी ने लिखा है, करेक्ट। फेज 1 जो आप कह रहे हो अर्श जी, बिल्कुल सही है। अर्श जी आपने कहा आप फर्स्ट ईयर के स्टूडेंट हो, लेकिन आप INI-CET के लिए रेडी हो ऐसा लग रहा है, वेरी गुड। हाँ, तो मैं कह रहा था कि आइनोट्रोपी मतलब कॉन्ट्रैक्टिलिटी एंड ल्यूसिट्रोपी मतलब रिलैक्सेबिलिटी एंड डायस्टोलिक फंक्शन। ल्यूसिट्रोपी मतलब रिलैक्सेबिलिटी एंड डायस्टोलिक फंक्शन ऑफ द वेंट्रिकल। पॉइंट नंबर 1, पेरिकार्डियल एफ्यूजन या कॉन्स्ट्रिक्टिव पेरिकार्डाइटिस, इसमें क्या होता है? देयर इज निगेटिव ल्यूसिट्रोपी। तो समथिंग लाइक दिस, जस्ट थोड़ा लाइन टेढ़ा हो गया लेकिन समझ लो आप। ये देखो, ये फिलिंग है, फिर आइसोवॉल्यूमिक कॉन्ट्रैक्शन, उसके बाद इजेक्शन, देन आइसोवॉल्यूमिक रिलैक्सेशन। अब निगेटिव ल्यूसिट्रोपी कैसे दिखती है? ये जो पॉइंट है ना, ये अगर यहां शिफ्ट होता है, दैट इज निगेटिव ल्यूसिट्रोपी। मतलब द ग्राफ विल गो लाइक दिस, ऐसे ऐसे जाके ऐसे। अगर ऐसा हुआ है ग्राफ में शिफ्ट, दैट विल बी निगेटिव ल्यूसिट्रोपी। क्यों? दो चीजें नोट करो। अच्छा, अर्श जी कह रहे हैं कि आपके लेक्चर से देखे, थैंक यू। दो चीजें नोट करो, व्हाई दिस इज कॉन्स्ट्रिक्टिव पेरिकार्डाइटिस या पेरिकार्डियल एफ्यूजन, क्यों है? लेफ्ट वेंट्रिकल इज अनेबल टू रिलैक्स, लेफ्ट वेंट्रिकल इज अनेबल टू एक्सपांड प्रॉपरली, और उस वजह से क्या होता है ड्यूरिंग डायस्टोल एंड फिलिंग, ये देखो एंड डायस्टोलिक वॉल्यूम जो नॉर्मली यहां होना चाहिए था 130 तक, वो अब यहां है, यानी 110 ml तक। इसका मतलब फिलिंग कॉम्प्रोमाइज हो रहा है। क्यों फिलिंग कॉम्प्रोमाइज हो रहा है? क्योंकि कॉन्स्ट्रिक्टिव पेरिकार्डाइटिस है, वेंट्रिकल इज अनेबल टू एक्सपांड, एंड इट इज अनेबल टू एक्सपांड, तो इट इज अनेबल टू अकोमोडेट द ब्लड ऑल्सो ड्यूरिंग फिलिंग फेज। तो आप देख सकते हो ये पॉइंट जो 130 ml तक था, दिस इज वॉल्यूम ऑन द हॉरिजॉन्टल एक्सिस, जो 130 ml पे था वो यहां आ गया है 110 ml। और दूसरी बात आपने नोट की, दूसरी बात नोट की, ये लाइन, दिस लाइन इज फिलिंग, लेफ्ट वेंट्रिकुलर फिलिंग, आपको पता है दिस इज फेज 1, फेज ऑफ फिलिंग, और ये स्ट्रेट लाइन है, मतलब वॉल्यूम इंक्रीज होता है, लेकिन वर्टिकल एक्सिस पे जो प्रेशर है वो इंक्रीज नहीं होता नॉर्मली। स्ट्रेट लाइन का मतलब क्या? प्रेशर डज नॉट इंक्रीज, वर्टिकल एक्सिस पे प्रेशर है ना, तो स्ट्रेट लाइन मतलब प्रेशर डज नॉट इंक्रीज नॉर्मली। लेकिन कॉन्स्ट्रिक्टिव पेरिकार्डाइटिस, जैसे जैसे ब्लड वेंट्रिकल में भरेगा, वेंट्रिकल इज अनेबल टू एक्सपांड प्रॉपरली, तो ब्लड भरेगा तो लेफ्ट वेंट्रिकल का प्रेशर बढ़ने लगेगा। ये प्रेशर बढ़ने लगेगा, यहां पर दिखाया है, लाइन स्ट्रेट होती है नॉर्मली, अब ये यहां आ गई है। सो दिस विल बी द कॉन्स्ट्रिक्टिव पेरिकार्डाइटिस, राइट? कुछ डाउट रहेगा तो आप जरूर मुझे पूछ सकते हो, बट आई थिंक कि आप लोगों ने ये किया है ऑलरेडी, सो यू आर ऑलरेडी अवेयर। लेट्स गो टू द नेक्स्ट। चलो देखते हैं। ये मेरे ख्याल से ना आपको थोड़ा टफ लगेगा, क्योंकि एस शेप्ड कर्व्स है, एस शेप कर्व है। सब लोग हो ना, सुन रहे हो ना मेरी बात? डाउट जरूर पूछो। एस शेप कर्व है और क्वेश्चन ये है, अ बीपी रेगुलेटिंग मैकेनिज्म इज शोन बिलो। तो सबसे पहले तो ये ग्राफ देख लो, एस शेप ग्राफ है। विच ऑफ द फॉलोइंग वुड बी रेस्पॉन्सिबल फॉर द शिफ्ट ऑफ द कर्व? तो आपको दो चीजें करनी है, एक तो आपको ये कर्व आइडेंटिफाई करनी है किस चीज की कर्व है, और उसके बाद वो शिफ्ट भी हो रही है, तो ये कब होगा ये आपको आइडेंटिफाई करना है। ओके, अर्श जी कह रहे हैं हार्ट फेल्योर स्मूथ स्पेशलिस्ट। आप लोग ऐसी आईडीज लेते हो ना कि नेम कॉल आउट करते वक्त, कोई बात नहीं चलेगा। चेंज ऑफ पोस्चर फ्रॉम सुपाइन टू स्टैंडिंग। ओके, और कोई कुछ कहना चाहता है? अर्श जी ने हार्ट फेलियर कहा। अर्श जी, आप शायद ना फ्रैंक स्टार्लिंग कर्व की बात सोच रहे हो दिमाग में। फ्रैंक स्टार्लिंग कर्व में ऐसा होता है, शिफ्ट ऑफ द पॉइंट, वो स्ट्रोक वॉल्यूम शिफ्ट होता है राइटवर्ड एंड डाउनवर्ड। ये वो कर्व नहीं है। तो चलो मैं एक्सप्लेन कर देता हूं, सुनो। और कोई कुछ कहना चाहता है? और कोई आंसर देना चाहता है? ये दोनों आंसर सही नहीं थे। सो एनीबडी? अभी आ गया सी आंसर। धनुष जी ने आंसर सी दिया है और स्मूथ स्पेशलिस्ट जी ने भी सी लिखा है। यस, इट इज सी। क्यों? एक्सप्लेन करता हूं फटाफट। देखो, सबसे पहले तो ये आर्टीरियल प्रेशर, हॉरिजॉन्टल एक्सिस पे आर्टीरियल प्रेशर है, वर्टिकल एक्सिस पे इंपल्स ट्रैफिक इन द नाइन्थ नर्व, इंपल्स ट्रैफिक इन द नाइन्थ नर्व। नाइन्थ एंड 10th नर्व दे स्टार्ट फ्रॉम द बैरोरिसेप्टर्स, और प्रेशर दिया हुआ है, सो मोस्ट प्रोबेबली दिस इज बैरोरिसेप्टर मैकेनिज्म का ग्राफ। पहली बात। अब देखो, एस शेप होती है। क्यों एस शेप होती है ये कर्व एस शेप क्यों है? बिकॉज एस शेप का मतलब है अराउंड 100 इट इज स्टीप. आप ये बात नोट करो, अराउंड 100 mm of Hg द कर्व इज वेरी स्टीप. स्टीप कर्व का मतलब क्या? स्टीप कर्व का मतलब है द सिस्टम इज वेरी सेंसिटिव अराउंड दैट ब्लड प्रेशर. अरे सीधी बात है, 100 mm of Hg के आसपास ये कर्व बहुत रैपिडली राइज या फॉल हो रही है. इसका मतलब 100 से बीपी जरा चेंज हो गया तो इंपल्स ट्रैफिक इन द नाइन्थ नर्व इज चेंजिंग ड्रामेटिकली. तो वो स्टीप पोर्शन यहां पे है, देख सकते हो आप. अभी आप देखो शिफ्ट हुआ है, द कर्व हैज बीन शिफ्टेड. इसको कहते हैं रीसेटिंग ऑफ द बैरो रिसेप्टर्स. दिस इज कॉल्ड रीसेटिंग ऑफ द बैरो रिसेप्टर्स. तो बैरो रिसेप्टर्स रीसेट हो गए हैं और अब जो फायरिंग वो 100 के आसपास मैक्सिमम करते थे, वही फायरिंग अब वो 120 के आसपास मैक्सिमम कर रहे हैं. इसका मतलब वो 120 mm ऑफ Hg को सेट हो गए, रीसेट हो गए. तो ये कब हो सकता है? हाँ, अर्श जी ने आगे लिखा है बैरो रिसेप्टर्स के बारे में, और गौरव सिंह जी ने लिखा है आर्टीरियल प्रेशर इज इंक्रीजिंग. करेक्ट. कॉर्टिसोल इंक्रीजिंग द MAP. लुक, सब लोग जो अटेंड कर रहे हैं उनके आंसर से ऐसा लग रहा है कि MAP इंक्रीज हुआ है, ऐसा दिखाया गया है. वो तो है, मीन आर्टिरियल प्रेशर इंक्रीज होता हुआ दिखाया है, लेकिन पॉइंट इज नॉट अबाउट मीन आर्टिरियल प्रेशर. पॉइंट इज अबाउट बैरो रिसेप्टर फायरिंग एट मीन आर्टिरियल प्रेशर. तो जो 100 को मैक्सिमम फायरिंग होता था बैरो रिसेप्टर्स का, वही अब शिफ्ट होके 120 को हो रहा है. दैट्स द पॉइंट. और ये क्यों होता है? सो जस्ट याद रखो कि बैरो रिसेप्टर रीसेटिंग ऑकर्स इफ द बॉडी इज कंसिस्टेंटली फेसिंग स्ट्रेस या कोई भी सिचुएशन जिससे बीपी चेंज हो और 2 दिन तक 48 घंटे तक वो बीपी चेंज रहे, तब जाके बैरो रिसेप्टर्स आर रीसेट टू द न्यू ब्लड प्रेशर. और वही यहां पे होता हुआ दिखाई दिया है. तो इज दैट क्लियर? आई होप इट इज क्लियर. इसमें इतना ही बताने लायक है कि बैरो रिसेप्टर सिस्टम, उसका अगर ग्राफ निकालो तो एस शेप है, एंड रीसेटिंग ऑफ द बैरो रिसेप्टर सिस्टम ऑकर्स आफ्टर 48 आवर्स. जो यहां पे दिखाया है कि पॉइंट हैज बीन रीसेट टू अ न्यू वैल्यू. 2 दिन तक ब्लड प्रेशर बढ़ा हुआ रहा, मीन आर्टीरियल प्रेशर 100 की जगह 120 रहा. तो अब बैरो रिसेप्टर्स का मैक्सिमम फायरिंग एट 120 mm ऑफ Hg. और ये कब होगा? ओके, क्लियर लिखा है सबने. गुड गुड गुड. ऐसे लिखो तो मुझे भी समझ में आ जाए. गुड गुड, थैंक यू. आगे बढ़ते हैं. नेक्स्ट. हाँ, ये तो धमाल है भाई. इसका आंसर आप देखो, दे सकते हो क्या? मैं थोड़ा फोन यहां पे रख के इसको थोड़ा एनलार्ज करने की कोशिश. एनलार्ज नहीं हो रहा है, ठीक है. मयूर पाटिल जी पूछ रहे कि इज दिस रिवर्सिबल? यस, इट इज रिवर्सिबल. देखो, 2 दिन तक अगर आप कंसिस्टेंटली एक हाई बीपी वाली सिचुएशन को फेस करते रहे, तो 2 दिन के बाद बैरो रिसेप्टर्स रीसेट होते हैं टू दैट न्यू वैल्यू. उनको लगता है कि अभी ये नॉर्मल वैल्यू है, और वो मतलब उस वैल्यू पे लेके जाने की कोशिश करते हैं. लेकिन अगर सेटल डाउन हो गए वापस बाद में, 48 घंटे तक बीपी एकदम नॉर्मल रहा, तो बैरो रिसेप्टर्स आर रीसेट. ओके. अगर वेसल वॉल उसकी कंडीशन ठीक है, कोई एथेरोस्क्लेरोसिस नहीं, बैरो रिसेप्टर सेंसिटिविटी अल्टर्ड नहीं, तो वापस से वो रीसेट होते हैं, रिवर्सिबल है. चलो ठीक है, यहां पे आते हैं. अभी यहां पे 2 कर्व्स दिखाई है, ये भी मेरा फेवरेट क्वेश्चन है. एक फ्लो वॉल्यूम लूप. फ्लो वॉल्यूम लूप इज शोन हियर. कैन यू रीड द फ्लो वॉल्यूम लूप? आई ट्राई टू जस्ट, वो इतना एक्सपांड नहीं हो रहा. ओके, आई रीड इट. मैं आपको रीड करके बताता हूं. ये पॉइंट रेसिडुअल वॉल्यूम ये 1000 ml है. फिर यहां पे 2000, 3000, 4000 ऐसा दिया हुआ है. टाइडल ब्रीदिंग दिखाया इन द रेड. ओके, टाइडल वॉल्यूम 500 ml है. टाइडल एक्सपीरेशन एंडेड एट 2000 ml. मतलब जब टाइडल एक्सपीरेशन हुआ ये रेड, तब आगे 2000 ml वॉल्यूम लंग्स में है. थोड़ा सा छोटा है ना डायग्राम, कोई बात नहीं, मैं एक्सप्लेन कर देता हूं, मतलब बोल के बताता हूं. रेड में दिखाया है नॉर्मल ब्रीदिंग, क्वाइट ब्रीदिंग. तो क्वाइट इंस्पिरेशन नीचे की तरफ, क्वाइट एक्सपीरेशन ऊपर की तरफ. और क्वाइट एक्सपीरेशन जहां पे खत्म हुआ वहां पे लिखा है 2000 ml. एट द एंड ऑफ क्वाइट एक्सपीरेशन, गाइस सुनो, एट द एंड ऑफ क्वाइट एक्सपीरेशन द लंग्स आर एट विच वॉल्यूम और कैपेसिटी? लिखो. एट द एंड ऑफ क्वाइट एक्सपीरेशन द लंग्स आर एट, बताओ. जब आप क्वाइट एक्सपीरेशन करते हो तो लंग्स किस वॉल्यूम या कैपेसिटी पे होते हैं? कोई लिख नहीं रहा है. ओके, मैं बता देता हूं. एट द एंड ऑफ क्वाइट एक्सपीरेशन, हाँ अभी लिख दिया, धनुष गौड़ा जी ने लिख दिया, FRC करेक्ट. FRC करेक्ट. एट द एंड ऑफ क्वाइट एक्सपीरेशन द लंग्स आर एट FRC. बराबर. ये मैं क्यों कह रहा हूं? क्योंकि देखो पूछा क्या गया है, कैलकुलेट द स्पेसिफिक कॉम्प्लायंस ऑफ द लंग. तो सबसे पहले करेक्ट, रॉकी सिंह जी ने भी लिखा है FRC, दैट्स राइट. सो एट द एंड ऑफ क्वाइट एक्सपीरेशन द लंग्स आर एट FRC. और यहां पे दिया हुआ है 2000. इसका मतलब पहला डेटा हमारे पास जो है वो है कि FRC बाय दिस डायग्राम इज 2000 ml. ठीक है. दूसरा पॉइंट, कैलकुलेट द स्पेसिफिक कॉम्प्लायंस. कैन एनीवन ऑफ यू डिफाइन और टेल मी व्हाट इज स्पेसिफिक कॉम्प्लायंस? नहीं, धनुष गौड़ा जी ने कॉम्प्लायंस लिखा है, उसके पास अभी आना बाकी है, मुझे कॉम्प्लायंस निकालना है. Specific compliance is compliance upon FRC. लिखो और ध्यान में भी रखो, अभी लिखने को टाइम नहीं है, क्योंकि आप अगर INI-CET को अपियर करने वाले हो तो अभी एग्जाम आ ही गई है. तो compliance upon FRC that is called as, हाँ मयूर पाटिल जी करेक्ट, compliance is चेंज इन द वॉल्यूम पर यूनिट चेंज ऑफ प्रेशर. ये है कॉम्प्लायंस. क्वेश्चन है कैलकुलेट द स्पेसिफिक कॉम्प्लायंस. स्पेसिफिक कॉम्प्लायंस, स्पेसिफिक कॉम्प्लायंस इज कॉम्प्लायंस अपॉन FRC. तो FRC की वैल्यू आ गई 2000. अभी ये डायग्राम देखो, ये डायग्राम आपको मैं पढ़ के बताता हूं. प्रेशर इज गोइंग फ्रॉम -5 to -8 सेंटीमीटर्स ऑफ वाटर and lung volume distension is by 600 ml. लंग्स का 600 ml का डिस्टेंशन हुआ है. कितने प्रेशर चेंज के लिए? 3 सेंटीमीटर ऑफ वाटर के प्रेशर चेंज के लिए. तो क्या आप बता सकते हो हाउ मच इज द कॉम्प्लायंस ऑफ दिस लंग? 600 ml डिस्टेंशन फॉर द प्रेशर चेंज फ्रॉम -5 to -8, मतलब 3 सेंटीमीटर ऑफ वाटर का प्रेशर चेंज. तो अब बताओ कॉम्प्लायंस कितना है लंग्स का? इस लंग का कॉम्प्लायंस कितना है? 600 ml का डिस्टेंशन वर्टिकल एक्सिस पे दिखाया, और हॉरिजॉन्टल एक्सिस पे प्रेशर फ्रॉम -5 to -8, मतलब कितना चेंज हुआ प्रेशर, सिक्स 600 डिवाइडेड बाय 3 मतलब 200 ml, 200 ml पर सेंटीमीटर ऑफ वाटर, ये कॉम्प्लायंस है। 200 ml पर सेंटीमीटर ऑफ वाटर डिवाइडेड बाय 2000, FRC इज 2000। हाँ करेक्ट, अभी मैं थोड़ा लैग की वजह से ना मुझे आपके आंसर थोड़े लेट दिखे। करेक्ट, इस लंग का कॉम्प्लायंस है 200 ml पर सेंटीमीटर ऑफ वाटर और FRC है 2000, तो 200 डिवाइडेड बाय 2000, ये था इसका आंसर, स्पेसिफिक कॉम्प्लायंस। आप बस इतना याद रखो कि आपको ये ऐसा ग्राफ देके पूछा जा सकता है कॉम्प्लायंस, मैंने उसमें थोड़ा सा और एडिशन कर दिया था, स्पेसिफिक कॉम्प्लायंस। तो स्पेसिफिक कॉम्प्लायंस इज इक्वल टू कॉम्प्लायंस अपॉन FRC, याद रखो इसको। आगे बढ़ते हैं, नेक्स्ट, चलो नेक्स्ट यहां पे आइडेंटिफाई द स्ट्रक्चर मेंशन्ड इन द डायग्राम बिलो। स्पाइनल कॉर्ड दिखाया है और एक पेन का एफरेंट पेरीफेरी से आके डॉर्सल हॉर्न पे जाके एंड हुआ है, फर्स्ट ऑर्डर न्यूरॉन, और यहां ऊपर से नीचे आने वाला एक पाथवे दिखाया है। इस पाथवे के 3 कंपोनेंट्स है। हाँ रॉकी सिंह जी, आंसर एक पहले का, हाँ ठीक है। नहीं, अभी आप इस क्वेश्चन पे हो ना, तो इस क्वेश्चन का, क्योंकि आप ABCD लिख रहे हो तो प्रीवियस क्वेश्चन का है कि इस क्वेश्चन का, वो मुझे समझ में नहीं, मतलब टाइम लगेगा उसको। ये देखो, पेन का फाइबर अंदर आया है और यहां एक डिसेंडिंग पाथवे दिखाया है। उस डिसेंडिंग पाथवे में एक यहां पे पेन इनिबिटरी कॉम्प्लेक्स ऑफ द डॉर्सल हॉर्न, वो कॉम्प्लेक्स है, और न्यूक्लियस रैफे मैग्नस। तो फिर ये स्ट्रक्चर क्या है? एनीबडी? अच्छा यहां पे देखो ना, ऑप्शंस दिए हुए हैं, इज इट लोकस सेरुलियस और पेरी एक्विडक्टल ग्रे, या रेड न्यूक्लियस, या वेस्टिबुलर न्यूक्लियस? हाँ मुझे लगा प्रीवियस क्वेश्चन का था ना वो, रॉकी सिंह जी, दैट्स राइट। अभी यहां पे आ जाओ, ये स्ट्रक्चर बताओ क्या है। न्यूक्लियस रैफे मैग्नस and pain inhibitory complex on the dorsal horn of the spinal cord। तो ये तीसरी स्ट्रक्चर क्या होगी? सबसे पहले तो आपने ये नोट किया ना कि दिस इज डिसेंडिंग एनाल्जेसिया पाथवे, एंडोजिनस एनाल्जेसिया सिस्टम, डिसेंडिंग एनाल्जेसिया सिस्टम है। तो उसके 2 कंपोनेंट्स तो नजर आ रहे हैं, पेन इनहिबिटरी कॉम्प्लेक्स ऑफ द डॉर्सल हॉर्न एंड न्यूक्लियस रैफे मैग्नस। व्हाट इज दिस? आइडेंटिफाई द स्ट्रक्चर। इसका आंसर रॉकी सिंह जी कह रहे हैं सी, नहीं, रेड न्यूक्लियस नहीं है, ये एनाल्जेसिया पाथवे का पार्ट है। धनुष गौड़ा जी कह रहे हैं ए। धनुष जी, लोकस सेरुलियस ऑल्सो इज ए पार्ट ऑफ एनाल्जेसिया सिस्टम, एंडोजिनस एनाल्जेसिया सिस्टम, लेकिन दिस इज पेरी एक्विडक्टल ग्रे। एक्विडक्ट ऑफ सिल्वियस है ना, उसके अराउंड, इसलिए पेरी एक्विडक्टल ग्रे, और इसलिए इसका आंसर था बी। तो याद रखो, एंडोजिनस एनाल्जेसिया सिस्टम, पेरी एक्विडक्टल ग्रे, न्यूक्लियस रैफे मैग्नस एंड पेन इनहिबिटरी कॉम्प्लेक्स। ये एनाल्जेसिया करता है, दबाता है पेन को। चलो आगे बढ़े, और आगे जाना है अपने को। हाँ ये अगला क्वेश्चन देखो, एक मिनट, आई जस्ट सी द चार्ज हियर, चार्जिंग, हाँ गुड, चार्जिंग है अभी भी। यहां पे एक पाथवे दिखाया है, विजुअल पाथवे sends one collateral to this structure and this structure is sending the signals to the cerebellum, and then cerebellum is sending the output signal to the spinal cord, और ये स्ट्रक्चर भी सेंडिंग सिग्नल डाउन ऑन द स्पाइनल कॉर्ड, और हैंड मसल्स लिखा है यहां पर, हैंड मसल्स. थोड़ा मैं बिग करने की कोशिश करता हूं, लेट्स सी. वो थोड़ा एनलार्ज नहीं हो पा रहा है, ठीक है, कोई बात नहीं, प्रोजेक्ट किया है इसके लिए. तो ये स्ट्रक्चर कौन सी है? इज इट प्रीटेक्टल न्यूक्लियस, इज इट एडिंजर वेस्टफाल न्यूक्लियस, इज इट सुपीरियर कॉलिक्यूलस, आप पढ़ सकते हो, या सुप्रा कायस्मेटिक न्यूक्लियस, क्या है ये? सबसे पहले बता दूं कि ये आई हैंड कोऑर्डिनेशन का सर्किट है. पॉइंट नंबर 1, आई हैंड कोऑर्डिनेशन. तो आई हैंड कोऑर्डिनेशन मतलब आई मतलब विजन, विजन का एक कोलैटरल भेजा जाता है एक स्ट्रक्चर को, और फिर वहां से वो सिग्नल जाता है सेरेबेलम को, एंड देन सेरेबेलम विल कोऑर्डिनेट द हैंड मसल्स. मैंने अभी रिसेंटली एक रील भी बनाया है कि क्या 15 साल का बच्चा जसप्रीत बुमराह को 6 मार सकता है? तो कैसे मार सकता है, या कैसे वो ऐसा इतना अच्छा खेल सकता है? आई हैंड कोऑर्डिनेशन अच्छा है. सो क्या लगता है आपको ये स्ट्रक्चर कौन सा है? मैं बताता हूं. दिस इज सुपीरियर कॉलिक्यूलस. सुपीरियर कॉलिक्यूलस, याद रखो, विजन के का एक सिग्नल टेक्टम को जाता है, टेक्टम को. टेक्टम में होता है सुपीरियर एंड इंफीरियर कॉलिक्यूलाई, तो वो ये टेक्टम का पार्ट है, सुपीरियर कॉलिक्यूलस, और फिर उसका सिग्नल जा रहा है सेरेबेलम में, एंड देन सेरेबेलम विल कोऑर्डिनेट द हैंड मसल्स. तो ये आई हैंड कोऑर्डिनेशन का सर्किट है, और कौन सी स्ट्रक्चर इन्वॉल्व है? सुपीरियर कॉलिक्यूलस. सो सुपीरियर कॉलिक्यूलस इज द स्ट्रक्चर सेंट्रल टू द आई हैंड कोऑर्डिनेशन. आप में से कुछ लोगों ने प्रीटेक्टल न्यूक्लियस लिखा है. प्रीटेक्टल न्यूक्लियस इज इन्वॉल्व इन द pupillary light reflex. Pretectal nucleus is involved in pupillary light reflex, यानी visual pathway to pretectal nucleus, Edinger-Westphal nucleus and constriction of the pupil. तो वो अपना visual क्या बोल रहा था? It is involved in the pupillary light reflex. जबकि इसका सिग्नल सेरेबेलम में गया है और नीचे हैंड मसल्स लिखा है, मतलब ये कोऑर्डिनेशन ऑफ द मूवमेंट के लिए है. और उसका कौन सा है पार्ट? सुपीरियर कॉलिक्यूलस. अच्छा, सुप्रा कायस्मेटिक न्यूक्लियस क्या करता है बताओ? मैं जानता हूं इसका आंसर आप सबको आता होगा. विजुअल पाथवे से एक सिग्नल निकलता है सुप्रा कायस्मेटिक न्यूक्लियस को ऑफ हाइपोथैलेमस. व्हाट इज द रोल ऑफ सुप्रा कायस्मेटिक न्यूक्लियस? चलो बताओ, आई एम श्योर आपको आता है सबको, बताओ. एनीबडी? तब तक मैं आगे के एमसीक्यू की तरफ बढ़ता हूं. हाँ, सर्कैडियन रिदम, करेक्ट, सर्कैडियन रिदम, दैट्स राइट. चलो आगे बढ़ते हैं. आइडेंटिफाई द स्ट्रक्चर शोन एज क्वेश्चन मार्क इन द इमेज. ये स्ट्रक्चर पूछी गई है, ये क्वेश्चन मार्क है. दिस इज द स्ट्रक्चर, दिस वन. तो सबसे पहले तो आप ये देखो कि सामने है सार्कोमेयर. कैसे पता चला? एक तो स्ट्रक्चर ऐसी है, फिर यहां पे सार्कोलेमा, सार्कोग्लाइकेंस लिखा है, मतलब दिस इज सार्कोमेयर, थिन एंड थिक फिलामेंट्स. और ये जो स्ट्रक्चर है थिन फिलामेंट से निकल के सार्कोलेमा तक जा रही है. तो इस स्ट्रक्चर का नाम क्या है? हाँ, मयूर पाटिल जी ने लिखा पैरावेंट्रिकुलरिस फॉर ऑक्सीटोसिन, करेक्ट, दैट्स राइट. तो बताओ, थिन फिलामेंट से निकल के सार्कोलेमा तक जाने वाली स्ट्रक्चर का नाम क्या? हिंट दूं आपको, हिंट देता हूं, लार्जेस्ट ह्यूमन जीन कॉम्प्लेक्स या लार्जेस्ट ह्यूमन जीन encodes for this structure. तो क्या है ये स्ट्रक्चर? धनुष जी ने कहा डी, नहीं, मायोमेसिन जो है ना, बताता हूं, एक मिनट। देखो यहां पे सेंटर में एक डार्क लाइन होती है, यहां पर मायोसिन फिलामेंट के अंदर वो जो सेंट्रल डार्क लाइन है उसमें होता है मायोमेसिन। यहां पर ये इट्स कॉल्ड एज एम लाइन, उसमें होता है मायोमेसिन। अच्छा, धनुष गौड़ा जी ने बी लिखा, बी, हाँ करेक्ट, डिस्ट्रोफिन, करेक्ट धनुष गौड़ा जी, दैट्स राइट। लेकिन एक बार लिखते तो भी मैं देख लेता, मतलब इतना एम्फेटिकली नहीं लिखते तो भी चल जाता, कोई बात नहीं। दिस इज डिस्ट्रोफिन, इट कनेक्ट्स थिन फिलामेंट टू द सार्कोलेमा। डिस्ट्रोफिन का काम क्या है? डिस्ट्रोफिन का काम है जो थिन और थिक फिलामेंट का इंटरेक्शन से जो टेंशन जनरेट होता है ना मसल में, उस टेंशन को मसल के मेम्ब्रेन तक पहुंचाना और मेम्ब्रेन से बाहर की तरफ पहुंचाना, ये डिस्ट्रोफिन का काम है। तो ये थिन फिलामेंट टू सार्कोलेमा, दिस इज डिस्ट्रोफिन। इट इज कनेक्टेड विद द डिस्ट्रोग्लाइकैन, और डिस्ट्रोग्लाइकैन के आगे यहां पर एक प्रोटीन है जिसका नाम है लैमिनिन। तो लैमिनिन वहां होता है, मायोमेसिन यहां पर होता है एम लाइन में, और फाइनली टाइटिन। टाइटिन कहां पे होता है यू नो ना, टाइटिन कनेक्ट्स जेड लाइन टू द थिक फिलामेंट लाइक दिस, दिस इज टाइटिन, ठीक है। बट इसका आंसर था डिस्ट्रोफिन। आगे बढ़ते हैं। ये लो, रोहित एंथनी जी ने मेरी तारीफ कर दी, शुक्रिया। मैं वैसे ही जरा डाउन फील कर रहा था, तो आपने तारीफ किया तो अच्छा लगा, थैंक यू। आगे बढ़ते हैं। हाँ समझ में आ गया रोहित एंथनी जी, अभी थैंक यू फॉर मेकिंग मी फील गुड, आई एम फीलिंग गुड आपने कहा तो। लेकिन अब यहां पे कॉन्सेंट्रेट करते हैं। ईईजी का एक पार्ट यहां पर दिखाया है, तो ये कौन सा पार्ट है, ये कब रिकॉर्ड होता है, ये पूछा है, ठीक है। तो बताओ ये क्या है, क्वाइट वेकफुलनेस या अलर्ट वेकफुलनेस या डीपर प्लेन ऑफ एनेस्थीसिया या स्टेज 2 ऑफ द एनआरईएम स्लीप। हाँ, धनुष गौड़ा जी ने बिल्कुल सही दिया जवाब, दिस इज डी, स्टेज 2 ऑफ द एनआरईएम स्लीप। क्यों बताता हूं। सी, क्वाइट वेकफुलनेस में अल्फा वेव्स होती है, अलर्ट वेकफुलनेस विथ आईज ओपन बीटा वेव्स, डीपर प्लेन्स ऑफ एनेस्थीसिया डेल्टा वेव्स, और ये जो दिखाया है ये स्टेज 2 ऑफ द एनआरईएम स्लीप है। लार्ज के कॉम्प्लेक्सेस, लार्ज के कॉम्प्लेक्सेस, अरे सॉरी ये है लार्ज के कॉम्प्लेक्स, और ये जो है दिस इज कॉल्ड एज स्लीप स्पिंडल, स्लीप स्पिंडल। तो प्लीज नोट डाउन, स्लीप स्पिंडल फॉलोड बाय लार्ज के कॉम्प्लेक्स। ये स्पिंडल शेप ऐसा आता है और उसके बाद लार्ज के कॉम्प्लेक्स। सो दिस इज टिपिकल ऑफ स्टेज 2 ऑफ द एनआरईएम स्लीप। और आईएनआईसीटी में और नीट पीजी में ये क्वेश्चन कई बार आ चुका है। ये कम से कम 4-5 बार आ चुका है पिछले 10-12 साल में, मतलब एवरी अल्टरनेट ईयर आता है, तो कर लेना। ईईजी इज अ कॉमन टॉपिक। चलो आगे बढ़ते हैं फटाफट। नेक्स्ट, आइडेंटिफाई द मिसिंग स्ट्रक्चर इन द सर्किट। एक सर्किट दिखाया है, आइडेंटिफाई द मिसिंग स्ट्रक्चर। और सर्किट क्या है देखो, सिंगुलेट गायरस, सिंगुलेट गायरस connected with a structure in the brain that is connected with mammillary body और फिर mammillothalamic tract। तो ये स्ट्रक्चर कौन सी होगी? अरे वाह, धनुष गौड़ा जी ने आंसर दे भी दिया। यस, हिप्पोकैम्पस, दैट्स राइट। ये पापेज सर्किट है, दिस इज पापेज सर्किट। जेम्स पापेज नाम का एक न्यूरो एनाटॉमिस्ट था, उसने इमोशनल बेसिस ऑफ आवर बिहेवियर ये बताने के लिए एक सर्किट बताया, तो वो पापेज सर्किट। इसमें क्या होता है, प्री फ्रंटल कॉर्टेक्स कनेक्टेड विद द सिंगुलेट गायरस, सिंगुलेट गायरस टू हिप्पोकैम्पस, जो अपना आंसर है एमसीक्यू का, हिप्पोकैम्पस। हिप्पोकैम्पस टू मैमिलरी बॉडी यानी कि हाइपोथैलेमस, एंड मैमिलरी बॉडी टू थैलामस, मैमिलोथैलामिक ट्रैक्ट, एंड देन बैक टू द सिंगुलेट गायरस। तो ये है पापेज सर्किट। करेक्ट करेक्ट, बाय फॉर्निक्स हिप्पोकैम्पस इज कनेक्टेड टू मैमिलरी बॉडी। बिल्कुल बिल्कुल, मयूर पाटिल जी बिल्कुल सही है। लगता है आप लोग INI-CET क्रैक करने ही वाले हो। दैट्स फैंटास्टिक, नाइस टॉकिंग टू यू। मतलब अच्छा लगता है कि आप सब लोग का नॉलेज एकदम ऐसा भरपूर है, नॉलेज से भरपूर हो। वेरी गुड। चलो नेक्स्ट, आइडेंटिफाई द सिक्रीशन। आइडेंटिफाई द सिक्रीशन। अभी इसमें क्या दिया है, ब्लड वेसल से एक प्राइमरी सिक्रीशन आया। ये प्राइमरी सिक्रीशन इज आइसोटोनिक, यानी वो सोडियम, क्लोराइड और वाटर ये सब है उसमें। और फिर ये डक्ट से फ्लो कर रहा है, तो सोडियम इज रीएब्सॉर्ब्ड एंड पोटेशियम इज सिक्रीटेड, क्लोराइड इज रीएब्सॉर्ब्ड एंड बाइकार्बोनेट इज सिक्रीटेड, ऐसा दिखाया है। सो व्हाट इज दिस सिक्रीशन? सेकेंडरी सिक्रीशन इज, मतलब फाइनल सिक्रीशन जो है वो हाइपोटोनिक है। प्राइमरी सिक्रीशन वाज आइसोटोनिक, फाइनल सिक्रीशन इज हाइपोटोनिक। तो ये कौन सा सिक्रीशन है? अरे सब लोग पैंक्रियाटिक जूस बोल रहे हैं। धनुष गौड़ा जी आपने पैंक्रियाटिक जूस लिखा है, सी नहीं, एक बार और कोशिश करो। हाँ, सलाइवा, दिस इज सलाइवा, करेक्ट, सलाइवा है। रिमेंबर, सलाइवा में एक प्राइमरी सिक्रीशन होता है जो आइसोटोनिक होता है, आइसोटोनिक टू प्लाज्मा, और उसके बाद व्हेन इट स्टार्ट्स फ्लोइंग थ्रू द डक्ट, सोडियम इज रीएब्सॉर्ब्ड but the duct is relatively impermeable to water. सोडियम निकल जाता है लेकिन डक्ट इज impermeable to water, वाटर रह जाता है पीछे, एंड इसलिए फाइनल सलाइवा विल बी ऑलवेज हाइपोटोनिक। सोडियम निकल गया, वाटर रह गया, मतलब हाइपोटोनिसिटी। सो दिस इज सलाइवा, करेक्ट। आगे बढ़े। ये देखो, अभी कंप्लीट द फॉर्मूले और इक्वेशंस गिवन बिलो। तो पहला, टू मेजर द एफिशिएंसी ऑफ अ होम्योस्टैटिक रेगुलेटरी मैकेनिज्म, गेन, गेन इज इक्वल टू करेक्शन अपॉन, करो कंप्लीट। होम्योस्टैटिक रेगुलेटरी मैकेनिज्म जो होती है तो उसका मेजर होता है गेन। हायर द गेन, मोर एफिशिएंट इज दैट होम्योस्टैटिक रेगुलेटरी मैकेनिज्म। तो गेन करेक्शन अपॉन व्हाट, करेक्शन अपॉन रेसिडुअल एरर, बराबर है मयूर पाटिल जी। धनुष गौड़ा जी ने लिखा है रीनल का इंफिनिटी होता है, बराबर। किडनी बॉडी फ्लूइड्स मैकेनिज्म इन रेगुलेशन ऑफ ब्लड प्रेशर, तो उसका गेन इनफाइनाइट है क्योंकि इसमें एरर 0 रहती है, ऐसा देखा गया है। दैट्स राइट। चलो एक ऐसे ही सवाल फेंकता हूं, ऑप्शंस नहीं दूंगा, ऐसे ही पूछता हूं। कौन सी ऐसी सिस्टम है जो फीडबैक फीड फॉरवर्ड टाइप की सिस्टम है? टेल मी वन सिस्टम इन द बॉडी विच इज नेगेटिव फीडबैक फीड फॉरवर्ड टाइप ऑफ सिस्टम। और जब तक आप सोचते हो, जस्ट 1 मिनट का ब्रेक लेते हैं, एक सिप ऑफ वाटर लेके वापस आता हूं। नॉर्मली हम ब्रेक नहीं लेते, बट गर्मी इतनी है ना कि एसी 18 पे चल रहा है तो भी ऐसा लग रहा है कि अरे बाप रे गर्म हो रहा है, गर्म हो रहा है। सो आप सोचो, आंसर दो, तब तक आई विल कम बैक। हाँ भाई लोग आ जाओ। अरे बाप रे, इसका आंसर भी दिया, हाइपोथैलेमस थर्मोरेगुलेटरी सिस्टम। फैंटास्टिक। मुझे लगता है जो ये लोग आके बैठे हैं, अटेंड कर रहे हैं, वो सुपर प्रिपेयर्ड है INI-CET के लिए। अच्छा पढ़ाई हुआ है आपका। मयूर पाटिल जी ने कहा हार्ट रेट इंक्रीजेस व्हेन वी थिंक ऑफ एक्सरसाइज। ये फीड फॉरवर्ड हो गया। फीडबैक, टेम्परेचर रेगुलेशन आप याद रख सकते हो, टेम्परेचर रेगुलेशन, दैट्स राइट। देखो मैं बताता हूं, एक्सप्लेन करता हूं। हमारे 2 टेम्परेचर्स होते हैं, एक शेल टेम्परेचर, एक कोर टेम्परेचर। शेल टेम्परेचर मतलब ये स्किन का, सरफेस का टेम्परेचर, और कोर मतलब अंदर बॉडी टिश्यूज का टेम्परेचर। तो सोचो अभी कि शेल टेम्परेचर कैन चेंज विद द सराउंडिंग्स। यानी आप ठंडी में गए, ठंडी में जाते ही शेल टेम्परेचर बिगिंस टू फॉल, स्किन टेम्परेचर कम होने लगता है, और फिर आगे क्या होता है? सिग्नल्स आर सेंट टू हाइपोथैलेमस एंड शिवरिंग इज इंड्यूस्ड। अब ये शिवरिंग क्यों हुई? टू मेंटेन द कोर टेम्परेचर एट अ स्टेडी लेवल। तो शेल टेम्परेचर का निगेटिव फीडबैक हो गया कि भाई उसका टेम्परेचर फॉल हो गया था, वो बढ़ गया, और कोर टेम्परेचर का ये फीड फॉरवर्ड हो गया कि भाई बाहर का टेम्परेचर कम हो रहा है, टिश्यूज का टेम्परेचर फॉल नहीं होना चाहिए, तो पहले ही कर दो शिवरिंग और मेंटेन करो। सो फीडबैक, फीड फॉरवर्ड इज टेम्परेचर रेगुलेशन। और ऐसी चीजें आपको नॉर्मली मैं ही बताता हूँ लेक्चर्स में, ऐसा मैं बोलने वाला था लेकिन उससे थोड़ा सा प्राउडी फील होता है ना, मैं ही बताता हूँ और कौन बताता है, ऐसा मैं नहीं करना चाह रहा था, बट आप अटेंड करोगे तो ऐसे एक्सक्लूसिव चीजें आपको मिलेगी। चलो आगे बढ़ते हैं। मेजरमेंट ऑफ वेरियस बॉडी फ्लूइड्स, बॉडी फ्लूइड वॉल्यूम्स। गाइज एक काम करता हूं, आप ये आंसर लिखो, तब तक मैं चार्जिंग को लगा देता हूं, वो चार्ज कम है करके बता रहा था, आप तब तक इसका आंसर दे दो। चार्जिंग चार्जिंग। हेलो हेलो, कैन यू सी मी? हेलो हेलो, क्या आप देख पा रहे हो? ओके। ओके चलो आगे बढ़ते हैं। तो बताओ, मेजरमेंट ऑफ वेरियस बॉडी फ्लूइड वॉल्यूम्स, इंडिकेटर डाइल्यूशन मेथड, V इज इक्वल टू I माइनस ए अपॉन क्या होता है? चलो मैं लिख देता हूं आंसर। V इज इक्वल टू I माइनस ए अपॉन C। V इज इक्वल टू I माइनस ए अपॉन C, इंडिकेटर डाइल्यूशन मेथड। मैंने ये इसके लिए लिया क्योंकि इस बार मैं एक्सपेक्ट कर रहा हूं कि बॉडी फ्लूइड कंपार्टमेंट में से एक या दो क्वेश्चन INI-CET में आने ही वाले हैं। ओके, तो जरूर देखना। कोई कुछ लिख नहीं रहा है। मयूर जी, धनुष जी, कम ऑन, बकअप, कीप इट अप, मतलब आ जाओ मैदान में। चलो आगे बढ़ते हैं। नेक्स्ट, हाँ आ जाओ। ये देखो, नर्व मसल में से रिलेशन बिटवीन नर्व डायमीटर एंड कंडक्शन वेलोसिटी, रिलेशन बिटवीन नर्व डायमीटर एंड कंडक्शन वेलोसिटी। सो माइलिनेटेड नर्व्स में क्या रिलेशन है और अनमाइलिनेटेड नर्व में क्या रिलेशन है, बताओ। चलो मैं लिख देता हूं। इन द केस ऑफ माइलिनेटेड नर्व्स, कंडक्शन वेलोसिटी इज 6 times द डायमीटर। 6 times, मतलब जैसे ए अल्फा फाइबर, ए अल्फा, ए बीटा, ए गामा, ए डेल्टा, वही तो क्लासिफिकेशन ऑफ नर्व फाइबर्स। ए अल्फा, उसका डायामीटर है 20 माइक्रॉन्स, तो कंडक्शन वेलोसिटी 120 meters per second, 6 times the diameter। और इन द केस ऑफ अनमाइलिनेटेड नर्व्स, मतलब टाइप सी फाइबर जो अनमाइलिनेटेड होता है, उसमें वेलोसिटी इज प्रोपोर्शनल टू द स्क्वायर रूट ऑफ द डायमीटर। इस बार एक क्वेश्चन क्लासिफिकेशन ऑफ नर्व फाइबर्स में से आने वाला है, तो इसलिए अच्छे से प्रिपेयर कर लेना इसको। ये लाइव चल रहा है ना, मतलब तुम लोग हो ना, सुन रहे हो ना, सुनो सुनो, मतलब सुनो, रिप्लाई दो, आंसर दो। आगे बढ़ते हैं। कैलकुलेशन ऑफ टिटेनाइजिंग फ्रीक्वेंसी ऑफ ए मसल। ओके, गुड गुड, सुनो सुनो। कैलकुलेशन ऑफ टिटेनाइजिंग फ्रीक्वेंसी, number of stimuli per second for a skeletal muscle. You know the formula? आपको एक किस्सा सुनाता हूं। चलो, बहुत टाइम से हम एकेडमिक कर रहे हैं, और ये किस्सा मतलब इसका एविडेंस है, आप मेरे चैनल पे जाके देख सकते हो। ये एमसीक्यू स्केलेटल मसल के ग्राफ के साथ मैंने डिस्कस किया मेरे चैनल पे यूट्यूब के। 15 दिन के बाद INI-CET थी। जैसे ही पेपर हुआ तो कुछ बच्चों ने वो वीडियो के नीचे कमेंट्स डाले, सर दिस एमसीक्यू केम एज इट इज, जो आपने बोला था वैसा का वैसा इन द INI-CET। अभी भी वो आप देख सकते हो यूट्यूब चैनल पे जाके, कमेंट्स भी रीड कर सकते हो। और ऐसा होता है कि टीचर ने बोला ये आएगा और वो आ गया, लेकिन ये बहुत ही एक्सक्लूसिव टाइप एमसीक्यू था। मतलब पहले कभी पूछा नहीं गया, डायरेक्ट ग्राफ देके पूछा गया था। चलो, हाँ, तो टिटेनाइजिंग, व्हाट इज टिटेनस? सस्टेन्ड स्टेट ऑफ कॉन्ट्रैक्शन ऑफ द मसल, फेलियर टू रिलैक्स, इट हैपन्स ड्यू टू हाई फ्रीक्वेंसी स्टिमुलेशन। तो कैसे कैलकुलेट करते हैं कि कितनी फ्रीक्वेंसी में वो टिटेनस में चला जाएगा? टिटेनाइजिंग फ्रीक्वेंसी इज कैलकुलेटेड एज रेसिप्रोकल ऑफ the contraction period expressed in seconds, 1 upon contraction period, reciprocal of the contraction period which is expressed in seconds, नंबर ऑफ स्टिमुलाई पर सेकेंड। अगर कैलकुलेट को करना है तो इस तरीके से कैलकुलेट करना है टिटेनाइजिंग फ्रीक्वेंसी। ओके, चलो आगे बढ़ते हैं। नेक्स्ट, ब्लड एमसीवी का फॉर्मूला बताओ, एमसीवी का फॉर्मूला बताओ। आई थिंक इस बार आईएनआईसीटी में एक क्वेश्चन ब्लड इंडाइसेस से रिलेटेड आएगा, ब्लड इंडाइसेस से रिलेटेड, मतलब आपको ब्लड इंडाइसेस दिए होंगे और कुछ कैलकुलेट करने के लिए बोला जाएगा, ऐसा एक क्वेश्चन होने वाला है। देयरफॉर आई टुक दिस। तो बताओ, मीन कॉर्पस्कुलर वॉल्यूम इज इक्वल टू पीसीवी अपॉन व्हाट? बताओ, पीसीवी अपॉन व्हाट? धनुष जी और मयूर जी, आप दोनों ही अभी आंसर कर रहे हो। तो बताओ, ए एच बी अपॉन पीसीवी इंटू 100? नहीं 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 नहीं, देखो मैं बताता हूं, सुनो। आपने एक ब्लड सैंपल लिया, उस ब्लड सैंपल का पहले आरबीसी काउंट किया और फिर उसका पैक्ड सेल वॉल्यूम किया, तो इतने आरबीसी का इतना पैक्ड सेल वॉल्यूम, इतना वॉल्यूम, तो एक आरबीसी का कितना ऑन एन एवरेज? दैट्स कॉल्ड एज मीन कॉर्पस्कुलर वॉल्यूम। इतने आरबीसी का मिला के इतना वॉल्यूम, तो एक आरबीसी का कितना? तो फॉर्मूला विल बी पीसीवी अपॉन, कम ऑन, फॉर्मूला विल बी पीसीवी अपॉन आरबीसी काउंट, पीसीवी अपॉन आरबीसी काउंट। और आपने जो लिखा है ना धनुष जी, वो है एमसीएच का फॉर्मूला। एक्चुअली एमसीएच का नहीं, एमसीएच का फॉर्मूला ये होगा एच बी अपॉन आरबीसी। अरे सिंपल बात है ना, मीन कॉर्पस्कुलर हीमोग्लोबिन एमसीएच कैसे कैलकुलेट करोगे? एक सैंपल लिया, उसका आरबीसी काउंट किया और फिर उसका हीमोग्लोबिन किया, आरबीसी काउंट और हीमोग्लोबिन किया, इतनी आरबीसी में मिला के इतना हीमोग्लोबिन, तो एक आरबीसी में कितना? दैट्स कॉल्ड एज एमसीएच, हीमोग्लोबिन अपॉन आरबीसी काउंट। हाँ करेक्ट। नेक्स्ट, सीवीएस। ये फॉर्मूला तो अभी इसके ऊपर कुछ आएगा कि नहीं, आई डोंट नो, आई एम नॉट श्योर, बस डाल दिया ऐसे ही। तो बताओ, एच आर मैक्स इज इक्वल टू फिजियोलॉजिकली अचीवेबल हार्ट रेट मैक्सिमम इज इक्वल टू 220 माइनस, बताओ एनीबडी, जो लोग भी सुन रहे हैं, 220 माइनस, कम ऑन। मैं लिख देता हूं, हार्ट रेट मैक्सिमम इज इक्वल टू 220 माइनस एज इन ईयर्स, दैट इज हार्ट रेट मैक्सिमम फिजियोलॉजिकली। हाँ अरे लिख दिया है आपने, धनुष जी ने और मयूर पाटिल जी ने, दोनों ने लिखा है। वेरी गुड, वेरी गुड, फैंटास्टिक। ओके, नेक्स्ट, नेक्स्ट क्वेश्चन, नेक्स्ट फॉर्मूला, कार्डियक इंडेक्स। ये बताने की जरूरत है क्या? बताओ, कार्डियक इंडेक्स इज इक्वल टू कार्डियक आउटपुट अपॉन, बताओ, ये बहुत सिंपल है। ये हार्ट रेट मैक्सिमम और ये वाला, इसके ऊपर आई एम नॉट श्योर कि इस बार आईएनआईसीटी में आएगा कि नहीं, लेकिन वो कॉन्ट्रैक्शन पीरियड वाला वो जरूर आएगा इस बार, एलवीपीवी लूप वाला जरूर आएगा, डायरोनेट डायग्राम एंड बॉडी फ्लूइड कंपार्टमेंट्स आएगा, देख लेना अगले संडे। हाँ, कार्डियक आउटपुट पर मीटर स्क्वायर ऑफ बॉडी सरफेस एरिया, दैट इज कार्डियक इंडेक्स, कार्डियक आउटपुट पर मीटर स्क्वायर ऑफ बॉडी सरफेस एरिया। किधर गया? लिख देता हूं यहां पर, कार्डियक इंडेक्स रे, मतलब कार्डियक आउटपुट 5 लीटर पर मिनट है, लेकिन ये कितने बॉडी सरफेस एरिया के लिए है? दैट इज कॉल्ड एज कार्डियक इंडेक्स, बॉडी सरफेस एरिया expressed in meter square. So cardiac output will be liters per minute per meter square of body surface area. All right, next. हाँ, अभी ये बताओ, करेक्ट 1.7 मीटर स्क्वायर होता है बॉडी सरफेस एरिया, आपने लिख दिया, वेरी गुड। आगे बढ़ते हैं, मेजरमेंट ऑफ कार्डियक आउटपुट बाय फिक्स इक्वेशन, फिक्स मेथड और फिक्स इक्वेशन याद करो और बताओ। कार्डियक आउटपुट इज इक्वल टू ऑक्सीजन कंजम्पशन अपॉन क्या है डिनोमिनेटर, बाय फिक्स मेथड। और यहां पर मैंने और एक मेथड ली है, डाई डाइल्यूशन मेथड, क्योंकि मेरा स्ट्रॉन्ग गट फीलिंग है कि इस बार डाई डाइल्यूशन मेथड या इंडिकेटर डाइल्यूशन मेथड के ऊपर एक एमसीक्यू आने वाला है, इसलिए मैंने इंडिकेटर डाइल्यूशन पहले एमसीक्यू में भी लिया था और अभी भी। सो बताओ, बाय फिक्स इक्वेशन कार्डियक आउटपुट इज कैलकुलेटेड लाइक दिस, ऑक्सीजन कंजम्पशन अपॉन, कोई लिखा है क्या? अरे धनुष जी ने लिख दिया है, फैंटास्टिक, यस धनुष जी, दैट्स राइट, अपॉन आर्टीरियो वीनस ऑक्सीजन डिफरेंस। तो अभी इतना लिख दिया है, तो ये भी बता दो कि वीनस सैंपल इज कलेक्टेड फ्रॉम वेयर? धनुष जी और मयूर जी, आप दोनों ही आंसर कर रहे हो अभी, तो मैं जस्ट पूछ रहा हूं, वीनस सैंपल फॉर दिस मेथड इज कलेक्टेड फ्रॉम विच ब्लड वेसल? क्या बता सकते हो? कोई कुछ कहना चाहता है? धनुष जी, मयूर पाटिल जी? नहीं, आईवीसी से नहीं है। मतलब आईवीसी नहीं नहीं नहीं, नॉट आईवीसी। वीनस सैंपल इज कलेक्टेड फ्रॉम पल्मोनरी आर्टरी, याद रखना इस बात को। वीनस सैंपल, देखो हमको रिप्रेजेंटेटिव सैंपल चाहिए, इट शुड रिप्रेजेंट द एंटायर बॉडी। अभी आर्टरी तो आप कहीं से भी ले सकते हो, क्योंकि सभी आर्टरीज में टिश्यूज तक जाने तक सेम ऑक्सीजन कॉन्सेंट्रेशन होता है, तो आर्टरी कोई भी ले सकते हो सैंपल के लिए। वेन कोई भी नहीं ले सकते, क्योंकि एक पर्टिकुलर बॉडी से पार्ट से आने वाली वेन, वो उस पार्ट ने कितना ऑक्सीजन कंज्यूम किया उसके ऊपर डिपेंड करता है कि रिटर्न जो वेन आ रही है वहां से उसमें कितना ऑक्सीजन रहेगा। लेकिन सारी वेन्स जब इकट्ठा आती है राइट साइड ऑफ द हार्ट में, तो ये सब वीनस ब्लड मिक्स हो जाता है और पल्मोनरी आर्टरी में जाता है, तो वहां से सैंपल लेते हैं। इट इज कॉल्ड एज मिक्स्ड वीनस ब्लड, रिप्रेजेंटेटिव सैंपल फॉर द एंटायर बॉडी। अच्छा, ये देखो डाई डाइल्यूशन मेथड, ये बताओ, F is equal to I upon, come on, complete it. Dye dilution method for measurement of cardiac output. F is equal to I upon, धनुष जी, मयूर पाटिल जी, मयूर पाटिल जी आप हो, मतलब अटेंड कर रहे हो ना? बताओ, by dye dilution method for the measurement of cardiac output, I upon, चलो मैं कंप्लीट कर देता हूं, I upon C into T into 60। हाँ, लिख दिया है, यस यस, करेक्ट करेक्ट करेक्ट। चलो आगे बढ़ते हैं। नेक्स्ट, चलो फटाफट कर लेते हैं। हाँ मयूर पाटिल जी, अगला क्वेश्चन देखो, मीन आर्टेरियल प्रेशर इज इक्वल टू डायस्टोलिक बीपी प्लस, आप बताओ, तब तक मैं भी लिख देता हूं। ये ऐसा आना चाहिए, ऐसा। डायस्टोलिक बीपी प्लस 1/3 ऑफ pulse pressure. 1/3 of pulse pressure. That's mean arterial pressure. करेक्ट करेक्ट, आपने लिख दिया आंसर, वेरी गुड। आगे चलते हैं, फटाफट देख लेते हैं नेक्स्ट। रेस्पिरेटरी सिस्टम, अल्वियोलर वेंटिलेशन इज इक्वल टू टाइडल वॉल्यूम माइनस ब्रैकेट है इनटू रेस्पिरेटरी रेट। ओके, धनुष जी ने आगे लिख दिया है 95 mm ऑफ एच जी। यस, मयूर पाटिल जी ने भी लिखा है, करेक्ट करेक्ट। इसका बताओ अभी। अल्वियोलर वेंटिलेशन इज इक्वल टू टाइडल वॉल्यूम माइनस समथिंग इनटू रेस्पिरेटरी रेट। मैंने ना एमसीक्यू लेने की जगह पे जानबूझ के ऐसा लिया, क्योंकि थोड़ा सा आपका थॉट प्रोसेस को प्रोवोक किया जाए विदाउट लुकिंग एट द ऑप्शंस। और मयूर पाटिल जी और धनुष जी ने दोनों ने करेक्ट आंसर दे भी दिया, मेरा ध्यान ही नहीं था। करेक्ट, टाइडल वॉल्यूम माइनस द डेड स्पेस वॉल्यूम इनटू रेस्पिरेटरी रेट। यानी एक ब्रेथ में इतना वॉल्यूम आपने इंस्पायर किया, उसमें से इतना डेड स्पेस में रहा और बचा हुआ अल्वियोलाई में पहुंचा, वो एक ब्रेथ में। और ऐसे कितने ब्रेथ्स है पर मिनट, सो दैट विल बी अल्वियोलर वेंटिलेशन पर मिनट। यस, आगे चलते हैं, नेक्स्ट नेक्स्ट नेक्स्ट। हाँ, ये बताओ आप। करेक्ट करेक्ट। अरे मोल श्री सिंह जी भी आ गए, वॉल्यूम ऑफ डेड स्पेस। वेलकम, वेलकम बैक। एक्चुअली आप थे, वेलकम बैक। अभी ये बताओ तो मैं मानू नहीं, वैसे मान चुका हूं, क्योंकि आप सही आंसर पहले क्वेश्चन से दे रहे हो, तो वो तो मान ही रहा हूं। इतनी गर्मी है ना, आपको क्या बोलूं, 18 पे एसी चल रहा है और तब भी गर्म हो रहा है। चलो ठीक है। अल्वियोलर एयर इक्वेशन, इट टेल्स अस अबाउट दी अल्वियोलर PO2। तो PaO2 is equal to FiO2 into P barometric minus PH2O माइनस। अरे बाप रे, क्या बात है। जेवियर जी ने लिखा है, धनुष गौड़ा जी ने लिखा है, फैंटास्टिक फैंटास्टिक। आप लोग कौन हो यार, INI-CET क्रैक करके तो नहीं बैठे? मतलब ये मैं सोच रहा था कि शायद नहीं आएगा आपको, लेकिन आपने दिया है। अच्छा, मोलश्री सिंह पूछ रहे कि सर आप कहां से पढ़ा रहे हो? बिहार झारखंड में इतनी गर्मी नहीं है। नहीं, यहाँ पर है। मैं आपको बताऊं, सुबह दोपहर को मैं जस्ट गया था, ये ठीक से नहीं चल रहा था तो बुलाने के लिए गया था। 42 के आसपास टेम्परेचर था, 42। खैर, ये इक्वेशन में पहले ब्रैकेट में ये है FiO2 इंटू P बैरोमेट्रिक माइनस PH2O, और इस ब्रैकेट में है PaCO2 अपॉन R। R is respiratory quotient. So this is alveolar air equation. हाँ, मैं बताना भूल गया, इस बार के INI-CET में इसके ऊपर क्वेश्चन आएगा। मार्क माय वर्ड्स, कैलकुलेट करने को आएगा। मार्क माय वर्ड्स। चलो आगे बढ़ते हैं फटाफट। नेक्स्ट। अच्छा, ये ऑलरेडी दिया है। अच्छा, मैं आंसर की को ढक के खड़ा होता हूं। मैच द फॉलोइंग आपको करना है, मतलब 1C 2B ऐसा करके आंसर बताओ मुझे। आंसर की को मैं कवर कर रहा हूं, आंसर की दी हुई एक्चुअली। आप देख पा रहे हो? टाइडल वॉल्यूम B, so 1B. इंस्पिरेटरी रिजर्व वॉल्यूम, अच्छा डेड स्पेस वॉल्यूम 5 का A बोला है, हाँ ठीक है, करेक्ट है. इंस्पिरेटरी रिजर्व वॉल्यूम 3000, रेसिडुअल वॉल्यूम 1200, और कौन सा बचा? हाँ, एक्सपिरेटरी रिजर्व वॉल्यूम 1100 ml. ठीक है, मतलब आपको आता ही है, नो टेंशन. ये तो आपने बाय हार्ट किया होगा ना, तो इसमें कोई प्रॉब्लम नहीं है. अगला देखते हैं. हाँ, अभी एक करके बताओ तो मानू आपको. 1 बाय 1 करते हैं ताकि आपको ऐसा पूरा एक साथ लिखने का जरूरत नहीं पड़े. 1 बाय 1 जाते हैं. बताओ, मीन आर्टीरियल प्रेशर. आई होप आप सब लोग अगले क्वेश्चन पे आ गए होंगे, क्योंकि मैं 1B 2C ये सब देख पा रहा हूं. मीन आर्टीरियल प्रेशर बताओ. 40 है, 120 है, 80 है, 6 है, 96 है, क्या है? हाँ, मयूर पाटिल जी ने कहा 1 is E. चलो, हो गया बराबर. मीन सिस्टेमिक फिलिंग प्रेशर, एनी आइडिया? मीन सिस्टेमिक फिलिंग प्रेशर. INI-CET में ही पहले 5-6 साल पहले क्वेश्चन आया था. व्हाट विल बी द प्रेशर इन द डेड? प्रेशर इन द डेड. एंड द ऑप्शंस वेर 0, 6 mm of Hg, 20 mm of Hg या 36 mm of Hg. तो आप तो सोचोगे 0. और मीन सिस्टेमिक फिलिंग प्रेशर, अरे वाह क्या बात है, 2D लिखा है सबने. फैंटास्टिक. यस, 2 is D, 6 mm of Hg. When the heart stops beating, ये पूछने की जगह पे बोल दिया प्रेशर इन द डेड. When the heart stops beating, हमारा जो सर्कुलेशन है ना, ब्लड, वो अलग अलग सेगमेंट्स में रीडिस्ट्रीब्यूट होता है और फिल कर देता है सारे सेगमेंट्स को. और फिर एक प्रेशर जो होता है उसको कहते हैं मीन सर्कुलेटरी फिलिंग प्रेशर, और वो 6 mm of Hg होता है. उसके बाद फिर सिस्टोलिक बीपी 120, डायस्टोलिक बीपी 80, पल्स प्रेशर इज 40. नेक्स्ट, आ जाओ भाई मैदान में. पेप्सिन, ट्रिप्सिन, अमाइनोपेप्टिडेज, टाइलिन और कोलिक एसिड, ये आपको मैच करना है. मीन सर्कुलेटरी फिलिंग प्रेशर इंक्रीजेस एज वीनस रिटर्न इंक्रीजेस. जेवियर जी ने कहा, बिल्कुल फैंटास्टिक. हाँ, पेप्सिन गैस्ट्रिक जूस, ट्रिप्सिन पैंक्रियाटिक जूस, अमाइनोपेप्टिडेज सक्कस एंटेरिकस, टाइलिन saliva. Tyline is an alpha amylase present in the saliva, and cholic acid in the bile. Yes, correct. धनुष जी ने सभी मार्क कर दिए, मयूर पाटिल जी ने भी किए, जेवियर जी ने भी 1D. करेक्ट करेक्ट, सही है. आगे चलते हैं फटाफट. नेक्स्ट. हाँ, ये बताओ तो मैं मानू. लेट्स गो 1 by 1. वेट, लेट्स गो 1 by 1. ओके, बताओ. ग्लूकोज एंट्री इनटू ऑल सेल्स. मैं एक एक बोलूंगा, आप जवाब देना. आ गए सब लोग? मैं देखूंगा अभी। अरे अरुण, अरुण RTH सेम, अरुण जी, आई कॉल अरुण जी, बिकॉज आप लोगों की आईडीज ऐसी होती है ना, एक ना एक, कि नाम कॉल आउट कैसे करे वही समझ में नहीं आता। मतलब अभी ये तो ठीक है, चलो आपका मतलब नॉर्मल नाम तो दिखाई दे रहे, धनुष, शैकाली और बाकी सब नाम दिखाई तो दे रहे कम से कम। अभी लास्ट टाइम मैं लेक्चर ले रहा था, एक की आईडी थी क्रेजी लवर और वो आंसर कर रहा था, तो अभी मैं उसको क्या कॉल आउट करूं? क्रेजी लवर थोड़ा अजीब लगता है ना। खैर, धनुष गौड़ा जी ने इमोजी भेजा है कुछ, चलो ठीक है कोई बात नहीं, यहां आ जाओ। विल गो वन बाय वन। अच्छा, आपने आंसर की रिफर किया, कोई बात नहीं, हरकत नहीं, चालेल। ग्लूकोज एंट्री इनटू ऑल सेल्स ऑकर्स बाय फैसिलिटेटेड डिफ्यूजन। सोडियम पोटेशियम पंप, प्राइमरी एक्टिव ट्रांसपोर्ट। Insulin secretion by pancreatic beta cell occurs by exocytosis. Oxygen going from alveolus to the blood, simple diffusion. And water movement from low solute to high solute concentration, osmosis. चलो आगे बढ़ते हैं। ये मैंने कुछ फैक्चुअल इंफॉर्मेशन निकाली थी बस आपको बताने के लिए कि इसके अराउंड MCQs आ सकते हैं। सो द मैन हु डिस्कवर्ड फाइबर्स इन द हार्ट एंड सेल्स इन द सेरेबेलम, पता है नाम आपको? परकिंजे सेल इन द सेरेबेलम, परकिंजे सेल इन द हार्ट। इफेक्ट ऑफ ऑक्सीजन ऑन CO2 लिबरेशन इन द लंग्स, कॉल्ड एज हाल्डेन्स इफेक्ट। इफेक्ट ऑफ CO2 ऑन ऑक्सीजन डिलीवरी, बोर इफेक्ट, और यह है हाल्डेन इफेक्ट। JG apparatus and Lasis cell is named after him। क्या आपको पता था वही नाम? GOORMAGHTIGH, और इसका प्रोनाउंसिएशन पता है आपको? गोमथी। तो प्लीज रिमेंबर JG apparatus, और उसमें जो एक सेल है लेसिस सेल, उसका दूसरा नाम है गोमथी सेल, GOORMAGHTIGH। Factor involved in the absorption of vitamin B12 is named after Castle, intrinsic factor of Castle। High frequency stimulation of skeletal muscle results in tetanus। टिटेनस के ऊपर क्वेश्चन आने वाला है तो मैंने फिर से ले लिया। प्रेशर रिलेटेड इंजुरी टू द नर्व ट्रंक इज कॉल्ड, बताओ, प्रेशर रिलेटेड इंजुरी टू द नर्व इज कॉल्ड, आंसर इज न्यूरोप्रैक्सिया, न्यूरोप्रैक्सिया। सलाइवरी अमाइलेज इज कॉल्ड टाइलिन। इस बार मुझे लगता है GIT में से अगर कोई क्वेश्चन आया तो सलाइवा के ऊपर आएगा, तो इसलिए मैंने सलाइवा बार बार लिया है। ओके, सिंड्रोम ऑफ postpartum pituitary necrosis is called Sheehan's syndrome, postpartum pituitary necrosis। J receptor in the lungs were discovered by Paintal, अवतार सिंह पेंटल, AS पेंटल। मैं पेंटल फैमिली को थोड़ा सा जानता हूं अच्छे तरीके से, चंडीगढ़ बेस्ड फैमिली रही है, और इनके ब्रदर की वाइफ IS पेंटल हमारे DY पाटिल में काफी साल तक प्रोफेसर एमिरेटस करके काम किया था। खैर याद रखो, J रिसेप्टर पेंटल, पिट्यूटरी एपोप्लेक्सी, जेवियर जी ने लिखा है, करेक्ट, यस। एंड द प्रोसेस डिस्क्राइब्ड एज सेल ड्रिंकिंग। सेल ड्रिंकिंग किसे कहते हैं? पिनोसाइटोसिस। तो सेल ईटिंग इज फैगोसाइटोसिस, सेल ड्रिंकिंग पिनोसाइटोसिस, सेल वॉमिटिंग एक्सोसाइटोसिस, सेल सुसाइड एपोप्टोसिस, सेल मर्डर, नेक्रोसिस। तो इस तरीके से ये है। ओ हो गया, ठीक है। तो थैंक यू फॉर listening to me very patiently. It's nice to interact with you. Thanks a lot. और आपको पता है आप कहीं भी मुझे रीच आउट कर सकते हो फॉर एनी डाउट रिलेटेड टू द फिजियोलॉजी। सभी जगह पे एक ही है अपना विवेक सर्स फिजियोलॉजी, यहाँ पे कनेक्ट कर सकते हो, या फिर और कहाँ पे, नहीं तो पीडब्ल्यू मेडेड के किसी प्लेटफॉर्म, मतलब टेलीग्राम हुआ तो अपना मेडेड का है अलग से चैनल, तो वहाँ पे आप कनेक्ट कर सकते हो। चलो, थैंक यू, बाय बाय।